Hello, everyone. Looks like we've got a lot of people joining online. We are really, really excited that you all have joined us today for the first annual International Space Ecology Workshop. Thank you so much for everyone all over the globe. As we're watching the chat box here, we have got representation from everywhere from Alaska through the US, the East Coast, the West Coast, through Europe. We've got some folks from Australia online and even some folks from the Philippines. So all over the globe, truly an international workshop today. And those of you who are in some different time zones and are up really, really late or very early, we are really appreciative of your devotion to this cause and to this field. So we're really glad to have you join us today. We will try to have some breaks in between our talks today so that everybody can take a moment to go get some coffee as needed. And also I am watching the chat box here. So if you can't hear me or have a question about something, go ahead and put that into the chat, and I'll try to keep an eye on that so that you can communicate with me. Looks like, I do need to mention before I go any further, we have mics turned off for the attendees for today because we have an extraordinarily large number of people registered, which was a pleasant surprise, but also makes things a little difficult logistically. So what we're going to do is actually just have folks be able to communicate with us through the chat. And if you have questions for the speakers, it goes into the Q&A box. So you should have Q&A. That's for presenting questions to the speakers that I will select and read them. And then the chat box for communicating with us. So we've had to unfortunately turn off mics just because there's going to be such a large number of people online to make this manageable. I'm going to be your MC for the day, your happy host. My name is Christine Escobar. I'm vice president and co-founder of a company called Space Lab Technologies. I have titled myself recently an eco-astronautical and systems engineer. So that's a new name I've made up. We'll see if it sticks. But basically my background is in ecology as well as in systems engineering. And now I'm studying bioastronautics at the University of Colorado. I'm a PhD student there. I basically got into this field, well, many, many years ago. I would say in the 90s. I was actually inspired myself by what you see behind me, Biosphere 2, by the idea that we can use life to support life in an uninhabitable environment like space, and sustainably live anywhere. The idea that we can use ecosystems to thrive in an uninhabitable environment inspired me long, long ago. And here I am today in a company called Space Lab, where we are developing technology and researching and developing technology for people to use to live and work in space sustainably so that we can stay there longer and go further. I'm principal investigator on several research projects like a microgravity lily pond. It's a hydroponic growth chamber for growing small plants, including little plants called duckweed. A lot of you know me as the duckweed girl that grows those aquatic plants. That's enough about me, but just a little bit about my background. And this workshop here today actually is something that I've always hoped would happen for a long time. So I think you all are probably feeling the same way, that we can get together and talk about this field. So, excuse me. I think that the fact that we are all here at these wide range of early and late hours across the globe today means that we really all share a common vision and common passion for this subject. And it's been, unfortunately, a really long time since a group of engineers and space biologists and ecologists all came together to discuss the ecological aspects of how we inhabit space sustainably. We believe, the organizers here today, that there is a need to promote and to organize our research on an international scale and hopefully reignite our interest, global interest in an ecological systems approach. given that there's so much momentum right now for deep space exploration, so much momentum and planning going on to perhaps move beyond Leo to the moon and Mars, we all share this mutual vision of artificial closed loop ecosystems. These ecosystems will exploit biological and ecological and electromechanical processes to support human life. Hopefully these systems can enable indefinite exploration of space and maybe sustainable living on Earth as well. So it's a really, really critical field to understand and to conquer. There's been a lot of needs assessments, right? I think many of us have seen different literature reviews and articles pointing out different gaps in knowledge for space ecology and closed ecological systems. But through all of those needs assessments, there's still all of these obstacles in knowledge and gaps in technology. And so all these obstacles remain in realizing this vision. And so we don't think that we can make any advances towards closing these gaps until we coordinate our efforts and hopefully socialize these concepts. So we really hope that this workshop today will provide you all and us with some inspiration and some momentum to that end, and that this workshop will be one of many. We'd like to hopefully see this as an annual event. So we've really, excuse me, we're really glad you've joined us for this adventure. What we want to accomplish today is to basically visit where have we been over the many decades of research in this field. Start to get to know who's who in the field right now, who are some active researchers, and start talking about what are the next steps for closing these knowledge and technology gaps. Maybe even draft up a space ecology, excuse me, Space Ecology Roadmap as a plan for future action of how we move forward and work together. Just a little bit about definitions. I love definitions. So I was thinking about what is this thing anyway? What is space ecology? Why are we having this workshop called Space Ecology Workshop? We might all have different ideas about what that means. So we have ecology, right? That's dealing with the relationships between groups of living things and their environment, and an ecosystem, a community of organisms and their environment functioning together. So what I think we would maybe define space ecology as is relationships of organisms to each other and to their environment in the space environment. Pretty simple definition there. I think today, hopefully, closed ecological systems for life support won't be a footnote or just a small session in a bigger conference or some bygone obscure field anymore. Today, we really want to immerse ourselves in this topic. We want to define what this field of study is and establish this unity of vision and a purpose. And I hope that we can accomplish that today. Here's our agenda. You guys all received a program, but just to review it again with you, what we're going to do today is have some 45-minute sessions with our different speakers throughout the day. There will be lots of opportunity for everyone to ask questions of the speakers. So they'll give their presentation. And then as you think of questions, please go ahead and put them into the Q&A box. And I'll be looking at those questions and then present your questions to the speakers for you. At 11:15 my time, you guys can translate, excuse me, at 11:15 a.m. Eastern time, correction. You guys can translate to whatever time zone that is for you. We'll also do a group brainstorming session, and I'll give you some more instructions when we get to that point about what to do and how that's going to work. But what we want to do during that brainstorming is start putting down all of these knowledge and technology gaps that we think still exist, and really we're trying to see, are there any that we are missing, that we haven't identified, and do we all agree that this is a complete picture of what some of the knowledge and research gaps are still to be overcome in this field. We're going to break for lunch after that first brainstorming session, excuse me. So this morning, first, we'll hear from Ray Wheeler, then Mr. Christophe Lasseur, and then William Dempster before doing the brainstorming. And then we'll break for a lunch break or whatever meal that is for you in your day. And then in the second half of our day, we're going to hear from Barbara Belvisi from Interstellar Lab and Cyrille Przybyla from the French National Institute for Ocean Science. Joel Cuello from the University of Arizona, and finally we'll hear from John Hogan from the NASA Ames Research Center. And then we're going to end the day with another brainstorming session where we're going to talk about where do we go now? What are next steps for the future? What actions can we take to close these knowledge and technology gaps? And that's it. That's our agenda for today. Once again, here's today's speakers. We are really grateful to all of you, our speakers, for taking the time to talk to us today and share with us your experience and knowledge and ideas for the future. And I think this was a really great collection of speakers that we've gathered for our first workshop here today. So again, we have Dr. Wheeler from NASA KSC, who's going to be starting us off here in a few minutes. Our organizers want to give a really big thank you to this great group of people that have come together to make this workshop happen today. We have Frida Tao, professor at the University of Washington. Patrick, you all know well, is the organizer and coordinator of all things technical and all of the emails you have been getting and all of the web communication. So Patrick has been doing an amazing job keeping us all glued together. We have Jane Shetzoff from from UCLA. Jane, sorry, I didn't put UCLA down there, and also is founder of a company called Propagule Ecotech. And we have Sherry Damlow, who's at the University of Florida. And Sherry, don't want to put you on the spot right now, but one of these days you're going to have to tell me what a space taphonomist does.
I study the interaction between the decomposing remains of organisms in the space environments.
Interesting. Great. Okay. I just realized that last night that I did not know. So just another little logistical note for everyone that is on the line. In your programs, you received a PDF of a program for the day. And let Patrick know if for some reason you don't know, you haven't seen that program, we'll get it to you. In there, we are including bios of attendees if you choose to submit one in what we're calling the who's who of space ecology. So you can look through your program and get an idea of who's who and who's doing what. And also there's a great references list provided by Frida Tao on closed ecological studies publications. And we hope that that reference list will be a resource that we can provide to this community in the future and keep it updated and keep adding to it. Okay, I just have a few tips for you for the day before I hand things over to Dr. Wheeler. Again, we have a staggering number of registrants today for the workshop. So unfortunately, we're going to have to keep microphones muted, but there are many ways for you guys to communicate with us and to communicate with the speakers. So if you have a question you want to ask one of the speakers, again, Q&A box, put that in the Q&A box, and then I will select questions, present them to the speakers for everyone to hear your question and the answer. If we don't get to all of your questions, we will actually make a really good, every effort we can to follow up with your questions post-workshop. Also, if you have comments and feedbacks, they're not questions, but just comments and you want to chat with each other, feel free to use the chat box at your leisure. And during those two group brainstorming sessions also, we're going to introduce you to a cool tool called Mural. And in Mural, you're going to be able to collectively write down your ideas on knowledge and technology gaps and actions we can take to overcome them. So this is kind of your turn to like go up to the board and write on the board. Also inside Mural, there's another chat box. You're free to use that as well. So we can have you writing feedback all over the place today. We want to get your ideas any way that we can. We're recording the workshop. audio recording, as well as we're recording all of the written comments for proceeding summary. So if you miss something, you're writing on the mural and you miss part of the discussion, you will get a link to the audio recording. You can go back and listen later. And finally, if you are having any trouble, let Patrick know is the best way. If you can message Patrick, he's our technical expert for the day. And if you're having any questions, please message him in Zoom. He's here to help you. So sit back and enjoy the discussion and have a wonderful experience today in our first workshop. So I'm going to take a few minutes before we introduce our first speaker. And for those of you who are just joining us, you can go ahead and get some coffee or whatever beverage you would like, or take a break if you need that, and we'll come back in about do we want to start right on the hour, Patrick, do you think? Yep. Okay, so we're going to start on the turn of the hour.
All right.
Hi, everyone. Just a reminder, one comment before we get started on the next talk, that we are doing an audio recording today, and we're also recording all of the written feedback. Just wanted to reiterate that one for everyone. We will be sending out a link to the audio recording, so you will all be able to access that.
Test, test.
We hear you, Bill. Hi there. Okay, so we're gonna go ahead and get started here in a moment. If I could gather everyone back. And our first speaker today, we would like to welcome Dr. Ray Wheeler. Ray is a plant physiologist and senior scientist at the NASA Kennedy Space Center. And he's in the NASA Exploration Research and Technology Directorate there at NASA Kennedy. And he's going to be talking with us today about some historical perspectives of closed ecological life support systems and bioregenerative research by NASA. So I'm going to hand it over to you, Ray, whenever you are ready.
Okay, thank you, Chris. Can you hear me okay? I can't see much when I'm in full screen mode.
We can hear you great, loud and clear.
Okay, thanks. First of all, thanks to the organizing committee for setting up this first and perhaps first of many annual space ecology workshops. This is really interesting and fun for me, and to see the worldwide participation is really exciting. As Chris mentioned, I'm a plant physiologist. I've been involved with plant research for many years, even in my graduate research and postdoctoral studies, and then working at Kennedy Space Center. And I've also become involved a bit with the general life support activities and research. As the title says, I'm going to give at least my historical perspective. It'll be biased toward plants. I'm a plant physiologist, and so I tend to think of the whole world revolving around plants, but clearly ecological systems involve many organisms that interact, as Chris showed in the definitions earlier. And I will focus largely on, as the title suggests, on NASA and US interests, but we're going to hear about European efforts, and I don't think, I'll mention a little bit, but there have been certainly activities all around the world. There were pioneering studies in Russia. The Japanese have had a number of very fascinating efforts over the years. The Chinese, China National Space Agency now is actively engaged. So there's worldwide interest, clearly. So just a little bit about human life support, and you've probably seen numbers like this. I mean, what we need for humans to stay alive on that left-hand graph are inputs. We need oxygen, food, and water. And, you know, it depends how you use your water. If you have hygiene and flushing and laundry and things like that, water budgets go way up. And so that you see a mass number there, and this varies depending on the human body mass and metabolic rates. And likewise, we have outputs CO2 from a respiration, metabolic solids, wastewater, things like that. So there are mass costs either way on this. So it becomes obvious that if you go into space, you need regenerative life support capabilities. You just can't stow and resupply these kinds of mass numbers. A lot of it's done today on the space station using physicochemical methods, but as many of the biologists and ecologists online know, you can use photosynthetic organisms like plants, and there are other photosynthetic organisms, and I'll mention those a little bit, to drive reactions to fix carbon dioxide at the bottom, you see there, and you need a lot of light energy to do that. And if you choose crops, you can get edible biomass. I show that as a single unit of carbohydrate. And then if it's aerobic photosynthesis, like most plants, you generate oxygen. So you're getting multiple life support functions. And of course, those then can cycle to the humans to provide food and oxygen and so forth. And then you have solid waste and liquid waste coming out of both of those streams. I don't show that in this diagram, but for materials closure, clearly those are issues that need to be considered too. Well, I want to go as far back, at least as I know, in terms of thinking about bioregenerative life support, and I'm going to refer to the studies of Joseph Priestley, an English scientist, theologian, philosopher. He was a very interesting fellow. This is in the late 1700s, and he did many studies, amongst which he did some studies in using bell jars, and he enclosed plants and either candles or animals in these bell jars. And I'll read a quote here from him. "I have been so happy as by accident to hit upon a method of restoring air, which has been injured by the burning of candles, and I have discovered at least one of the restoratives. It is vegetation. When I first put a sprig of mint into a glass jar standing inverted in a vessel of water, it had continued growing for some months, and I found that the air would neither extinguish a candle nor was it at all inconvenient to a mouse." So I think we could probably give Priestley the credit for conducting the first bioregenerative life support experiment. And this these concepts were used and built upon and even written about before this, but I show one example here from the US Navy, where they were considering the use of algal bioreactors for submarines to generate oxygen. This is in the 1950s. There's some well-known names here, at least to some of the plant community: Alan Brown, Melvin Calvin, Nobel Prize winner, Bob Kraus, Jack Myers, or Reynolds. Some of you might know these names, but these were individuals that were on the advisory panels for this naval document. And even in the 1960s, early 1960s, people were thinking about how to use photosynthetic organisms and ecological systems, in this case, algae. This is a diagram from a paper that Bob Kraus wrote in 1962. And it shows a very complex, what he called a recyclostat, to cultivate algal systems, harvest the oxygen, harvest the biomass, and then how you might even configure that for a lunar surface mission, the bottom part of that figure. This is 1962. And here we are, 60 years later, thinking about trying to do these same sorts of things as we go back into space. There were even space flight experiments. This picture shows is from a paper by Herb Ward and colleagues, where they conducted, as far as I know, the first space flight experiment for the US that involved higher plants and algae. They use chlorella algae and they use duckweed, Chris, Spirodella in their case, I think was the genus. And they measured the photosynthetic rates. The chlorella experiment didn't work well; it failed. But the the duckweed experiment did. And on that bottom right-hand graph, you can see, it's hard to read, but there's a time plot showing oxygen levels rising over time, and then CO2 levels, carbon dioxide levels, going down over time. And of course, things reverse during the dark periods, but you see a very classic photosynthetic gas exchange curve. And so Ward and his colleagues demonstrated it can be done in space. And this was with duckweed. So just some general observations from the studies in the 1950s and 60s. Again, I'm focusing on US studies primarily here. A lot focused on algae and some cyanobacteria. And one of the favored organisms was Chlorella pyrenoidosa. I hope I pronounced that correctly, but it was a thermal tolerant strain of chlorella. And at the time, it was touted as being the most photosynthetically efficient organism that we know of. And so you could push it very hard in controlled reactors, but there were other species tested. You see in that second line, some of those are algae, some of those are cyanobacteria, and we'll probably hear a little bit more about things like spirulina from Dr. Lasseur later. They worked a lot on development of culture systems. They use chemostats, turbidostats. These are continuous harvesting systems. And there's a lot of challenges with that in terms of algae and maintaining the nutrients and harvesting the biomass and different ways to do it. And they even got to the point, this is again in the 1960s, where they connected these culture systems with photosynthetic organisms to heterotrophic organisms like mice and monkeys. And there were even some short tests with humans, and I don't have the citation for this, but so this was all happening in the 1960s. And one thing that stood out from these studies, they were very interested in trying to balance things completely with through biological solutions and ecological solutions. And so they were interested in things that were referred to as assimilation quotients and respiration quotients. And so this is the CO2 produced divided by the oxygen consumed in the case of respiration, or the CO2 assimilated divided by, boy, I hope I have that right. Well, anyway, it's a ratio of CO2 and oxygen. And what they found, and this goes to some studies that the leader of a lot of these efforts at the time, Jack Myers at the University of Texas, that it was difficult to balance these major metabolic gases in closed systems if you only had biological solutions, at least in simple systems. You could do 95% of it, but things weren't quite perfectly balanced. And then I got this quote from a paper by Herb Ward and Miller below, and he said, there's a general misconception in the scientific community, this is from 1966, that the success of bioregenerative approach depends on the development of a biologically and chemically closed ecology with complete material balance. While this may be the ultimate goal, few consider it possible, if indeed necessary. So let me apply my translation to that statement. You can use biological and ecological approaches to get you 90 or 95% of the way there. There are some of these to balance everything very precisely in terms of materials flow, metabolic gases. You sometimes have things that don't quite balance, but these can be resolved very easily with chemical engineering solutions. And so this is not to say that bioregenerative life support systems aren't competitive. They are, and they are the way we'll need to go to be more autonomous and more Earth independent as we venture farther into space. But we'll always need engineering support for any of these systems. That's my translation to this. A few specific comments. So on algae, they were highly focused on algal studies at the time. And so the algae were definitely very photosynthetically efficient. I mean, they're just hard to beat in that regard. And they're very volume efficient. You can get a lot of algae into a small volume, relatively speaking. And they were energy efficient in terms of if you use electric lighting, for example, you could submerge the lights right in the reactor, so you can absorb all the light. This is a bigger challenge when you get to higher plants. Is some of the inefficiencies show up because you end up wasting some light. It shines on the floor or the walls, something like that. On the negative side, though, when they began to think about using algae more for just oxygen generation and CO2 removal and sort of getting multiple functions out of them in terms of food value, there were some processing challenges with algae. They have lots of cell wall materials. They tend to be very high in nitrogenous compounds like nucleic acids and protein. And so, having them as a sole or a major source of food presents some challenges in terms of balancing and processing foods. And there were palatability problems. They just tasted like algae. But maybe many of these types of things can be solved now with modern techniques of engineering and selection. It was difficult to maintain chemostats and production systems for long term. You'd have to shut down and clean up. Sometimes you would get other organisms that would get into the system, and bacteria and maybe even protozoans that would cause havoc. And if you're in a weightless situation, microgravity, because algae are aquatic organisms, you have to deal with liquids and weightlessness, and so you have phase separation, gas liquid separation challenge. But these are dealt with. The other thing is algae don't transpire water like higher plants, so they don't have aerial organs like leaves that act as evaporators. And so with higher plants, if you're clever, you can kind of couple them with waste processing approaches. And they might not be convenient for point source labs. I don't know about that last one. So good positives and some negatives with algae. And here's some general reviews that came from that time. You see a classic paper by Elliott Myers there, and then a review by Miller and Ward. And on our organizing committee, we have Frida Taub. Professor Taub was involved with some of these studies in the 1960s and wrote this great review paper in the reviews of ecology and systematics. So a little more just from NASA perspective. These two documents are dated 1966, and they talk about closed life support systems and bioregenerative life support. So NASA as an agency and the Russians and other groups around the world were thinking about these things in the 1960s. And in the case of NASA, a lot of the focus for the biological work, the bioregenerative aspects, focused on chemosynthetic organisms and especially hydrogen metabolizing organisms. And I'll get to this in just a minute. At that time, the genus was called Hydrogenamonas, and I'm not sure I'm pronouncing that right. I think later it was changed to Ralstonia, and now it's Cuprovitus, and I'll ask John Hogan for a fact check on that. But point being, they were hydrogen metabolizing bacteria. And the idea being for life support in space, you would run electrolysis systems to generate oxygen. You'd need water. You'd split that water. The oxygen would go to the humans, but then you'd have leftover hydrogen. And so they wanted to explore using that hydrogen as a feedstock for these organisms that could then generate biomass from that hydrogen. And then, as I mentioned, there was also work with algal systems. So there wasn't much work. They talked a little bit about higher plants, but not really much work by NASA at that time with higher plants. So now I'm going to fast forward about 15 years or so to about 1980. And in the late 1970s, NASA started up what they called their CELS program, Closed Ecological Life Support Systems program. And people have referred to it in different ways. They've called it Controlled Ecological Life Support System, Controlled Environment. I think the original name was closed ecological life support systems. And so this is when they drew on the ecological community to come up with suggestions and ideas of how they might proceed. And so this was sort of the heyday for ecologists in terms of NASA's engagement for bioregenerative life support. And you see some documents here. A group at Purdue, Hofhound Mitchell, advised on some selection of plants. There were just general guidelines and systems performance. A group affiliated with the Complex Systems Institute at the University of New Hampshire, Barry and Moore, Mel Avener, and others were involved in advising. Boeing Corporation at the bottom there, you see Olson and Olson. So anyway, a sort of a fundamental change in NASA's focus now. And so that's when people began to take a more of an ecological approach, including higher plants in some of these. And here I show some pictures of some of the people involved. You see Mel Avener there. There's Frida. She's one of our conveners of this whole meeting, and I hope she chimes in on some of these things. Lawrence Slobotkin, Dan Botkin, Claire Folsom, Bassett McGuire at the University of Texas, Frank Salisbury, my academic advisor. This is a picture of Frank and Cleon Ross when he was still at, when Frank was at Colorado State. He later moved to Utah State. Bob McElroy at NASA's Ames Research Center in the upper right there. Bob really led the NASA's CELS program. There was a NASA headquarters manager, a fellow named Jim Brett, but Bob was really the person that was the corporate memory and kept things running for many years. My colleagues at Kennedy Space Center, Bill Knott, John Sager, and two longstanding university investigators. Frank Salisbury was one at the top, but Kerry Mitchell at Purdue and Ted Tibbetts at Wisconsin. So I'm calling some of these folks as trailblazers for the CELS program. So a little closer look. This is really a busy chart, but hopefully you'll bear with me on this. It shows a timeline of some of NASA's activities, and I don't bring it up to the present, since I'm kind of focusing more on the historical. So you see the CELS program at that top gray bar starts up around 1980. And at the time, there was research at NASA's Ames Center in Moffett Field, California. And then there were lots of university grants. So it was, and this doesn't show all the university grants. And there were individual grants that were renewed through several cycles. And then as you go farther, you see these yellow boxes where they were called NSCORTs. The first one was at Purdue, then there was one at Rutgers, and then at Purdue. John Hogan on today, humbly speaking, was involved with the Rutgers NSCORT. So these were multi-investigator and in some cases multi-institutional grants to study bioregenerative life support. And then some other NASA field centers got involved. Kennedy Space Center in the mid-1980s, and the main focus there was a large-scale facility, a demonstration facility called the Biomass Production Chamber. And I'll show you some pictures of that. And then NASA's Johnson Space Center became involved. And the plan was for Johnson Space Center then to conduct human studies with the bioregenerative life support and combined physicochemical. And they did some of this, but they were going to build up to a large closed facility called Bioplex, which never was completed. And so NASA doesn't have any large-scale demonstration test facilities anymore. And as many US federal agencies do, they use not only grants, but they use what are called Small Business Innovative Research Grants, SBIRs, to develop technologies. So that's that bottom line. And I show scattered in here some spaceflight experiments that were done that were specific to bioregenerative life support. There are lots of space biology experiments that were done over this period, but the ones I kind of note here, like the potato leaf experiment, the mere wheat experiment, growing Mizuna in the Lada chamber on the ISS, these gray bars. Oh, the other thing I'll say is, you go back to the top gray bar, The name of the program changed to Advanced Life Support in the 1990s and early 2000s. And even for a while, it was called Exploration Life Support. And then it was called Life Support and Habitation Systems. And there were reorganizations in NASA. And so, you know, this is what happens in an agency like NASA. Right now, there's really no focus program, though, in NASA for bioregenerative life support. So, okay, let me show you some pictures of where I worked for many years at Kennedy Space Center. This was a large-scale demonstration facility, and I'm going to do a time check, keep going here. It was called the Biomass Production Chamber, and it was a hypobaric test chamber that was used in the Mercury and Gemini program in the 1960s, but it was converted to a plant growing system. So you see air ducting on the outside and lamp ballast, those silver boxes on the racks on the outside, and hydroponic tanks. And so this closed chamber provided about 20 square meters of growing area in a closed atmospheric volume. It used electric lighting, obviously a lot of power for that, and a lot of air circulation. But the idea was to take the findings, the fundamental findings that were coming from the university researchers, and then test them on a larger scale and do them in a closed system. So we wanted to get a little more realistic in terms of what we might encounter for space. And by using these recirculating hydroponic approaches, we could recycle all the water, we could condense the humidity, and then send that right back to the plants and so forth. So, and in aerospace engineering jargon, this is referred to as technology readiness level. So the university experimenters were sort of at a low TRL, technology readiness level, in terms of their research. And then this would be a higher level to demonstrate that it's a little more close to the real thing that you might envision for space. As it turns out, this chamber, because it was cylindrical and it was standing up, we had to stack the hydroponic growth trays. And in the left-hand side, you can almost see all 4 levels. So you have these shelves of hydroponic trays, and then there's an electric light bank above it, and then another shelf of trays and another light bank and so forth. And unbeknownst to us at the time, this was probably one of the first examples of vertical farming in the world. Some of you might have heard that term now. This is an area that's expanding rapidly in controlled environment agriculture, especially in urban settings. And NASA did this because it was a volume efficient approach. And that's the same rationale that are now being used when you're confined in urban settings. How do you get a lot of production from one city block, for example? Well, you stack up multiple shelves in a vertical farm kind of arrangement. And so this is kind of an interesting spin-off that NASA had a hand in terms of what's happening nowadays in controlled environment agriculture. There was a lot of work with recirculating hydroponics. So I'm showing some examples here of university researchers, wheat and sweet potatoes and rice at the bottom. And then we did some studies with potatoes at Kennedy Space Center. So using a particular form, what we call nutrient film technique, it's recirculating hydroponics, but you don't have a lot of standing water. You could grow these subterranean crops like potatoes and sweet potatoes. So that was a revelation for us. And it turns out now this is an approach that's used by seed potato propagation stock growers. They typically use greenhouses, but they use these same techniques that NASA explored. And we wrote about it. And so it's been, information that's been helpful in commercial agriculture. Another unique thing from some of these studies is and this was largely through university researchers, by using very high light levels, CO2 enrichment for C3 crops. If you elevate the CO2, you enhance photosynthetic rates. Use of hydroponics to eliminate water and nutrient stress. If you push the plants hard, you can get just enormous yields. With wheat, the group at Utah State could exceed world record yields from field settings by a factor of 3 or 4. Work that I did with Ted Tibbetts at the University of Wisconsin showed we could get about 200 metric tons of potatoes per hectare, the equivalent. which was twice the best yields ever reported. So the point being, in controlled environments that we might use in space, if you have the light intensity and you can provide the nutrients and the water, you can reduce the growing areas. So you have to do these trade studies, but you can get in very high yields from some of these systems. Another unique thing in terms of the studies was because in space you're going to have to be closed and you have to think about materials balance, you can monitor a whole canopy or whole stand gas exchange. And we were focused on the plants, but clearly there are microorganisms in these studies. They're contributing to these metabolic gas balances. And so I show a picture of one of the first of these efforts, Steve Schwarzkopf at Ames Research Center in the upper left there. A system he called sort of that was evolved and improved on at Purdue called Minitron chambers. Kerry Mitchell's group at Purdue, the upper right there. Lower right, then we did the same thing, but in our very large closed chamber at Kennedy Space Center. And then Johnson Space Center had about a 10 or 11 square meter chamber that they could close and get gas measurements as well. Just some data to show that the plants do exactly what you expect. They generate oxygen in the light, they consume CO2 in the light, and the reverse in the dark periods. That's that right-hand graph. But these closed systems also allowed us to measure trace gases, things like ethylene gas, which we worry about with plants. Plants make ethylene, they use it for essentially hormonal control of growth and development. And we can measure lots of other biogenic and even non-biogenic gases, things like siloxane compounds, which are a troublemaker right now in the space station. There was a lot of the plant selection came from just selecting from existing genotypes that were out there from plant breeders. And we're still relying on that very much. And an example of that, on the left side, you see rice plants. These are natural, these are genotypes that are already available. So you can see you can get really small rice cultivars that from the International Rice Institute in Manila, Philippines. But there was also some focused breeding work, largely done at Utah State with wheat. And NASA has also sponsored some genetic engineering work. One example is a protein-enhanced sweet potato line. There's a group at University of California, Davis that's infused a parathyroid hormone protein into lettuce. This could act as a countermeasure for bone loss in low-gravity environments. There's ultra dwarf tomato that's been genetically engineered by a group, Robert Jinkerson at University of California, Riverside. So there are tools available, selecting from existing genotypes, traditional plant breeding, or even genetic engineering or gene editing now. So across all of this work, you just have to keep the economics of life support in mind. And we always have to worry about what are the energy requirements? What's the mass of the system? What's the volume? How much crew time? I mean, crew time is valuable in space. Are these systems reliable? What are their failure rates? And in the case of plants, the work that I've been connected with mostly over the years, lighting tends to dominate some of these trade studies because it's pretty power intensive, energy intensive. So just to show you how light works, this is a plot of yields that we got from that large biomass production chamber. The y-axis shows yield. It's plotted as a rate, grams of dried mass per meter square per day. And then integrated daily light is shown on the x-axis. So you see yields are sort of a linear function of light. So light's really important to push the plants hard. And just to give you a calibration point, there's a bright sunny day on Earth and a bright sunny day on Mars. And so, as I mentioned earlier, if you push the plants harder with more light, photosynthetically active radiation on that x-axis, you get more and more yield on the right y-axis. So yield goes up with light, but that means in turn that the area required per human in meters square goes down because you're getting more yield per unit area. So these are the kinds of trades you can do. You need more power per unit area, but you can shrink your volume and potentially your mass. So that's why I say you always have to revisit the economics of life support in any of these systems. a big breakthrough in terms of electric lighting was the development of LEDs as a plant lighting source. LEDs, light emitting diodes. The concept, they've been around since probably 50 years or so, but the concept of using them to grow plants was patented by a group at the University of Wisconsin through NASA funding. And I saw one of the attendees, Bob Morrow, is online. Bob was part of this development, and his name is on that patent. And so this is something that evolved out of NASA-funded research. The concept of using LEDs to grow plants. They were handy. They're solid-state devices. They use electric direct current, so forth. But they weren't very efficient in the 1990s, and they were very expensive. But now they've improved remarkably, and they're super efficient because of materials breakthroughs, and they're used everywhere now in plant growth research and plant-controlled environment research. So another thing that NASA contributed to that has spun off into commercial controlled environment agriculture. As I mentioned, at Johnson Space Center, the plan was, and they began testing humans in closed systems with plants. And you see Nigel Packham there looking out a porthole from a small chamber where he lived for 15 days, and it produced all the oxygen that he needed to keep him alive, wheat plants. And then this was expanded to some larger chamber studies. Here, it was called the 20-foot chamber, and they used 4-person crews, and they did a 30-day test, a 60-day test, and then a 91-day test. And for the 91-day test, they had a small chamber inside that grew lettuce, sort of a salad machine type concept. And then they connected it to an external chamber that had the wheat in it, 11 square meters of wheat. And then they took the oxygen from that plant chamber and sent it to the human habitat, and then took the CO2 from the human habitat and sent it to the plant chamber. So, but that's where it was, the next step was this Bioplex facility, and NASA stopped the funding for it, which is unfortunate. I think it would have been a very interesting and informative facility. So there were efforts to test plants, in particular for bioregenerative life support. There are lots and lots of other fundamental biology space flight experiments with plants, but I'll mention a few here just based on bioregenerative life support theme. One was a series of tests to grow wheat on the Russian Mir space station in a plant chamber called SVET. And you see Shannon Lucid, a NASA astronaut there, tending to those plants. And then early on the ISS, the International Space Station, the Russians had a plant chamber called LADA. And you see a European astronaut there helping out with one of the LADA experiments to grow Mizuna plants. And then in the 1990s, Ted Tibbets and Ray Buell and I was part of this. We conducted an experiment to see if we could grow potato tubers and weightlessness in space. And so we used a small plant chamber called the astroculture chamber, and it worked. We got little potatoes to form at the base of these leaves. So now, currently, there are plant chambers on the space station as well as some of these prior ones. The veggie plant chamber is shown in those upper photos. It's a sort of a minimal plant growth chamber. It has a light bank, and then the plants are grown in little rooting packets of solid media and time-release fertilizer. And a number of studies have grown leafy greens, and the astronauts have even harvested these and eaten them, and it's been a big hit with the astronauts. They really love it to have fresh food to supplement their diet. Veggie still, we're still trying to improve the watering system, and NASA's, still working on watering techniques for weightlessness. That's a challenge. There's another chamber called Advanced Plant Habitat shown on that bottom photo. It's a slightly larger than veggie. Both of these are about 0.15 square meters, so they aren't really that big, but that's big for space. And that bottom photo shows a picture of some chili pepper plants that were grown last year, and it went for 137 days, and I think it is the longest contiguous plant experiment ever conducted in space. And it made peppers, and the astronauts ate them, and they were, I think, pretty hot. So we can interview some of the crew on that. So my idea of sort of a time progression here of bioregenerative systems that involve plants, we're here at the ISS, supplemental food is about what the best we can do. We're just constrained in terms of volume and power. But we can take what we learned there and use that on transit vehicles, maybe going to Mars. This concept has been called a salad machine, and NASA has an effort going now called the Ohalo chamber, which is essentially a version of this salad machine that they want to build and test on the ISS. Then when you get to planetary surface systems, maybe you begin to expand up. You grow more food, generate more oxygen bioregeneratively. You couple this with water processing, and there are lots of ways to process water and other biological ways. I'm just kind of mentioning plants here. And then if you go back to the same place, you keep scaling up, expanding more, and then maybe eventually you get to a more autonomous system. And with that Herb Ward comment in mind, that you're always going to need some engineering help to kind of completely balance these systems.
Hey, Ray,
just
5 minutes. 5 minutes for me.
Great. Okay. Thank you. Thank you. So this just graphically shows the same thing. We're at the left. We're just doing supplemental things. I think we're relying on physicochemical life support, but as we go forward, bioregenerative systems will become more and more important. So maybe we can conduct a priestly experiment on Mars, where we use plants and instead of mice, we have humans in the closed system, and perhaps where the sprigs of wheat, potatoes, lettuce, and others will not at all be inconvenient to their human companions. So thanks very much for this opportunity to talk with you, and I really look forward to the conversations today. So that is it, and I will stop sharing.
Thank you so much for that great talk, Ray. I never get tired of looking at this great body of work that has been accomplished. I see Frida applauding there.
Yeah.
I'm applauding as well. We have go ahead, Ray.
Well, I was going to say, if Frida has any fact checking on the history there, please chime in, Frida.
I'm always in awe of just how far we've come and how much we've learned. And I hope that we won't have to relearn too much as we move forward, but that's why we want to make sure that we educate ourselves in all this great research and these facilities and what they've taught us, so that we know how to move forward. So there's lots of questions for you, Ray, and I wanted to tell everyone this is working great, actually, as you're putting Q&A into the your questions into the Q&A box, and also into the chat room. I'm catching those as well. We won't have time for all of your questions for Ray right now, but at 11:15 Eastern, when we do the brainstorming, if there's any questions that haven't been answered, we'll be able to come back to them. So just to let you know that. The great thing about being MC for today is I get to choose which questions I want to ask first. So the one that really I'm excited to ask you about, because I think it's very important and relevant, is Emily Matula's question. Emily's asking, she says, inevitably, the political climate influences NASA's involvement in space ecology. Do you see current and future programs expanding under NASA with hopefully the recognition of climate change, or will the focus start to shift more to the private sector?
You want my comment on that?
Yeah, we would love your comment on that.
Yeah. I mean, you're spot on, Emily. I mean, the political environment affects government agencies like NASA. And we're going to hear from Christophe, Lasseur, and in agencies like ESA. Any space agencies, you're subject to the political environment. But I agree with your observation that things like global climate change are, now a more compelling argument that we can go back to our political leaders and say, look, we really need to understand what's going on in closed systems. the Earth's biosphere is a closed system. It's got enormous buffers, but we've seen that humans are very successful at even perturbing something as large as planet Earth. I mean, the CO2 is rising. So I think we need to use these kinds of arguments to go back to our political leadership and our respective administrations and say, please support these efforts. They're important, not just for space exploration, but for understanding what's going on Earth.
Okay. I think maybe we'll have time for one more question. There's a question about microgravity. I think I'm going to leave that one actually until later for a little more time. But one question from Davy Souza. Regarding the use of higher plants, how do you envision the cultivation of natural stimulant plants like coffee in long-term space missions?
Yeah, well, that's interesting, and woody crops in general. I think there's a role they will fit in somewhere along the time sequence. Right now, the tendency is for rapid growing annual type things like leafy greens, because you don't need a lot of volume and you can harvest them quickly. But I think as we get to more mature systems, diversifying our crop inventory to include things like spices and stimulant coffee, things like that, fruit trees, small berries. I think those are all possibilities that we need to think about. An interesting thing is, how do you get them to Mars? How do you get, especially clonally propagated or tree grafted type things to Mars? That's an interesting challenge. But I think it's certainly doable and something to consider.
Okay, well, thank you very much, Ray. Hopefully you'll be able to stay online with us throughout the rest of the day, and we can get back to some of the other questions from the group when we do our brainstorming session. Next up, we are going to hear from Christophe Lasseur, who's the head of the Melissa project from the European Space Agency, or ESA. Christophe's going to be presenting to us about Melissa, the European project for closed life support systems. And are you able to share your screen for Christophe? Excuse me. Oh, looks like it's working there. And I think your mic is muted.
Right.
There we go.
Do you have it full screen?
It looks like it's in the PowerPoint software there, but we can see full screen, yes.
You can see full screen or not yet?
Not slideshow mode yet.
Should be okay now.
There we go. That's perfect.
Perfect. Okay. So, good morning, good afternoon, good evening to everybody. I think it's very nice to see this worldwide community on this Saturday following this activity of Space and Ecology Workshop. It's certainly a pleasure for me, as Dr. Wheeler mentioned and Professor Taube, to see activity restarting on the US. I will be back on this, but when I joined this life support community as a PhD student in early 1987, the reference was clearly on the NASA side, and things have evolved worldwide, and it's certainly something that the community shall keep in mind for the future. Yeah. So you could see that there is a few colleagues which are as well co-author of this presentation. These are mainly my colleague from ESA. Yeah? So this number was already presented by Dr. Wheeler. No, not so much difference. Maybe in terms of water, I see 26 liters. On the other side, we are a bit less demanding in Europe. But anyway, the value are quite high whatsoever are the number you take. Today, probably you know that the standard mission reference is go down to 650 days and is not anymore 1,000 days. But as soon as you will have to go to food and probably to waste, you will need biological processes as well. So this means that now, at least in Europe, the message is rather clear. If you want to stay in space for a long duration, you will have to consider what we call a circular economy. This is maybe the fashion way to present life support these days in Europe, but it is certainly important to keep it in mind. Yeah? So the challenge we are facing in Melissa project, and it's clearly, again, as Dr. Wheeler mentioned, a community of life science experts, but as well of engineers. Yeah? And the challenge we often consider for us is how to assemble the different processes in order to have a full closed loop. Yeah? Keeping in mind that today, at least the way we see it in Europe, is an holistic ecosystem. It's not at the level of understanding who could fulfill the space requirement. So we are more on, let's say, an assembly of technology, of processes, including, of course, a biological one, in order to have this highest degree of closed life support system. Yeah? So on the NASA side, in the past, they mainly used the equivalent system mass, the work of Alan Drysdale, which has been quite extensively published. In Europe, we have almost the equivalent. It's called ALICE, which stands for Advanced Life Support System Evaluator. You will see that the criteria are almost the same. The only difference is on European side, we are doing multi-criteria analysis, and on the NASA side, everything is converted to mass. But that's really how we, let's say, today trade off the different subsystem and the system in order to progress in time for this high degree of closure. Things which is important on this very long-term project, you need to stay abreast of new technology arriving, and they are sometime at TRL 1, 2. Also the rest of the loop maybe at TRL 5 or 6, 7. And you need to have the tool to be able to reconsider the impact of this technology or the interest of this new technology arriving, potentially fully financed by terrestrial application, who could impact seriously your system, such as, for example, LED did a few years ago. That's something you need to keep in mind in your evaluation tools. So here is the concept of Melissa. Consider it, please, as a concept. The crew produce waste. Then you have three processes which are there to mainly transform the waste in the proper substrate for two biological processes, which are the higher plant on one side and the microalgae on the other side. So clearly, be back as well to the talk of Ray Wheeler, considering the two opportunities with advantage and weaknesses, depending in fact of where you are in your mission. Are you considering only the transit phase to Mars? Are you considering a Mars surface? Are you considering potentially a moon base? This is of course depending on the requirement, and this is what will help you. So of course, we like to present it like this. It's circular, it's easy to understand. If we would put all the arrows, it looked much more as a network than a really circular system. But some of the message that I will pass later on are easier to catch if you present it as a full circular system. So such a system is by definition totally unstable. And what does it mean? It means that you have to pilot the different flux between these different processes. Yeah? And to be able to do that, you need to have a very and almost extreme understanding of the behavior of these processes. And as it is a circular system, you cannot do it, I would say, post-mortem. You need to be predictive in the control of your flux, and you need to really act before the processes, and you need to drive it. Otherwise, your system will be, by definition, unstable. Just to give you some ideas, the matrix of input we are considering for all the Melissa processes is somewhere in around 25 elements. So that gives you already the information you need to know to be able to pilot such a system. So this means that in fact the loop you see here is more when it's approach, let's say engineer. This is how engineer will see an artificial ecology. So you see that we try to be able, of course, to go very much in the detail at different level, and I will be back on this. So what are the challenges that we have been facing, and nothing new on our side? I think it's the same for all who are trying to study this in Japan, in Russia, in Europe, in US. And we, of course, welcome newcomers such as China, Australia, and so on. Of course, it's a circularity. How to design and have a circular system, keeping in mind that if you consider a full circular system, the efficiency of the full loop will be the efficiency of the, let's say, the worst processes. So that's something you need to consider. And this is where already the advantage of a network is quite interesting. Of course, your different processes you are trying to use, either biological or physico-chemical, by the way, they have to be compatible in terms of processes. I mean, the stoichiometries has to be compatible, and this is true in terms of static and in terms of dynamics, because in space, we don't want to have big buffer. Of course, if you put big buffer everywhere, your system will be almost self-stable, but this is not what you want. You want to avoid big buffer. You want to avoid pumps. You want to avoid mass. So you need to be good in terms of dynamic control. And being back to the 80s, specifically be back if you look at this quite famous workshop around 1985 in NASA, organized by Bob McElroy, you see that there was already some challenges. How can we control biological processes, but be deterministic? Yeah? And that's really where it starts. Yeah? And you cannot limit your understanding only to carbon and nitrogen. I see the main part of the community is well aware of this, but when you explain this, people are sometimes surprised that extremely low quantity at the level of ppb can block your complete system. Of course, you will have multi-cycle. In fact, will have different family of cycle. You will have the gas cycle, you will have the hydraulic cycle, and you will have the solid cycle. And they are completely different level. So you will probably be challenged in terms of speed by the gas one, but you will have to consider the solid and the long-term drift. Of course, in a closed system, and we have seen it with COVID, biosafety may be an issue, and of course, product has to be accepted by the crew. Yeah? So Melissa started in 1987. It's quite a, let's say, a mature project now. Funny enough, it started by a first very preliminary experiment of oxygen CO2 exchange on microbial base. And this was done by a group in the suburb of Paris. And funny enough, at that time, the PI was in Berkeley and met Bob McElroy, who informed the PI that the results were quite successful, and the PI was not yet informed. So that's quite a funny story already between the exchange of NASA and Europe. So it started from Airbus, mainly with two countries, Belgium and France. And of course, it has gained maturity. Now we are somewhere around 45, 50 organizations distribute over 12 to 14 countries. So all this collaboration, and I think it's important to keep understanding this, because you need, for this very long-term effort, to be able to keep the motivation of all the actors. There is doubt that the space agency will be able alone to finance this activity. At least this is what we have experienced, a few of us in this group today. So the idea of motivation, the idea of collaboration, the idea to be able to keep as well noble task for all the actors is something important. I have doubt that it could be under the umbrella of one space agency, be able to finance everything over the years. So in Melissa, there is 15 partners, European partners who have signed a memorandum of understanding. This is, of course, maybe not, but coordinated by ESA, and they follow some of the ESA rules in order to be able to progress in this collaboration. And what we have observed, this is detailed, but what we have observed over the last years is that due to the synergy with terrestrial circular economy application, we had to revisit this collaboration agreement. First of all, to allow several directorates of ESA realize that ESA is a big machine, a bit as smaller than NASA, but still, and we have to be able to put on stage different directors signing Melissa collaboration agreement, and this we did. We did it successfully this year, so we are very proud of this. And of course, now we are as well putting on the table way to strengthen the IP issues. We have spin-off company in order indeed to foster the collaboration with terrestrial development, because we believe that on this system, on this topic of environment, sustainability and things, we are one of the actors, but we are certainly not the most active one anymore. And so that's something which I will keep on repeating. So the Melissa group is roughly around 100 people in Europe, very diverse community, microbiologists, chemical engineers, system engineer, medical doctors, and so on, all trying to work together over 14 countries, so bringing as well the biodiversity of European culture. So this is very rapidly the ESA group. Again, foster, first of all, on ECLS engineer and life science, but integrating as well now all the issue related to terrestrial application. You probably don't know, but in ESA there is now a new direction related to commercialization. So that's very important. This is why we have now 2 experts, and of course, the reinforcement of the IPs and procurement and legal issues. So, Melissa starts first ESA contract in 1987 in the R&D directorate of ESA, and all over these years, 30 to simplify, we never had a full declared customer, because of course the main focus of ESA was the International Space Station, robotic activity for Mars, but no official declaration that Europe will participate to any human mission to Mars. And this is changing now with the Terrae Novae program, which, as you can see in this document, has been drafted in September and now has been approved and will be presented to the Ministerial Council. And if you look carefully, so the date, it's really now, I would say, but if you look more carefully, even in detail, you will see that human transitability is something crucial, and Europe wants to participate. Europe wants to be active in advanced life support system and considering ground-based analog as a first step of the life support system development. You hear from Dr. Wheeler that it has been a challenge to have this after the facility he was involved with. Bioplex never occurred. BIOS in Russia is suffering. CIF has been refocused mainly on nuclear issue. So today, except the Lunar Palace and the Melissa pilot plant, there is no clearly ground demonstrator of a full closed life support system, and I think it's crucial. Rapidly, how we organize our activity in ESA related to Melissa, what we call the, let's say, the enablers, which are mainly the work which is academic. You have here it in red. Then in blue, you will have the techno demo, what we've put in space to validate this different block. And in green, which are more, let's say, other activities, which are not only focused for life support, but are very useful, such as the behavior of multi-phases processes in microgravity. And you will see now that we are proposing to keep on the activity. Things important, 3 of our demonstrators are entering in phase CD for oxygen CO2 conversion, for urine treatment, as well as food complement. So that's quite in line, which has already been mentioned before. And of course, we have a very long view, which is quite usual in space agency. So a system such as Melissa, or any closed life support system, is a very complex one. And you cannot characterize it only by asking to maintain a proper environment of all the processes. You really need to dive in the understanding of the processes itself, following the Lavoisier principle, what goes in, what goes out, what is the balance of the mass, and can you do it almost to simplify to the complete Mendeleev table. This is why we are going down to stoichiometries, of course in nominal mode, but in limitation. As soon as you will have biological processes, you have evolution, and that's something very important when you deal with engineers which are specialized of structure, of electronics, of thermal. They are not used to have processes which are evolving in terms of nature over time. So that's very crucial because you will have a lot of stress. And you may even take advantage of this different behavior to stabilize your process. It is not sure that the assembly of the technology you will have is an assembly of processes which are all functioning in steady state. There is strong possibility that some of the processes may be unstable to reach the system stability. So you should take advantage of this diversity of biological processes. Of course, if you are able to do that, then you will not have only stoichiometry, but you will have the metabolome of your complete system, and you will be able to select different pathway, different behavior for these processes. You need to characterize the processes in terms of kinetics and so on. That's of course crucial, but you cannot forget other things which are the hydrodynamics, especially in microgravity, where you will have the gas, liquid, and solid behavior, which are not that simple, specifically that you may have several gravity levels. Then you have all the issue of energy, of crucial. And then you have, of course, what this should have been translated, the organic constraint. In fact, the hardware itself, can we, if we play with the stoichiometry of a process, as well have the possibility to play with different pathways in terms of hardware. And that's certainly one more challenges. So you have it here represented once again, the different level of characterization. And then when we have all this input, you see the 5 blocks at the top, then we are able to implement this in simulation, mathematical simulation, analysis of this simulation in terms of degree of sensitivity, in situ experiment, either on ground, either in space, and of course keep on working up to a full integration of these processes together. So just to give you an example of one of the MELISA processes, which is more focused on nitrogen, very conventional nitrification process, quite classically used, at least on Earth. So you will have ammonium entering and you have nitrite, and this is done by 2 different family of microorganisms that we call Nitrobacter and Nitrosomonas. For this simplification, this one will be in autotrophic condition. If you go directly to urine, you will have to work in heterotrophic condition. But to be able to already understand and control this process, who looks quite simple, going from ammoniac to nitrate is just a triple oxidation process. You will have, due to the fixed bed reactor, because these bacteria are more acting as a catalyzer, then you will have to cut your biological processes in different slides in order to understand all of them and then put them back in order to have the full processes characterized. And if you are able to do that at the level of the exchange, then you will have, of course, to look at the dynamics, hydrodynamics, the gas-liquid transfer. How do you transfer oxygen to a column of water in microgravity? How do you recover any gas which is produced? What is the exchange between this gas and your microorganisms? This is what you need to know in order to be able to have a full control, such as what you have here. You see that here we vary the oxygen concentration of this column, and at the top right, you will see the prediction in red of what should be, of what, let's say, our prediction are mentioning, and in blue are experimental spot, who are, for this specific case, perfectly spot on. And you need to do that for all the processes in order later on to be able to pilot the complete loop. And what we do with, let's say, I would say microbial processes, we are of course trying to do it with plant, it's at another degree of challenges, as Dr. Rille know perfectly. If you look already at all the different flux you have in a plant, including, of course, the roots part, which will be colonized, potentially in axenic condition, where you will need to know what you do. Yeah? And this, if you consider as well at the level of the leaf in microgravity, you see it's already quite complex, because it's not only input-output, it's full balance of different type of processes, leading, for example, to demonstration in microgravity. This is a work which is well known. This is done during parabolic flight. This is a work of Lucie Poulet that Dr. Wheeler know quite well, to demonstrate the behavior of the gas at the leaf surface. So what we are trying to do in Melissa, and I think it's one of the specificity of this project, is really we are trying to reduce the plant or the organisms in terms of complexity reduction, in order to be able to go down at the organ levels, and progressively rebuild the plant in order to have a full deterministic understanding of the plant or of the bacteria or the microalgae, for example. We are not today going to empirical modeling, for the main reason that empirical modeling are for us considered as post-mortem, and they will not give us the possibility to react in time in terms of dynamic for the mastering of a closed loop life support system. Yeah? And of course, you cannot limit your understanding and characterization to, let's say, nominal processes. You will have to consider much more than this, and of course, you have to go down to DNA, RNA, and of course, going later on to proteic and metabolic pathways, in order to integrate in your studies the stressor, just as, for example, the radiation. And that's clearly that we will expose our processes to radiation, and what will be the impact? All our system will evolve is, of course, something which is quite important to understand. Yeah? And not least, we need as well to be sure that what we produce is accepted. You are all well aware that the organic, organoleptic quality of food varies if you are on Earth or if you are in microgravity, due mainly to the fluid shift in the human body. And this is why we participate as well with some Melissa recipe to bed rest, where we are able to evaluate the acceptability of our recipes. Yeah? Specific hardware which has been developed for Melissa. This plant characterization unit, which is in Napoli, where you could you have a very high degree of airtightness in order to be able to study traces element as well, but you can as well sample the root zone in axenic condition in order to be able to follow either the root development, but as well the microbial community at the root level. Things which is important, we have this facility in Napoli, in Italy, and we will most probably duplicate it in Norway in the coming years. Yeah? We have as well the Melissa pilot plant, which is a facility which is located in Barcelona, in the campus of the university, where there we are progressively integrating the different process of Melissa. This pilot facility is sized for 1 human. Today, we are mainly working with human waste, except CO2 oxygen, which is produced by rat. Today, the crew compartment of the Melissa facility is a colony of rat, but for those who will be able to participate to the Melissa conference in 2 weeks, you will have, in terms of breaking news, the decision that we will move to human facilities. Yeah? So access to space. Those who are active in the space domain know that you are never credible if you have not validated your technology in space. So you need to fly. You cannot restrict yourself to ground research. This is why in Melissa we already have quite an active list of, let's say, precursor of the Melissa loop. And here is the logic of this development. Very simply, the first line is focused on the oxygen CO2 cycle. The second line is focused on the urine, and the third line is focused on plant and food. So back again to the difficulty to fly techno-demo, even if you are a space agency or do accesses, and the way that very often people are doing, at least on our side, is to bid on the scientific calls, which are organized at international level. ILSRA, it's an international board, which involves as well our colleagues from Japan, Canada, and NASA. And if you are selected there, then your experiment will fly. So we enter in techno-demo via the good quality of science. And this is why Artemis, Uranus, and WAPS are currently in development and focus on the strong result that we have obtained with this precursor. We were able to propose a techno-demo, which are already looking much more as a lab support demonstrator. Biorat 1 for oxygen CO2, Biorat 2 for urine treatment, and PFPU as the precursor of a food production unit. Yeah. So just give you some example of the microalgae. I think Dr. Wheeler mentioned several times Arthrospira or Spirulina platensis, which is of course presenting such interest due to the very high content in protein, as well as growing at a very high pH, who, let's say, limit quite seriously the contamination, because you can grow spirulina up to pH 10, 10.5. So at this level of pH, of course, limitation in terms of contamination is quite good. Yeah. This was flown for around 6 weeks, 4 photobioreactors on board the space station, and we were able to demonstrate the good result of the prediction of the kinetics of the growth and oxygen production, but as well the technical separation of oxygen during these 6 weeks. And we were able as well to bring back these 4 photobioreactors and demonstrate that there was no microbial contamination at all, despite a very, let's say, very much difficulty on the post-movement of the experiment. Was already on board, and we had almost 2 weeks of delay compared to the planned launch. And when you are dealing with a biological experiment, is not that simple. Yeah? Now, this good result allow us to progress in a techno demo, which is again much more important for us, because this is something we could even consider for a Mars transit habitat. And you have here our techno demo, which is in development. We are finishing phase B2 at the moment, and we will soon enter in phase CD. So you see, it's already starting to be a beast. 200 to 300 kilo in microgravity is not already something which is light. It's already a big piece of hardware, very important and very solid for us to be able to show the robustness of our processes. So I demonstrate to you, I hope, the need to characterize all the processes, whatsoever is their family, chemical, biological, physical. You need to do the same level of characterization. And you need, of course, to demonstrate these processes when you integrate them, because you are aiming to have a closed loop. And a closed loop means several processes together. We have, I already mentioned, the Melissa pilot plant. And this is the main result of what we have achieved so far. I like to present it this way. CO2 coming from the rat plus urine coming from human allow us to reach oxygen plus protein. This has been demonstrated during 18 months. And if you follow on this screen the blue line, which is oscillated at different steady state between 19% and 21%, you see that whatsoever is the rat activity, we are able to pilot our processes in order to keep a very good oxygen concentration. Things important, if you look a bit carefully in this drawing, you realize that part of the oxygen produced by the photosynthetic processes is feedback to the previous processes, which is nitrification. Yeah? So already in terms of piloting is something which is not so trivial. because you are producing oxygen, but part of the oxygen is injected in the urine treatment in order to have the proper nitrogen source for your photosynthetic processes, which is himself depending of the CO2 produced by the rat. So you clearly see that if you don't master these processes quite well, then this system will already never stable. So the next step, and that's a bit of a, as we say in French, a clin d'œil to NASA here. We will now do the connection between the crew compartment and the higher plant chamber. We have already demonstrated that stability is quite good. So we're gonna do it first only crew compartment and plant. Then of course we will add the urine treatment previously of the plant chamber, and in parallel of the plant chamber we will put the microalgae. And this is where we are very active at the moment. So normally I would say over 2023 this result should be achieved if everything goes fine. Yeah. So something which is important, at least in Europe, is the how to finance such an effort of life support development over years. And the answer we are trying to establish is the synergies with the terrestrial sustainability challenge that we are in Europe considering. So a few examples of technology which are issued from Melissa and are becoming commercial. We have created already 6 spin-off companies coming from Melissa. Here is one of them. It's called Hydrome. It's a urine treatment. So you are able to produce a biocide that you use to clean water, and like this, let's say, reduce considerably the water need for the toilet. This has been implemented in a public park in Belgium over summer 2022 and is now deployed in 2 schools in Flanders. Roland Garros tournament, what the French Open is doing here is because in 2022, we recycle all the grey water coming from the sportman and sportwoman in order to reduce, of course, water consumption, and this was quite successful. And in 2023, we will implement as well a urine treatment unit. Yeah? Some of you who are interested from the sea know that boats are not the best example of sustainability these days. They have a very strong tendency to quit the national water area in order to drop all the waste in the sea as soon as they are in international area. And this is why with the Foundation Belém, Melissa signed an agreement in order to implement within this sailing boat, this historical sailing boat that you have on the left, Melissa technology to recycle black water, yellow water, and gray water, and act as an ambassador of what could be done on other boats. CO2 recycling is often an issue if it is already ventilated in the air, so you really need to go at the source of the CO2 production. This is a concrete production site, and where we are able to, let's say, connect our system on the CO2 production and recycle it in oxygen, but of course produce biomass who can have values, for example, for feed or food. Yeah. Another example of biomass production, which is will be implemented in French Guiana. It's in progress. It's 100% funded by the Minister of Environment in France, so no link at all with space in terms of funding, and it will be the biggest platform of microalgae production for biofuel. Yeah? This is a building which is in construction in Paris, in the south part of Paris, of course, exposed to south. And there on the facade of this building, you will have a colonize of microorganisms, and you will be able to produce a biomass with a commercial value. Yeah. So it's another way as well to trap CO2. Last but not least, the when you do research, sometimes you are facing the serendipity approach. This is exactly what happened here. We discovered by total coincidence that one of the Melissa microorganisms have a very strong effect on the bad cholesterol. So we take 15 patents, we test it on human during isolation campaign, and we are now putting this spin-off on the market. Hotels and habitation, consumption of European is somewhere around 100 liters of water a day per European citizen, 200 if he is in the hotel. So you see already is a very good civil behavior. And this is why we are focused on recycling water in the hotel. And this one is implemented in an hotel in Monaco. For you who are dealing with centralized, decentralized wastewater treatment, Monaco is really a cheese with a lot of holes, so you cannot organize a centralized water treatment, and this is why decentralized system as this one makes sense. Yeah. We as well try to participate to something which is more robust. There is area in the world where the protein is a real challenge, or there is no cattle, very limited quantity of animals, you cannot feed them. So this is why in Congo we participate to this activity where we are producing microalgae in order indeed to sustain local population, which is in fact quite an interesting return, because for those who know a little bit the history of spirulina, it was discovered in Lake Chad, of course used worldwide, and the fact that it's used back now with a bit of, let's say, technology is quite a nice return.
Christophe, you have 5 minutes remaining, 5 minutes
Okay.
left.
I rush, I've almost finished. Another very good example of collaboration with terrestrial, probably not so known under the name of Biomérieux in the US or in the rest of the world, but probably much more in the world of Biofire. This is a development we did with Biomérieux starting in 1998 to 2013. We thought at that time that to guarantee the microbial security on board space station, we need to do a fully automated PCR. This is what we developed. And in fact, now it's the first platform worldwide of COVID-19 detection. Education, this is really part of the gene of Melissa. This is why you see on this picture, we have a large number of PhD, and we organize as well Melissa Summer University in order to be able to teach and benefit of this very strong motivation. I was pleased as well to participate to other places worldwide with my colleague of GPL. And I have to say a few words about this one, because Melissa get inspired in the early days about the work done by Bob McElroy, how to gather motivation all over the country with the best American university in order to push the different topic, microbiology, algae, control system, and so on. Bob has always been a very nice person with me, and he was as well working with some of my NASA colleagues that I had the chance to meet in that time, such as Mark Lees, Ray Wheeler, and I think I invite everybody to read the sales document, because there is a lot of information there. Last but not least, the Melissa conference will be in 2 weeks in Toulouse. Registration are still open. It's probably a bit late for you to travel, but you can participate remotely. And of course, if you want to come, you are welcome. The registration are still open. And just to mention that we have every 3 years the Melissa Summer University, where we welcome PhD. We are not obviously Melissa actors. And on this, I would like to thank you very much for this invitation and this opportunity to present you, Melissa. Thank you very much.
Thank you very much, Christophe. That was a wonderful talk and introduction for many of us to Melissa Project. I think we're also very excited to see all of these spin-off technologies. I think today more than ever, we all realize the important implications of this work on our sustainable systems for living on this planet, as well as off of it. So that was very impressive. I'd only have, I think, time maybe for one question for you, and there's some other questions as well that we could talk about later on today when we do the brainstorming session. One question is, what factors contribute to how comfortable we are relying on a biological solution, like a nitrogen fixing soil bacteria, versus a physical chemical solution, like a nitrogen bioreactor. How do we know or determine which is the more reliable or dependable solution?
Mhm. So the trade-off metric that we always use is the ALICE one for Europe. So we really look at the mass, we look at the energy, and we compare with physico-chemical processes or other processes. Yeah? Of course, the challenge is the knowledge. Are we able today, or do we have the same knowledge of biological processes versus physico-chemical one, even if the TRL is the same? Yeah. Or what do we know in terms of biology who will, let's say, not become a weakness in the future, even if the ALICE criteria or the ESM criteria are the same? And this is why in Melissa we include a knowledge readiness level, where we consider that the 5 levels are presented has to be understood to have a proper knowledge readiness level of the processes. And so you have to consider this is not only hardware, is as well the your understanding of the processes, which has to be integrated in the trade-off of the technologies. And that's something which is not always easy to explain to deciders.
Thank you very much. Thanks, Christophe. We're going to move on now to our next speaker. Our next speaker that we have is William Dempster, Bill, who is the Director of Systems Engineering for the Biosphere 2 project from 1984 to 1994. And he's going to tell us about the Biosphere 2 concept, its purpose, and give us a visual tour, which I think I'm very excited to see. So with that, I'm going to hand over to Bill. If you could go ahead and share your screen.
Yes, thank you very much. It's a great pleasure to be able to present to this group. And thank you so much for organizing this whole conference. Here goes share screen.
That looks great, Bill. Thank you.
Okay, great. Okay. I know many people know about Biosphere 2, some may not. But in any case, I'm going to give you a tour. So here is an aerial view of Biosphere 2. And as you can see, this is a very large facility. And before I go into some detail about the different sections that you're seeing in front of you, I'm going to take a tour through some of the sections inside, and then I'm going to come back to this same picture. But first, I'm going to just put up a little screen about what is a biosphere. And let's see, is our sidebar blocking this in your view? It is in my view.
No, it is not. We can see the whole screen. So if you want to take a moment to see if you can move that sidebar out of your view so you can see.
It's okay. I don't need to move it for my purposes.
Okay. Looks great to us.
Okay. So anyway, what is a biosphere? And it's a complex, multiple interdependent life forms that are indefinitely self-sustaining and evolve. And it's materially closed within negligibly small limits, of course, regarding the materials. But it is open to energy and information beyond its boundaries. And why did we call our project Biosphere 2? Well, because Biosphere 1 is the planet Earth, of course. A recycling closed systems, many people might not think of it being as a closed system just because it's so big and they don't see that it's closed, but it's huge. and it's closed and it's sealed by gravity. So now I'm going to show a little video inside tour of Biosphere 2. We're going to go inside to the top of the tower. That's the library. This is inside the human habitat area. The agriculture area where food was grown to feed the 8 occupants. Included domesticated animals. Rainforest. Ocean and savanna. Marsh. Desert. Marsh again. Ocean. Technosphere. These are the systems that support the operation in the basement area. Okay, that was a brief tour, but I'm going to go specifically through certain individual areas. This is an overview of the agriculture area. This is about a half an acre, and it was where the food was grown to feed 8 biospherians, that is the 8 crew members, and also the domesticated animals, which included chickens, pigs, goats, and fish. This is the rainforest. This is very early in the rainforest before it grew up to touch the ceiling at the top. An artificial mountain that was built there, and with a waterfall falling, there was a pond at the top of that, which you don't see in this view. But it filled the whole top of that artificial mountain area. And the pond and the rainforest waterfall fell down to this stream, which ran into the savanna area. And here is the ocean. This is approximately 800,000 gallons, about 3 million liters. About 20% of it was brought from the Pacific Ocean, specifically to include the microbial life that is in real ocean water, and the rest was mixed from local fresh water mixed with commercially available sea salts to create a ocean. Marsh, the mangroves here were collected from the Florida Everglades, and it was immediately adjacent to the ocean and had water flowing from the marsh into the ocean. And again, the desert. What you see through the space frame in the background is one of the two expansion chambers that we called lungs. That is connected to this interior space by an underground tunnel. Now here is, again, the first view that I presented, but with labels showing where the different areas are. So going first to the wilderness areas, this is the rainforest on the left. That's about 20,000 square feet of footprint. The long section here is the savanna, the ocean, and the marsh. And then the desert on the right, slightly smaller. And in front is the agriculture area, which is about a half an acre where the food was grown. And this was the habitat in the sense that it was the living quarters for the 8 crew members. And also in the front here, there was a brief view of that in the video. There was a central office area, which included working desk space and computer connections for each individual biospherian. The top of the tower was a library, which was the very first inside view that we had in the video. The animal bay was here in the bottom. That's where the chickens, pigs, and goats were kept. And in the behind there, in the ground floor area was a workshop for maintenance, included a full machine shop. There's also an exercise room down in here. The airtight footprint of this is over 3 acres, 1.25 hectare. And the enclosed volume is 7 million cubic feet, which is about 200,000 cubic meters. And I want to emphasize that all of this is an airtight space. This is one of the 2 expansion chambers, and I'm going to go into much more detail about what that looks like inside later on. And there are 2 of them. One is here on the right. This is the south lung over here and the west lung here. And in the corner, you just see a little bit of the energy center, which I'm going to go into a little more as well. We provided electric power from here. We could generate our own, but we were also connected to the electrical grid, but also heating and cooling by circulating chilled water from this center into Biosphere 2, all in sealed piping systems, so that none of that heated or chilled water that came from the energy center ever mixed in any way with any of the ecological systems water that was inside. Now I'll speak for a moment about the purpose of Biosphere 2. There was a lot of, I think, confusion about this. fed by sensational stories in the media. But the basic system idea was to create a laboratory for global ecology in which ecosystems can be experimentally studied. So this was a experiment, and to provide a laboratory for doing ecological experiments. And we called it Biosphere 2, a closed system. The degree of sealing was certainly less than 10% a year atmospheric leakage. My personal opinion is that it was significantly less than that, but I could conclusively demonstrate by testing that it was less than 10% per year leakage. Experimental system configured with 5 major natural biomes, which I mentioned before: rainforest, savanna, ocean, marsh, and desert, and anthropogenic biomes of human habitat and agriculture, which are human-created biomes. And our purpose and intention for this is to use the method of progressive approximations. To start with a first approximation, observe results, modify to a next approximation, and repeat and continue. And the intention was to continue in this manner for 100 years at a physical scale and a time scale which are appropriate for studying biospheres, which of course, drawing from planet Earth, are physically very large. Well, we couldn't build the whole planet Earth, but that what you saw so far is what we could build as a practical facility. Collections, we stocked over 3,000 plants and animals. This is Linda Lay and Peter Warshall in South America doing collections for stocking Biosphere 2. Here they are at a termite mound. This is, we included termites in Biosphere 2. And now immediately shifting to the Caribbean, where our coral reefs were collected for going into the ocean. This is Gay Alling diving in the Caribbean off the Yucatan Peninsula. And support of this diving activity was by this sailing ship platform, which is a sailing ship that the same team that started Biosphere 2, we built this ship a decade earlier. It's an 85-foot Chinese junk design that's about 25 meters long. It has sailed the world oceans for 40 years, currently in dry dock in Spain for a major rebuild. Here's what it looked like from a visitor's point of view, looking inside to the agriculture area. And here is Mark Nelson, one of the 8 Biospherian crew members, greeting through the glass Oleg Gazenko, a Russian scientist, on the left, and John Allen on the right, who was the inventor of the Biosphere 2 idea. This is living quarters. This is Mark Nelson's apartment. This is his living room. And on the left, in the corner, you can see a spiral staircase going to the second floor, which was where his bedroom was. He was looking out through the window over the agriculture biome. Here, 1 year in, and by the way, the crew of 8 people were inside for 2 years. This is 1 year in, photograph taken of them on the beach of the ocean taken from the outside through the glass. And here is the shop, the maintenance shop. This is Mark Bantillo doing some work on a lathe that was inside the machine shop. And here is Gay Alling again diving in the ocean, this time inside Biosphere 2. You saw her diving in the Caribbean before, collecting coral reefs. And this illustrates how temperature and humidity control were working in Biosphere 2. This is a cross-section of the agriculture area. And what you see here is the air circulation. This is the growing beds on top. And in the basement underneath is the air handling unit. The air is taken into the air handling unit and is first cooled by evaporatively cooled water, then chilled water, then heated water if necessary to reheat, which controls the humidity. Of course, condensate drips off of these, and that's a method of recollecting water, which is very pure, of course, because it's essentially distilled water. And then the air circulation goes through the space where it is heated by sunlight and becomes warm and humid again from evapotranspiration, and then recirculates. Now at the bottom, the glazing system was airtight, sealed with silicon seal. The bottom was sealed with a stainless steel liner on top of the concrete slab, which joined with silicone to the bottom of the glazing system. So concrete is nowhere near airtight, of course, and so there was a stainless steel liner completing the seal. This is one of the air handling units. You only see about half of it in this picture. This is 12 feet wide by 9 feet tall by about 30 feet long. And its air discharge is 50,000 cubic feet per minute. And there are 25 units like this to manage the temperature control and humidity control in Biosphere 2. So that's a very big operation, big energy intensive operation to control the temperature and humidity. This is the energy center where the chilled water and evaporatively cooled water was generated. These three cooling towers evaporate water to cool it. On a hot summer day when cooling requirement was maximum, we would evaporate about 200,000 gallons every day. And these other buildings are the other parts of the energy center with generators and chillers. This is one of our three electric generators. That's 1.5 megawatts. There were two others, also 1.5 and 2.25 megawatts generators. This is one of the three chillers, 200 tons. That's a English unit of description, meaning 12,000 BTU per hour of cooling capacity is what is meant by 1 ton. And this is a 200-ton chilling unit. And these pipes were the chilled water circulation that went to Biosphere 2. But again, these pipes were all sealed so that none of the water that was delivered from here to Biosphere 2 ever intermingled or circulated with the ecosystem waters that were enclosed inside. Now we're going to study the construction of the lungs, the expansion chambers. What you saw before, the domes, were merely weather covers for the actual mechanism. What you're looking at here is a tank, which is 158 feet in diameter and 24 feet tall. And in the center of it is a aluminum pan, a dish, which is 92 feet in diameter. And right here in the corner, you see the mouth of the underground tunnel, which connects underground to the main Biosphere facility, so that the air that's contained in this expansion chamber is continuous with the air in the main facility. And this shows the membrane being placed around the edge of the pan. Here they are drawing the membrane. So you see the edge of the pan there, and this is the edge of the tank here. And overhead is the dome that you saw in the previous pictures, built from space frame. And then this is a view from inside the lung when it is partially inflated. To give you a sense of scale, of course, you see people here in the foreground on the left. And this pan, which would rise up and down due to the expansion and contraction of air within the main facility. And there were two lungs like this. The pan would go up and down approximately 20 feet every day. And that was the degree of expansion contraction of the air. And this illustrates the system for neutralizing the pressure. If you think about that pan, which weighed about 15 tons, and the membrane, which weighed about 3 or 4 tons, resting on the air that's trapped inside, the weight of it would create pressure of the contained air. Not very much pressure, actually, only about a ½ inch of water column, which doesn't seem like very much. But it was of interest to make it completely neutral, the pressure difference between inside Biosphere 2 and the outside surrounding atmosphere. So the design was to have this small fan in the corner here, which was controllable, and it would blow air from underneath the weather-covered dome to the outside, which created suction under the weather cover inside, and that suction would uplift the pan. And by controlling the operation of the fan, you could exactly neutralize the amount of suction to match the weight of the pan, so that that would give the pressure of the air inside to be just exactly equal to the outside ambient pressure of the biosphere. And by exactly, I mean within a tolerance of about 1,000th of a PSI. That's 8 Pascal was the plus or minus range of the pressure inside Biosphere 2 compared to the outside ambient pressure. Now I'm going to show a graph of what carbon dioxide looked like inside Biosphere 2 during the 2 years of closure. And you see seasonal fluctuations. Started off at ambient concentration of CO2, which is around 400 parts per million, and went up during the winter when sunlight and photosynthesis were low to between 3,000 and 4,000 parts per million. And then in the spring and summer, it descended again to about 1,000 parts per million. Then in the next fall and winter, it rose up to nearly 5,000 parts per million. And then the next summer down again. to this time about 2,500 parts per million. And then as we were heading into fall, it was starting to rise again. This is a more detailed examination of 4 days in the CO2 graph. The CO2 is on the top for 4 days, and the ambient sunlight light level is in the bottom. So what you see is as the sunlight comes on in the day, the sun rises and the sunlight goes up, then you see the carbon dioxide, which had been rising all night long, begin to fall as photosynthesis takes over until it reaches the low at sunset, and then it rises again from respiration at night and then falls again and rises and falls. And notice on this particular day was a cloudy day, so sunlight was quite low. And then you see these disturbances in the CO2 curve during the photosynthetic period. There are little bumps in the curve there. And we used to say, of course, you don't need to look out the window to know whether it's sunny or cloudy. You can just read the CO2 graph.
Hey, Bill, you have 15 minutes left. 15
Right.
minutes.
Got it. And this is showing oxygen. Oxygen declined over 2 years, starting at 21% and declined after 16 months to about 14 and a half months, in which point we injected oxygen to rise it back up to 19%. And that's a whole story, which I won't try to explain right now, but maybe in questions. This illustrates a method of detecting leaks in the liner. The liner is underneath the water flood here, which is about 1 foot deep. We flooded the basement with water and then pumped air under pressure in channels that were under the liner, so that if the welded seams had any flaws, we would see bubbles coming up. And we did, we found a few flaws and fixed them. And this shows the evaluation of leak rate, which was by trace gases in the atmosphere. So insofar as leakage occurs, that is exchange of outside air with inside air, you will then see the progressive dilution of the trace gases. And we used helium and sulfur hexafluoride, and also an independent test was done by Columbia University using krypton. And the dilution rate is very slow. Here's 350 days showing the sulfur hexafluoride going down at a rate of maybe 8% per year. And what do we need to do? I'm going to compare to the development of rockets. Well, rockets for 100 years have had people building them and playing with them and doing all sorts of possible experiments. And my point is that in order to understand how to make closed systems work, depending on what you mean by work, say to go to Mars and provide life support, we need to do something similar. We need to build hundreds, we need to build thousands of these closed systems and do all sorts of experiments. And so that's my point about what we should do. And biospheres, if they're really closed, they're tricky, as we saw with the decline of oxygen. So there's some further information. I'm going to suggest people look at this documentary film, which was released in 2020. It's 2 hours, and there's other references to some books, and there are publications as well. So with that, I'll open the floor to questions.
Thank you, Bill. I think I can speak for us all that we are just so happy to have been able to see this presentation. And for those of us who have not seen it before, it is amazing to see what an engineering feat Biosphere 2 was and still is. And for those of us who have seen it before, to be reminded of that fact. I have several questions for you. So I'm going to, I'll just start here at the top. Joya Massa is asking whether the Biosphere 2 designers, when they were selecting the sizes of the different terrestrial ecosystems, how did they select the size in proportion to one another? Do you have any insight into that?
Well, I'll only say that in our first talk about doing the project, we had the idea that it was much smaller, but as we really began to get into it and study the idea of what could be the representative size of a biome that we were drawing as from planet Earth, we felt that it had to be significantly larger. And so that drove the size up. And of course, then there was the question of feeding a crew of people. Of course, the number of people is somewhat arbitrary, but we chose 8 or provided for the possibility there could be as many as 10 in Biosphere 2. And that drove up the size of the agriculture area. So we ended up with half an acre.
Okay, thank you.
And over 3 acres for the entire airtight footprint.
Someone has asked, did you encounter any engineering challenges developing Biosphere 2 that you think would be an obstacle for a more permanent space station? Engineering challenges that might create a problem for building something like this in space?
I'll say yes, in the sense that my particular focus was on making it airtight, and that was really the main thrust of my work. And we found out the hard way when we built a smaller test module and tried to seal it with gaskets and so on, and found out that it leaked very, very badly. Then we went to another system, but we also, thinking about the problem of the pressure differential between the inside and the outside. If we didn't have the expansion chambers that we called lungs and just had it sealed, then the problem is that the expansion and contraction of the air will create tremendous pressures. And that facility, as you saw, simply couldn't have withstood those pressures. It would have basically exploded or have been crushed by excess atmospheric pressure from the outside. So if you're going to go to Mars, where there is essentially zero pressure in the atmosphere, 1% of Earth's actually, you're going to have to deal with the pressure. And so how are you going to deal with that? And that goes to the shape of the enclosure and whether or not it's an inflatable. And if you have even a pinhole anywhere, you're going to have a very high rate of leakage. My estimate from testing, which I did by an independent method with Biosphere 2, is that the total sum of leaks was about a cumulative hole of 3/4 of an inch diameter in the entire facility. That's about the size of a finger. That's the accumulative number of holes that existed, which I could quantify with too long an explanation to go into here. But the prospects for loss of atmosphere in a facility that's in space or on Mars are very, very difficult.
What material was that long membrane? What was it made out of?
Hypalon.
Did you
That's
say hyphalon?
chlorosulfonated polyethylene.
Okay, thank you.
And it was about 3/8 of an inch thick.
It's very, very impressive.
And it was fabricated in 2 layers with the seams of the 2 layers staggered so as there would not be a seam anywhere that would have a continuity from inside to outside.
Greg has asked that CO2 absorption into the concrete. Does anyone know if it's stabilized over time since the original experiment?
We don't have quantification of this degree of stability, but I'm quite sure that it did stabilize because, of course, eventually as the CO2 absorbs into the concrete, it's creating calcium carbonate. It was absorbing into the concrete because it was reacting with residual amounts of calcium oxide and forming calcium carbonate. And of course, as the calcium carbonate formed, it was reducing the amount of calcium oxide that remained and forming, transforming that to calcium carbonate. So the answer is it certainly must have been, but we don't have a quantitative measure of that. Although core samples were taken in 1994 of the concrete, and we could see elevated concentrations of calcium carbonate compared to unexposed concrete that had higher levels of calcium oxide and lesser levels of calcium carbonate. So I think the answer is yes, but I can't give you a quantitative answer.
Thanks. Talking about sensors, well, Ray Wheeler was asking whether you guys measured methane in the enclosed air, but also I was wondering what other kinds of things were sensed and measured, and it must have been a tremendous amount of sensors throughout the facility, but methane in particular.
There were about 1,000 sensors in the whole facility, mainly recording temperature and humidity, but also light levels and soil moisture. And there were sensors for gas, but mainly for gas sensing. What we were doing was taking samples and running them through a gas chromatograph. And we were also exporting samples for analysis by outside laboratories. And yes, there was some methane there. And unfortunately, I don't remember what that number was. But it didn't get to a level of being a serious concern.
Sorry, I have so many questions here, just trying to pick the there's been some questions about insects. I don't know if this might be outside of your area, but well, Ray asks, well, he hadn't heard about termites in Biosphere 2. He's wondering how the termites did. And another person was asking, actually, this was Alan Johnson, the fact that there was over 3,000 species. Do we know what level or how many of those species persisted over time, or were there ones that died out? And were there any that were deliberately added in afterwards?
There were none that were added in. I don't know the numbers between plants and animals. Yes, some species went extinct. In particular, there were birds originally, and they did not survive. But we also, I'll mention, by the way, included some primates, bush babies, some very small primates. There were 4 in the beginning, and there were 7 at the end, and we didn't add them. They were reproduced on their own. I don't have quantities that I can talk about how many species survived. I think there were significant numbers of extinctions. There was an article claiming that all the pollinators died, which is not true, because it is true that bees did not survive, but there are other insects creating pollination. There were fish in the ocean, they survived. There were corals, they survived. There were small animals like lizards and snakes. And there were many insects that survived, but I don't have any numbers to give you.
Okay, I think we have time for maybe one more question, and then I'm going to move over to the next segment of our day.
May I interject? Sorry,
Yes.
one thing. In anticipation of extinction, we followed the strategy of overpacking. We put in more species than we considered were the essential number, and anticipating that many would die out. I'm sorry, go ahead with the other question.
Oh, right. So the idea really was that species packing would allow some stability to occur naturally for the species to, what's the word I'm looking for? Basically Darwinian effect for the most, why am I'm blanking on the actual term here, well suited to the environment to survive. So yeah, I'm familiar with that idea that species packing was in order for the best assemblage to win out. So I think one last question, there's some others here that I may pose to you later, is basically if you had the chance to do it all over again, what would you change? And also part of that question, I would ask, if you were to do thousands of these facilities, what do you think that they would look like? Would they be more simple? What pieces would you keep? That's two questions,
well,
sorry.
Basically, at this scale, I wouldn't really change anything for starting. The point of the experiment was to learn. And of course, one of the major learnings, of course, enormously mentioned in the press, was the decline of oxygen. And we added some artificial lighting to increase the photosynthesis and the food production. So that was a big lesson to learn. I can hardly imagine that anyone will build a facility this large again, or they might sometime, but really much smaller facilities can be built. There should be room-sized facilities or desktop-sized facilities, and there should be many of them built at universities, even at high schools. People who like to tinker should be doing them. We might have tried to make less concentration of richness in the soil, which would reduce the rate of CO2 and evolution and the rate of oxygen loss. But that's a very subtle question, because that resource in the soil, being rich, was the source from which the biomes had to grow from infancy to maturity, and there had to be a carbon source. So the question of excess CO2 or oxygen decline is much more complex than just the simple one of being at a steady state at some later stage. So I'm sure many will build facilities in different manners in the future, and hopefully they will learn from what we have done or did. I will mention, of course, if some people don't realize, the pictures I showed were from 1990, 1, 2, 3, and 4. So the current facility is quite different as it's being operated by the University of Arizona.
All right. Well, thank you so much, Bill, for joining us today. I'm so glad to have you with us, and so many things that I hadn't seen before. So it was a wonderful presentation. Thank you.
And you're very welcome, and I thank all of you.
Okay. I'm going to go ahead and Patrick, do you think we should take a few-minute break, or do you think I should roll right into the next segment before lunch?
I think we've got a couple minutes for a break if we want to before we jump into the mural introduction and lunch.
Okay, so tell you what, folks, we're going to reconvene at, let's see, 25 after. So for those of you who need to take a little break, and then when you come back, I'm going to provide a little instruction on how we're going to do this brainstorming session before our meal break. So we'll see you again in 6 minutes. Bill, if you wouldn't mind, if you could hit the stop sharing, and I'm going to go ahead and load my next set of slides here.
Okay. There we go.
Thank you. Hello, everyone. And we are going to go ahead and get started again. Hope you had a nice little break there. So to recap a little bit and to give you a little guidance for this brainstorming session here before our lunch or other meal break, why develop an ecosystem in space? I think we all have a general idea of why that's the case, but what I like to remind everyone is that living in space is a tremendous sustainability challenge. The further that we travel from our planet, the more it's going to pay off to reuse and recycle whatever we consume rather than carrying all these supplies with us and then throwing away the waste. And I believe that if we can learn to do that in an uninhabitable environment like space, we can live sustainably anywhere, even on Earth. And so while packaged food stores, they have a limited shelf life, they lose quality over time, we have plants that can provide us regenerable, nutritious food, turning carbon dioxide into oxygen, purifying wastewater, and making us happy, reducing our stress. And plants and other organisms like bacteria and fungi and all these other components of our system can produce many products also that we might use in our daily lives, like medicine and fuel and clothing. But it's really hard. This is not easy to do in a space environment. We need all the same things to grow a plant or support other organisms in space, like water and gases, minerals, and light. But you have complications doing this. You're extremely limited in how much you can carry with you on a spacecraft and how much volume you have available for a closed ecological system. You have microgravity and reduced gravity. We haven't talked much about that yet, and I think we will. This really complicates water transport and air mixing and root aeration, and it can confuse organisms' gravitropic responses. You've got high radiation, you've got reduced pressure sometimes. You might have elevated CO2 levels. And by the way, you're also sometimes launching at very high accelerations and have very high vibration loads to be exposed to. This can physiologically stress all of the organisms out. And sometimes it can be really hard in such a small isolated container to control chemical releases and buffer these processes and buffer all of these components and transfers of material. So if something fails, you can't just go to the store and buy a new part. So there's also reliability aspects we have to think about in our testing. So keeping all of this in mind, in the next segment between now and 12:15 Eastern time, we want to reflect on everything excuse me, that we have learned since the 1950s and 60s. Through decades of research in these ground-based test facilities, so much has been accomplished. I've just shown some examples here, some we've talked about today, like Melissa, like the NASA Lunar Mars Life Support Test Project and the Biomass Production Chamber at Kennedy. And then also there's work going on in this field in Japan and in China. There's the CES facility at the University of Guelph there. Eden, we haven't pointed out, in Antarctica. So there's a lot of work that's going on, right? And there's also a lot that we've learned in microgravity in the space environment on how the space environment affects organisms, especially plant growth. And so just some of the microgravity space agriculture or crop production facilities that have been flown and tested are shown for you there. So we've demonstrated some enabling technology that we can use in microgravity to grow plants as well as other organisms. And we've shown feasibility that bioregenerative life support can be safe and reliable, but I think we still have a long way to go. We're just beginning to understand the importance of plants' microbiome, beginning to understand the importance of modifying the plant's environment to control nutritional quality and not just yield. The list goes on and on. I'll let you kind of look over this. But what we'd like for you guys to help us with today in this first brainstorming session is to identify and prioritize needs and gaps. We've looked at a lot of different needs assessments, literature reviews, and we've organized some of these existing knowledge gaps into 3 groups: organism level questions, ecosystem level questions, and technology needs. And these are some of the topic areas there that you see, some that questions that remain that we need to research and technologies that we need to develop. So keeping that in mind, what you're going to now see on Mural, let's see, I'm going to ask in the chat, which I've lost my chat, Patrick. There it is. Aha, okay, everyone should see a link in the chat to our mural. And we'd like you to tell us if you see a missing topic, to add one, if there's certain questions or capabilities that remain unanswered, knowledge gaps and technology needs. We want you to go ahead and put them on the board. And also there's a question on this mural regarding what you think is most critical to answer first. So some tips and tricks for you here as everyone is getting over to that mural. You can just put that link into your browser. And I'm not going, I have just a picture of Mural here on the screen for you. This is not the live one. Maybe I should actually share the live one, just one moment. Okay, you guys should be able to see the live mural on my shared screen here. You'll see some cursors moving around. Some people are putting some stickies up, notes up on the board. Just some tips for you though. On the very right side of your screen, there's called an outline. That can help you navigate to the different sections. So if you want to look at organism level, you can click on that topic and it'll zoom in to that part of the board for you. So I'm going to do that. So I'm zooming into organism level. If I want to look at technology needs, I can click that there. You can click on your screen and drag this around to get to any place on the board. You can use your mouse to zoom in and zoom out. You can't see. You can make your text as small or large as you want to, but try to keep it a little smaller so everybody else can fit their ideas on the board. See, some other tips for you. All you got to do to add a sticky note is just double click on the screen. Just like I did there. It's pretty simple. But on your left, if you want some more fancy tools, there's some text you can add, titles and comments and other text boxes. This is really important, folks, as you're adding your thoughts and ideas to this board. If you accidentally remove someone else's comment, please try to put it back, click that undo button. Here's the undo button up there. Actually, I'll go ahead and try it out. So I just put a sticky here. I click undo, it went away. So try to be careful to be respectful of everyone else's content. And one final tip for you guys. There's a comment feature. It's a little confusing to use. We just don't recommend you use the comments, so probably just stay away from them. There is up here, there is a chat box. Patrick says hello in the chat box. Feel free to use that. We can talk in multiple ways today. Many chat boxes available. So with that, I'm going to bring those tips back up and share that again. So we're gonna just let you all brainstorm and put your ideas about knowledge gaps and technology needs. What questions do we still need to answer in order to realize closed ecological systems, especially for space life support? And as you are doing that, we'll keep going until 12:15 Eastern time. So that's about 40 minutes for this brainstorming session, excuse me. And please go ahead and put questions into the Zoom chat or the Mural chat if you have any troubles or technical difficulties.
If we have any lingering questions from some of the earlier talks, maybe we could discuss those verbally while people are on Mural so that we can kind of spark that conversation as well.
Absolutely. So I have a list of questions, Patrick, I've prepared just for that occasion. So there's many questions that we didn't have time to get to for our panels. So before I start, let's see, do we have all of our speakers here online? Let's see, Christophe, are you still online? Kind of doing a roll call here. Although he may be busy brainstorming on the mural. Ray, are you online?
They're probably taking a well-deserved break.
Yeah, probably. So I might wait a few minutes before I
Sure.
do that.
Ray's online. I just couldn't get to my mute button fast.
Sorry, we should slow down a little bit. Let's see. Bill, looks like I see you're still online there.
I'm still here.
Okay. I think I'll wait another moment and see, because I had some questions for Christophe as well. I'll tell you what, Ray, let me go back to, I think there were some specific ones just for you that I would like to ask. I don't know if I should keep harping on this closure question, but I think it's a big question that is unanswered, Ray. But I'm wondering your thoughts on if we know that closure is unnecessary, how do we know what level of closure is necessary? How do we answer that question?
Yeah, I don't know. It's I think, I mean, if you want full autonomy and Earth independence, then you know, high closure will be needed along with maybe local resource use, in situ resources. So you're still probably open to in situ resources, but maybe don't need to import things from Earth. So, yeah, I think it's a question for, you know, further study and debate. I think it's sort of a cost benefit analysis. You probably can get there, but it maybe becomes prohibitively expensive. And maybe at some point you say it's just more effective to, are we going to build our own pumps and compressors and sensors when we get to Mars? Are we going to import some of those from Earth, or computer boards or things like that? So I'm thinking closure in the sense of a lot of the technology needs as well, not just closure.
And I think really, like you just said, it comes down to cost savings versus perhaps additional complexity that might affect reliability, and how much effort you have to make to get there. Is the a prior boss of mine used to say, is the juice worth the squeeze? the benefit you're gaining over the cost to get there.
But Chris, I will add sort of an observation from over the years of, you know, working back and forth with ECLS engineers, and I saw there was a question about ECLS versus cells. I think as a community, as a bioregenerative community, we need to be cautious in dealing with space agencies, for example, and not overstate our case. And by that I mean, you know, say, yes, we can contribute we will be needed to improve autonomy, but not say that we're going to take over kind of attitude. I think you tend to you encounter resistance in space agencies when you make those kinds of statements. Now, from an esoteric perspective, studying closed ecological systems, that's fine, and understanding them. The applications of them, it's an evolutionary process to me. That's just purely a Wheeler observation, and others might have different opinions on that.
There was a question that, actually, just a note for everyone online, the Q&A box, as these questions are being answered, some have been answered in writing. So if you click on the Q&A, the answered tab, you can look at some of those responses. So feel free to read through those. One of those questions, Ray, that you answered in the Q&A box was, oh goodness, I've lost my train of thought. I think it was about the fungi, and what assemblages of, have there been experiments using assemblages of different plants with fungi?
Yeah, there was one question about increased complexity. Will that give you more capability to improve closure? And I think yes, probably so. I think this is a question for more systems ecologists to think about and analyze. Again, I think the economics enter in. Is it more cost effective to get the last 10% closure or whatever biologically, or get that with chemical engineering approaches? So you just have to do those trade studies. The one on fungi, what was?
Just whether there's been experiments with symbiotic relationships between plants and fungi.
Oh yeah. So it probably depends on your horticultural approach. If you're using conventional hydroponics with nutrient fertilizer salts and water, and you're managing it in the standard approach, you're going to get organisms that'll colonize, and some of them might be beneficial, but the things like that mycorrhizal fungi add, where they help mine nutrients from solid media, you might not need that in hydroponics, but then there might be some more interesting biochemical relationships that between fungal organisms and higher plants and bacteria and things that can promote beneficial effects that I don't understand completely, but there might be others that can talk to that.
Christophe on the line? Okay, sorry, Ray. I thought I would see if he had anything to add from a Melissa perspective on that one, but I think we'll come back to that. Lori Irwin had asked earlier, Ray, whether the biomass production chamber used a dehumidifier to recapture and manage humidity.
Yeah, we did. We had standard copper condensing coils with an external chilled water supply. The types of technologies that Bill was showing for, not the scale, but the same kinds of principles that Bill showed for Biosphere 2. So, and when we first started, we were just dumping the condensate, and then we were adding deionized water back to the nutrient solution, hydroponic tanks. But then we closed that water loop, and because it was a copper condensing coil, we had to run the condensate through an ion exchange column to remove copper and zinc, and there was a couple other elements that would kind of ablate from the condensing coils. They're trace elements, but in hydroponic systems, which is what we were using, there's virtually no buffering capacity, and so we had to control things like copper. But yeah, and then we would recycle that condensate right back to the plants. Now, in a functioning system, you'd send that clean water to the human habitat, and then you'd retrieve the human water, the wastewater from urine and hygiene and whatever else that you would process. And if it's to some advantage, you couple the plants along with into that process as evaporators. But yes, we were condensing the condensate or the humidity.
Thanks. I had a question. Ray, I'm going to keep picking on you, and then I have some more questions for Bill, if you don't mind. Do you think, you mentioned those world record yields for the potatoes? Well, there was probably world record yields for other plants as well. Do you think those record yields could be maintained over long durations for multiple life cycles? Or what do you think are the challenges with maintaining those high yields?
Yeah, I think they could, and bear in mind, I was talking just one production cycle. So in a controlled environment, you maybe could get 3 or 4 field production cycles in 1 year. So, those numbers would be even higher. One of the keys to that, especially with wheat, for example, was extraordinarily high light levels. The group at Utah State did these studies. Bruce Bugbee and Oscar Monje was part of this. And so you can push grasses like wheat and rice with really high light intensity. And so they were using levels that were like at least twice, if not 3 times that of maximum field kind of light intensity. And the wheat could hang in there. So it just says that you can push these plants harder. And the other thing that it said to me in terms of field agriculture is there is still untapped potential in our crops if we can manage them accordingly. Now, field settings are more challenging, obviously. You can't control everything. But the potential is there.
Oh, last question for you, Ray, then I'll leave you alone or open it up to our panelists. Is do you have any particular lessons learned from BPC that you think were the biggest surprises, most surprising, unexpected?
Half of our, I'm just putting a rough number, half of our headaches were associated with the hydroponic system. Sensors, pumps, leaks, just kind of mundane stuff. And so, and they could be resolved with better design approaches, higher quality hardware and sensors and so forth. But, that's the way we went into it. Unfortunately, we did not gather good crew time data. We were really research oriented, so we were more research than operations oriented. And so we have some crew time data, but it was research intensive. So I'm not sure how meaningful it is. The other thing, final observation general, and there's probably lots of others. We did not really have any biological failures. And by that, I mean, with one exception where we had an obvious pathogen get into the system, it was very likely a Pythium organism and it affected some wheat. And so we pulled the plants out and cleaned up and started again. The plants performed very predictably. Our failures were really in the engineering and the hardware side. A pump would fail. A blower would fail, the power would go out. We had a hurricane and we had to shut things down for 3 days. The plants and the biology were actually quite predictable. And maybe we were just lucky with pathogens, but if you use safe practices and you develop HACCP programs and protocols to mitigate introducing nuisance organisms, pathogens, things like that, I think you can manage those. But that's an area you always have to keep on your radar.
Yeah, that thought occurred to me earlier looking at, I believe it was Christophe's presentation mentioning biofilm, and we always mention biofilm in pathogens. And there seems like we have not begun to understand how to maintain a balance between pathogen control, biofilm control, so it doesn't clog up all of your pumps and tubes without killing all the good microbes. I think that's a huge research question, is how do we keep the good guys and get rid of the bad guys while still controlling those biofilms, or make our technology more robust to biofilm. It can work regardless. So that's
I see Frida had a comment.
Frida, go ahead.
Oops, you're muted, Frida.
At the Algal Biomass Organization meetings, which are going on right now, there's one study, for example, if they added particular bacteria to their algal culture, they prevented rotifer infestations. So, you know, that was an interesting way of solving a problem. The problem that I've been having in my research, which is looking at very small closed ecological systems, where I try to publish papers comparing our results to lake results. The reviewers say, why did you bother doing this? So I haven't even bothered trying to put in any National Science Foundation proposals or anything else. There doesn't seem to be an understanding that closed systems are something worth studying in terms of the ecologists that I've come across.
I'll chime in there too, Frida. We did observe rotifers in our hydroponic systems occasionally, but they were, if they're grazing on bacteria and algae, we didn't really care.
Well, but that was good from your point of view.
Yeah, yeah. The other, this is kind of an interesting one. For only one crop, but it has sort of ecological relevance. Only one crop, rice, where we added silicon to the nutrient solution. Rice just likes to have silicon. It grows better. It's kind of a borderline essential element for plants, but it makes the plants sturdier and so forth. Well, when we did that, we got a bloom of diatoms. All around the edges of our hydroponic trays, we saw brownish biofilms and things growing, and we thought, what in the world are these? And our microbiologist extrayer looked at it and saw these little spiky organisms with beautiful silicaceous designs and spikes and patterns, and we had diatoms. Now, they went away once the plants shaded everything. Just like any algae, they languished and kind of died off because they weren't getting enough light. So we didn't really worry about it, but it was an interesting observation in that our systems were silicon deficient for diatom colonization until we added silicon, and there they showed up.
Yeah.
So that was kind of a neat thing.
I've been watching the Algal Biomass Organization for studies where algae are used in any significant quantities in human or animal diets. Most of the studies are as food additives, which is anywhere from 1 to 5%. There are a few studies. I think there's one study, I've forgotten whether it's rabbits or some small mammal, where up to 15% of the diet was from algae. But I haven't seen any big breakthroughs where microalgae are the major part of the diet in any form. I'm still looking. I'm still watching.
Is Bill Dempster still there? There you are.
Yeah,
Hello,
I'm
Bill.
right here.
I have a few Biosphere 2 questions, if you don't mind.
Go ahead.
Well, the one question that I've had somewhat of a debate with some mechanical engineers in the past about the pressure control in spacecraft greenhouses. And I think that the lung idea personally is elegantly simple and robust because of that simplicity. But I would just like to find out how you would respond to what you think the advantages of the lung are over, say, just using vacuum pumps and compressors to control that pressure. If you didn't have a variable volume and you're just using active pressure control.
Well, I'll just comment that if you're talking about going to space, lungs are really irrelevant because on Earth, the pressure difference would naturally be fairly small. It would, the pressure inside and outside, they're already going to be fairly close to each other. I mean, in terms of an order of magnitude. Lungs work for closed systems in a ambient pressure, which is comparable to the ambient pressure you want to have inside. But if you, they wouldn't have any particular relevance if you were on Mars. But you're right about the simplicity, and it completely passive. You don't have to do any. There's no active equipment necessary. I mean, my little fan under the weather cover dome was just a refinement. It would and did work fine without the fan at all. There were some times when we turned the fan off. But trying to do it by anyway, what are you going to do in a closed system if you do use pumps? You're going to have to take some air and put it somewhere, or take it from somewhere, which means that then you have to have a storage tank that you have to put under high pressure or evacuate to vacuum or something like that. It just gets smaller, that's all.
So, oh, go ahead.
No, that's pretty much the thought. Go ahead.
Okay. Another non-hardware question. Emily Matula asked earlier whether there was concerns about CO2 exposure near 0.5%. I guess that was the 5,000 ppm over long durations. Don't know if you can speak to the physiological aspects on that. But were there concerns about those high CO2 levels for a long time?
Well, I can't, from any expertise or knowledge that I have on my own, all I can say is I don't think that we suffered anything. And then, of course, then I think most people know that some people have studied carbon dioxide levels in people's houses when they have a party, for example, and they see similar thousands of parts per million of CO2 under those circumstances. But that's, of course, it's true that that's not long-term. And my own sense of it, but that's just intuitive, and I don't have study data on it, is that you'd probably be perfectly fine up to 10,000 parts per million and then begin to think about it. But that's just off of the top of my head. I'm not qualified really to answer the question very well.
Let's see here. Oh, Nathan, I'll go ahead and address your question. Nathan O'Neill had a question for Frida, for Professor Taub.
Okay, I'm unmuted.
Hello there. Nathan was wondering if you have some good links for algae organizations that you were speaking about, and any additional recommendations for literature on algal bioproduction and spaceflight. So I'll start with directing this person to the program for starters, but then I'll let you take that.
Well, if you just look up ABO, Algae Biomass Organization, they've got a website, they've got a lot of information on it. The big hurdle that they're facing right now is the dichotomy between growing huge amounts of algae for biofuel, which is technically feasible, but the price is yet high. If you can have some other product like a pigment that can earn a whole lot of money for a small mass, then maybe. So it looks like the field is technically feasible, but not yet economically feasible, except that there are a few companies that seem to be doing pretty well. So they're really, really pushing on that mass culture of algae. But they've got a lot of different members and a lot of different interests in it. Right now, their meeting structure is that they are meeting from essentially 8 o'clock in the morning until 11 over most of the month of October. So, you know, you can join the organization, or you can pay registration fees. There is a registration fee for their workshop. So that's one of the organizations. And then there are several basic organizations. The Ecological Society, for example, is a very long-running organization, and they put out a journal that has beautiful, beautiful micro photographs, which is one of the reasons I get the paper version of it. So there's an overwhelming amount of data out there, but none of the answers to the questions you're going to ask.
Thank you.
There's a big push also on macroalgae right now that turns out to be feasible for human food, for pharmaceuticals, for auger production. So there's a lot, there's enough information there to bury you.
On macroalgae, Professor, I was wondering, often wonder about the feasibility of macroalgae production in spaceflight, given that in a aquaculture sense, it takes apparently a lot of water.
Well, it not only takes a lot of water, but most of them are marine. I don't know of any freshwater macro.
Right.
And in terms of production for food, for example, they regard it also as a way of removing nitrogen and phosphorus from water. So they get credits for that. They get CO2 credits. And there seems to be an increasing industry, in addition to some open ocean macroalgae, where if a storm is approaching, the whole thing goes down in the water column.
Okay.
So I don't know of any space applications that would be relevant at the moment, but a lot of research going on.
I would love to have a seaweed salad in space, but I'm thinking that's probably not going to happen. So,
well, I mean, we don't know if you're going to sometimes have a problem in a closed ecological system of building up salinity, for example.
Yeah.
But there's a lot of information out there. There are organizations that really focus on helping you grow these things.
Yeah. And also, I know that on the salinity piece, it's important to try to find species that can, that are, what's is the word halophyte, that are salt tolerant, that can take up a lot of salt, because in order to be able to process urine, that's helpful. So that's why another reason I like duckweed. It's good for that. Actually, I would like to pass a question back over to Bill, if that's okay. Hi, Bill.
Sure.
I had a question about lighting. So I remember reading articles about sometimes during the 2 years that Biosphere 2 operated, there was an unexpected number of cloudy days. It's normally very bright and sunny in Arizona, and it was unexpectedly cloudy during that time period. if you guys had to do it all over again, did you consider artificial lighting to supplement the sunlight? And, what were some of the reasons behind relying on sunlight?
We added artificial lighting after the first 2 years.
Okay.
We started a second experiment, by the way, which only ran for 9 months, in which we weren't present to finish.
Mm-hmm.
But yes, we added artificial lighting specifically because the crop growth was lower than we expected and attributed to low lighting.
Mm-hmm.
We had 2 El Nino years in a row, as it turned out, which was cloudy in the wintertime in particular. It was very, very cloudy, and light levels were very low. So the answer is yes. And so in answer to the other question that I was asked, maybe I didn't answer well enough about whether we would have done something different, is yes, we would do something to ensure more light, which basically would have to be artificial lighting, because there was significant shading from the space frame. And then the general shape of the structure brought in shadows early morning and late afternoon, which didn't help either. So lighting increase, definitely. That goes along with Ray's comment about the Utah studies about getting a lot of production out of wheat. But it's generally true, our lighting was too low. And even though we said that we had much world, so to speak, world record productivity in our plant growth, that's also like, so what, in a certain sense, because we had a super controlled environment of temperature and humidity. And compared to field, of course, what else would you expect? But lighting certainly should be more, and we would have to turn to artificial lighting because you kind of can't do more than we already had with the glazing.
Okay, another question I have for I think this is for Ray, actually. Hi, Ray. Lori was asking whether you used mycorrhiza in your experiments, and if so, was there yield effects or increases? And I would ask that actually of Christophe if he was on the line as well.
Well, I can answer quickly, no.
Okay.
We did not. Most of our systems were hydroponic systems. Now, they might have been there unbeknownst to us. We don't know. But we did not deliberately look for them or add them to our systems. When you get into solid media and people talk about using, in situ regolith in the future and things like that, maybe there are, good roles for things like mycorrhizal fungi. Yeah. I don't know about for Christophe, though, if he's on.
Christophe, are you there? I guess he's gone and taken a break.
Hey everyone, I just wanted to point out we're about 5 minutes away from the meal break. 5 minutes.
Hey, great. Thank you, Sherry, for keeping me on track.
It's what I do.
It's very helpful. I guess there's another, well, I have two kind of loaded questions to ask, probably for Ray. I would like to hear Christophe's response maybe later when he returns as well. Actually, this is one for Frida, for you as well. I know that in past conversations, back when folks were discussing closed ecological systems in the 60s and 70s, one of the debates between ecologists and engineers. There's two big debates. One's reliability. Put that aside for the moment. But the other debate was whether or not to control evolution. How much control, evolution or no? What do you guys think?
I don't think you have any choice. Evolution is going to occur. There have since been studies where E. coli are transferred each day, and they now have 12 or 15 years worth of DNA data as to how, I don't know whether they started with a single genotype or multiple genotypes, but you can't stop evolution from happening. Somebody can disagree with me.
Seems like Ray is nodding his head in agreement.
Well,
Seems
one
reason-
thing you can do is continually kill off anything that appears to be a mutant. I mean, if you have an organism, an agricultural organism where you know what it's supposed to look like, and you kill off anything that looks aberrant, maybe you can prevent it that way.
Bill, you had a comment?
I was just saying it seems logical to me what Frida said, so, but I'm not an expert.
I like that idea, Frida. So it's about fitness to the environment, right? Whoever survives is who's most fit, and killing it off, making it an unsuitable environment for those that you
But
don't want.
Ray has his hand up.
Ray.
Yeah, another thought, and I'm sure we've all talked about this on and off over the years, is, and this I think it would affect evolution, is that when you leave and become a biological island as you go off into space and you head to Mars, you now no longer have movement of genetic materials in and out of your system like we do in our own laboratories, in our own field gardens, and wherever. I mean, bioreactors and so forth. And so your genetic inputs are now limited, and you have what you have. It's a classic island biology kind of issue from an ecological perspective. And
But
so
you can
that's
lose
You
things.
yeah,
You can lose things.
yeah.
And apparently, the rate of mutations, I like to think that the fact that I'm error prone and I very often mistype, apparently that happens a lot in genetics too, where you didn't start with that organism and you didn't plan on that organism. Well, I think we're seeing it with the viruses that are going around right now.
Mhm. Well, that raises the question of, even though you don't have genetic material coming from outside the system, you still can have mutations. And so then the question becomes, is a mutation viable or beneficial or something like that? And I don't know if studies have been done of mutations that occur and try to label them in some way that says 90% of them are not viable and only 10% of them are viable or beneficial or something like that. But that would be a very interesting question in that context.
Well, an example would be if what we want to harvest is the seed, and there was a mutation in which a lot more went into the vegetative growth, but not the seed. It would be a disadvantage from our point of view of reducing yield, but that plant might be better able to outcompete others.
Let's see. It looks like it is time to break. We have a lot of activity going on the mural board. Everyone, this is really exciting to see. I just can't tell you how thrilled I am to see that the mural seems to be working out well. If anyone is having any issues or difficulties, I'd like to do a quick poll in the chat. If you think there's any negative feedback on the mural that we should know about, seems like there's a lot of positive, but if they have any criticism or advice, we'd like to know how we can improve. Go ahead and let us know in the chat there. You are free, everyone, to keep working on the mural. It's still going to be there throughout lunch or whatever meal in whatever time zone you are in. And so you're free to keep brainstorming, but we're going to go ahead and break for 45 minutes, excuse me. We'll be reconvening at 1 p.m. Eastern. That is 11 a.m. Mountain Time, 10 a.m. Pacific Time. But we will be reconvening in 45 minutes. And at that time, we're going to hear from Barbara, as well as Cyrille and John Hogan. So much more to come. And thanks, everyone, and have a nice break. Hello, Barbara. Nice to meet you in person.
Nice to meet you too. Nice background.
Yeah, one of our presentations earlier today was a tour of Biosphere 2, but I like to hang out here.
I am a huge fan of Biosphere too. I love this place.
Have you ever visited?
Yeah, I was there last, I think it was April or May.
Wonderful.
So I stayed there a few days. Yeah, I love the space.
Well, thank you for hanging in there for this late talk today for you. It's 7 p.m. there, right?
Yes, exactly. I'm glad the connection is working. That was a big. I was a bit scared of that, but it's all good.
Great. Well, I'm going to be offline here for just a moment, and we're going to convene back in about four or 5 minutes. In the meantime, you are, let me stop sharing my screen. I think I'm sharing. And you're welcome to go ahead and load your screen or screen share. I want to check to see if that works.
Hello. It's on the moonpad.
Oh, looks good. We can see your screen.
Yeah. And when I do that, is it full screen?
Yep.
Okay,
Great.
cool. Okay.
Okay. Everyone, I've been working through the lunch. I'm going to run off and get some coffee, but I will be back in time.
Okay.
Okay, I'll be right back.
See you, Christine.
Oh, and I meant to introduce you, Barbara. Patrick online there. You can see Patrick is one that's been sending all of our emails and our technical support for the day and basically keeping us on track. So
super cool.
Great to meet you, Barbara. Really glad you could join us.
Me too. I'm super happy. Where are you right now?
I'm in Seattle, so you can see this is about as bright as it'll be until next summer. This is what we're expecting the weather to look like.
And you have some LEDs behind you and plants growing in
Oh,
hydroponics.
yeah, of course. Lots of stuff going on back there. These are all my tropical plants and carnivores.
Yay!
I saw the biopod unveil.
Yeah.
Oh,
Cool.
exciting.
Yeah, that was really cool. We've been working on it for some time. It's actually quite fast for from product design to manufacturing, but it's
you
cool.
guys are opening up a factory.
Yep. Yeah. So we actually have already 25,000 square feet in south of Paris already. So we have a lab and we have prototyping area and we've been assembling the biopod at the facility already. But we need to build much bigger place to start that production, but it will take some time. We start with a small factory, so we can at least cover the first 100 units, but we need to build much more. So we'll do it in Europe, in France, and then we have to scale in the United States, but it will take probably 2 years. So
That seems pretty fast for scaling.
yeah, 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 we're trying. But that's cool, and at the same time working on the space product. So
yeah, I'm curious. I hope that you go in a little bit on what the difference will be between the terrestrial version and the lunar version.
And the lunar version, yeah, I'll explain a little bit. It's going to be we have a 5-year program. So to turn the terrestrial one into lunar biopad. So, but I'll talk a bit about that, yes, during the presentation.
Awesome.
Hi, Jane.
I got the camera on there, but I'm not directly out of bed. I took not directly. Yeah, the subtitles are a disaster.
Oh, really?
I found them pretty helpful.
I think it depends on exactly how the person talks.
Ah.
Yeah.
Okay,
Is it not picking up European accents very well?
Oh, I just closed it because I know it totally screws up my speech in the most interesting ways.
I will try to articulate.
So far, I think it's been pretty good as far as my ability to understand. I think everybody has been okay with our different dialects across this globe. Okay, so
Yeah.
Patrick, I'm gonna I think just have us jump right in.
Let's do it.
Any objections?
That's it.
Okay. Welcome back, everyone. I hope you had a nice break. And for those of you who didn't take a break, there was some activity still on the mural. And also I'd like to mention that as the talks progress throughout the second half of today's workshop, feel free to continue putting your ideas and thoughts down on Mural. And as you've been doing, chat away in the Zoom chat. And also if you have questions, as a reminder to our speakers today, this afternoon, go ahead and put those questions into the Q&A, and I will do my best to present those questions to our speakers for you. So this afternoon, we have with us, very excited to introduce Barbara Belvisi, who's the founder and CEO of Interstellar Lab. And Barbara, you'll have to tell us the title of your talk because it just escaped my memory. I apologize. So we're very excited to hear Barbara tell us about Biopad and what's going on at Interstellar Lab. So I'll just hand it over to you.
Thank you, Chris. Thank you for putting this event all together. It's a wonderful gathering of awesome people. Some I've known from some time, so it's glad to see the community gathering and the work that everyone's doing, which is a very important work because if we can't grow food in space, the future of humanity there is, I think it's going to be complicated or robotized, but yeah, so what I want to talk about today, so let me just share here. Can you see my screen?
Mm-hmm.
Yep. Okay, great. So Interstellar Lab, we're a company. We've been around. So I founded a company late 2018. So it's been 4 years. And as a company, our purpose is to help growing life everywhere. So today I want to talk about the future of farming on Earth and in space, because actually it's very there is a lot we're learning from space design that is applying on Earth to solve some of the key agricultural problems that we have here. And there is a lot of things we can actually do right now on Earth to start, building the system, the knowledge, the automation, all the software, and part of the hardware before we are able to bring system in LEO and then on the moon. So I will explain a bit more about how we are pushing it. Just a quick recap on the vision for those who don't know about Interstellar Lab. So everything started with this vision of becoming multiplanetary species, which is, I think, like many of you, was a child dream, and it's still I still consider myself having a very vivid child spirit. So this dream is still one that I have, and I'm a big defender of the, of becoming a spacefaring civilization, and that it's a very, very exciting future. But I believe there is a need for a narrative where the future is not about leaving the Earth and settle on other planet, but the future is about, how we can at the same time protect the life that we have on Earth and bring this life with us together in space travel. And so that led us to the core of the company, which is really to build 12 modules. So everything started, I think Christophe, I saw Christophe Lasseur here. So you see me presented the company 4 years ago at the very beginning, Ray Roller as well. And so everything started with a vision of a lunar station, and the station will be composed of different modules. From this vision of the station, which at the time was called eBios, and the station was a combination of controlled environmental module, and we move over the 2 first years from like this big bioregenerative station to actually focusing on the core module, which is the module dedicated to plant growth. So as a company, we develop, we build, and we manufacture a controlled environmental module, and we adapt depending on where we have to deploy them. But the vision is to use them. I mean, right now on Earth, it's more than a vision. We actually are commercializing the product. Is to use those pods to do indoor farm agriculture. So very efficient, uses less resources. And I'll explain how we turn this like lunar pod into a space pod, build a space pod, and how we're going to go back to the moon after that. But so using those pods on Earth, this module for sustainable farming, and also bringing them in the future in space so we can help expanding life in space. And so the team, not everyone is here, but we are around 30 people now. And it's very, very multidisciplinary, because I think, and I believe we all agree about that this field of, agriculture in space require a lot of different discipline, whether it's hardware and design, engineering and system, manufacturing and assembly, software and artificial intelligence, but also agronomy. So when I started the company, it was very important to surround myself with all those people that have this knowledge, because it's a team project, and more than a team, it's also a community of engineers, scientists, and different people working on that. So but at the very essence, I believe that if you want to deal with biological life, and especially understanding how to grow plants and understanding the plants, you need scientists and agronomists. So the agronomist team is actually a very big part. Not everyone is in the slide, but it's a third of the company as coming as a botanist or plant science background. And it's very important to understand what the plants need if you want to build the system around it. And otherwise, I think it's hard. So the vision as a company that we have is, so we start. It's called Interstellar. We start with multiplanetary before going interstellar. The travel technologies hasn't been built yet, so we can travel interstellar. But the vision for us is really to look at the Earth, and then Leo, and then the Moon, and then Mars. And so one step at a time, how we can develop system modules inside which we will grow life. And of course, the first planet that we know that has life right now is the Earth. So we started with this planet, also because we're living here, so it's much easier to build a greenhouse on Earth than put one on the Moon right now. But then the next step is going to be low Earth orbit, and for that we have 2 different approaches. Which I will show at the end of this presentation, but it's an inflatable module to be attached to space station, but also cubic system that are different version, but kind of similar, but different in their automation and the watering system than VG and APH, but also bringing some more growing capabilities in Leo because we need data and we need testing. Our plan is also to be ready by 2027 for for the lunar Artemis base. And by doing the different iteration between now and 2027, that we need to do on the biopod design, because the biopod, which is the name of the first product you see here, that is designed for Earth, that will not work as it is right now on the moon. So there are like a couple of, more than a couple, but there are some iteration that we need to do before we be able to install one on the moon. And then the target is to be ready for Mars by the 30s. We haven't defined like a proper roadmap strategy yet, because it's coming a little bit later down the road. But that's our vision, and that's as a company what we're gonna build, a different range of product that we're gonna work on. I'll start with Earth, and I'll explain a little bit more about the product we've been developing, because it's because the one we're gonna work to put on the moon is gonna be very similar in terms of how it works. So everything around some of the basic principles on how you control the environment will be very similar. The software will be similar, the automation, the material, the watering system, the atmospheric system is going to be different hardware, of course, but the automation very similar, and the access to the plant catalog. But you'll understand. So the biopod, it's actually designed as if it's designed as a completely sealed structure. So it was inspired, as I said at the beginning, we started first with a conceptual design for a lunar base, and then we took the module actually in this lunar base that was dedicated to plant growth and did an adaptation on Earth. So this back and forth between space and Earth, I think is very important in our industry, because it's space has very strong constraints when it comes to mass, when it comes to efficiency, when it comes also to redundancy, to minimize the input and and maximize the output if you want to feed the astronauts. But so we took all of those principles and we developed a product that fits and meet the terrestrial requirements. But it's in concept, it's very similar to a space product. So it's the biopod is designed with a composite base, which we assemble on site. And so it's composite like you find, in aircraft and in rockets. So it's it's structural to the biopod. It's helping us getting all the hardware and all the system that we have inside, but also it's creating a very good thermal insulation. So it's very warm inside where you have all the hardware. And so we need to protect and to isolate the base, and so composite was a very good material for us. So you have this composite base, then at the entrance you have an airlock. So actually you have two doors that are creating separating the inside of the biopod from the outside. Inside the base you'll find all the hydraulic and atmospheric components. So for a biopod to work right now, you don't need anything but access to power. So it has a little, actually big cable that you just plug into the grid, and then it works by itself. When it comes to water, it's equipped with 3 water tanks, and so you just need to fill up the water tanks. There is a kind of a hole where you can put the pipe on the side. You fill up the tank and then you close it, and then you don't have to touch it. So inside the base you have the water system with the 3 tank. The tanks are connected on one side to the automated nutrient dosing solution. On the other side, they are connecting with a water purification treatment unit, which is composed of a UV disinfection system and a combination of different filters. It's equipped also with dehumidifiers, so we get all the water that evapotranspirates through the plant in the atmosphere, we get it back into the system and we purify it. When it comes to the air, so the biopod is an inflatable structure, so it breathes. So we have blowers units that are taking the atmospheric air from the Earth. Obviously, that will be different on the moon because there's no atmosphere. It might be similar but different, but something interesting we could do on Mars by taking the CO2 that is already in the Martian atmosphere. But on Earth, so we take the air from the terrestrial atmosphere, and we actually equipped the biopod with a CO2 scrubber, so an amine CO2 scrubber, and so we scrub the CO2 from the outside atmosphere to elevate the level of CO2 inside the biopod. So we enrich the atmosphere inside the biopod up to 1500 ppm. We can do much more. more, but plants don't really like more. They can get burned. So it's a good level. And so we pump CO2 from the atmosphere, we store it, and we put it in inside the biopod. And of course, we have an AC unit to cool down the system. All of that is fully automatized. So there are two computers kind of that are attached to the different hardware on which we run programs. So we've been developing different programs depending on the type of climate you want to recreate inside. So the biopod is really autonomous. You don't have anyone controlling it. If you want to control it, you can do it, but it's autonomous by design. And then on top of the raised floor, there is all the aeroponic system that we designed so it can be modular. So it means you can play with the LEDs. And so depending on the size of the plant, and we want it to be modular and also independent in terms of the irrigation for each racks. So meaning you can play with the spectrum of light, blue and red, and you can play with the timing of irrigation as well as the nutrient solution that you want to go in by racks, meaning you can have different type of plants. They all together in the same pod. They will all need to be able to live in the same temperature and humidity environment. Then on top of the biopod, you have an inflatable membrane. The material we use on Earth is called ETFE. It's a very durable, very resistant material, also very sustainable, and it lasts over 50 years. On Earth, we only do 2 layers. There is one part, this one was a test. We can do fully transparent, we can do fully opaque, or we can do a combination of both. Currently, we are doing combination of both just to test the difference on how the light from outdoors is actually affecting the plant growth. And so we do cushion, it's one piece membrane, this is where the innovation is, and we do a cushion air without any supporting aluminum frame. And so we have this transparent membrane that is taking light from the sun, but is also authorizing us to have very good insulation, because when you have a cushion, you create air is a very good insulator. The vision for the moon will try to see, ETFE is a very good candidate. On the version for the moon, we need to test a different approach. We would like to keep the transparency, not because it's for the moon, it's not because it's we're going to take sunlight from the sun. That's not really the reason. It's more of a design thing that I would love to see, a greenhouse on the moon where through the greenhouse you can actually see the plant. I think it's very inspiring, but it's going to be a combination of different material and probably a 4-layer membrane with a cushion of aerogel. But we need to do a lot of testing before that, and this is part of our 5 years roadmap to transform the biopod. So on Earth, it's very sustainable because it's, it was initially designed to operate in space. As a concept. So it takes CO2 from the atmosphere. It produces a lot of biomass because it's a closed system. So you can really recreate the perfect condition for the plants to grow. It reuses all the water and it reduces land footprint on Earth. So we've been designing this product that's actually solving initially for space and adaptation to Earth. And right now on Earth, it's solving a lot of the problems that there are in agriculture. As I said, it's fully automated. So we have a very nice application where you can check all the different levels of CO2, humidity, temperature. The biopod is equipped with a lot of sensors that are measuring in real time. So you can look at what's going on inside in real time. If some of our customers, they want to have access to play with the different scent points for like R&D purposes. Some other customers, it's purely commercial. They want to grow vanilla, they want to grow vetiver, and so they just run the program. But you have the possibility in the app to have access to both. And this is very important because when we design it, we thought also as well, as at the space version of our product. And what you want to limit, of course, is the human intervention. So you want to limit crew time. You want a system to be the more autonomous as possible. So astronauts in the future, they don't have to spend too much time in the greenhouse, even though we expect them that they will really enjoy the weather and the smell, and probably will spend a lot of time in greenhouses. But I'm kidding here. But so so this application, and this is where I'm saying that there is so much we can do from designing terrestrial product and doing this back and forth. Actually, the design of this application that we're doing for our terrestrial customer will be adapted to our next space product. So we can really leverage the knowledge that we have on Earth and to develop space product. The plant catalog. So our vision for the future of farming is the combination. It has to be a combination of three things. The first one is, of course, you need a hardware. Without a greenhouse, it's going to be, or a closed system, it's going to be really hard to grow plants in space. The second part is that you need software and automation, because it's very key to limit crew time and to automate all the systems. So you need to develop the intelligence and to plug the hardware really with the software and AI part. But last but not least, you also need to know how to grow plants. And so for that, what we've been developing is what we call the Biopod catalog, which is a library of recipes on how to grow plants. So for instance, we've been growing vanilla for a year and a half right now. We will publish very soon because we're having we work a lot with Wageningen on vanilla. Vanilla doesn't seem like a key species to grow in space. It's actually super strategic on Earth. It's disappearing. It's getting harder and harder to get access to vanilla. But so for vanilla, we've been growing this plant for a year and a half, and so we develop based on all the data that we gather from the experiment and the experience of growing this plant. We developed a program, and a program is a combination of time and set points. So based on the cycle of the plant, we define from day 1 to day 10, every day, every morning from 9 to 11, the temperature is going to be, I'm saying figures that are not correct. It's going to be 24. Then we want the light to go more in the red spectrum versus in the morning, the more in the blue spectrum. And so all of this data, we turn that into code, and this code is programmed that you can run the Biopod. And it's based on actual knowledge on how to grow plants using our system. And so we started a year ago actually building this catalog. We have a lot of different species and we adding more and more, but it's really all the product that we are developing, whether it's on Earth or in space, it's really the combination of the three, which is the hardware, the automation, the software, and the plant catalog, really understanding what the plant needs. On Earth, and we talk a lot about, how space, and especially in agriculture, can help us design better system to tackle some of the key issues we have on this planet. And Biopod on this planet is actually, a solution for 6 of the Sustainable Development Goals that have been, put together by United Nations. And it's and I think we need more of those technology that are coming from space, whether it's watering, water treatment unit. This is reverse osmosis, and NASA has been doing that for like many, many years. It's a spin-off book, is an amazing book where you can find every year the application of space technology back on Earth. But again, space is a very good tool to help us project and actually find solution here. I will finish quickly because we're running out of time with on the terrestrial part. But on the market here, and actually our terrestrial customers are potential customer for space. So we work a lot with cosmetic, perfume, everything that is around using natural ingredients, so flowers and plants, whether it's for food or whether it's for perfume and creams or soap, they all need natural ingredients, and so it's a huge market. Among those, those are our top customers, and so we work lots with them, and they have a big interest as well in developing this type of in going in space. Why? Because in cosmetics, pharmaceutical, perfume, health, they usually use some specific molecules that are going inside plants. And what we know from the state of art is that when you send some plants in space, because of the environmental condition, and in this case, meaning more solar radiation, but most of it's the lack of gravity, it's triggering, it's a stress to the plant, and the plant will trigger the production of more of certain molecule. And so there is So that's why also in our roadmap, we're planning on sending a lot of crops into space, is because we want to see how it affects the production of specific molecule that can be used in pharmaceutical or in cosmetic industry. And I'll talk about which is the Madagascar periwinkle, which is a plant that is used to treat cancer because of two alkaloids it produces. And one of the experiments we've been working on for a future mission is actually to send this plant and to look at how microgravity is impacting the production of those alkaloids. But so all those terrestrial market is also a potential market to finance future space mission, and they're interested in testing and see how they can grow things in LEO, but also they would love to be part of the future lunar base and being one of the players that will grow plants in space. So it's very important to add those players into the space stream, because there's going to be the one. NASA cannot finance everything, so money also needs to come from customers, and those guys can be customers. So here we have 194 domain, which is a lot. We absolutely don't have the production capabilities now. We have 10 biopod in production and one installed. Those are some of the pictures of the team installing the biopod. Even when I share the pictures, but it's much bigger when you see it in real than when you actually look at the picture, even though you can see the scale, but by looking at the size of the people next to the biopod. But so we designed it so it can be assembled very quickly by a small team. So it's a team of 7 people who actually assembled it. And the first one, it took some weeks, but the next one's going to be more easier. That's some picture of the biopods right now. So it's been working for a month right now. Yeah, a little bit over a month. We actually we have tagetes, so it's marigold, so it's a flower. We actually are harvesting them right now. We did a whole cycle, in 1 month, a whole growth cycle of this plant. We have vanilla growing there. We have a Madagascar periwinkle is actually this flower that you can see here on the top, the little cute white flowers, and that's inside the biopod, so it's very purple because of the LEDs. So when you actually go inside, you have to wear goggles so you don't see, when you're inside, you don't feel like you're in a purple or a pink pink bubble. But yeah, so that's the system working now. So we have 12 different species growing in the biopod right now. Among them, 2 endangered species, 3 type of fruits, and the rest is for pharmaceutical and cosmetic application. So those are real pictures. So those are actually pictures from the unveil we did exactly 1 month ago. So 22nd of September was the unveil of Biopark to the general public. And so at the time we didn't let people enter. Now it's possible to come and visit. We have our customers coming every day. And so people were on top at the facility looking at the biopod. But those are real pictures. They look like sci-fi, but this is the reality. And the racks are moving. So here, when you look, this is a view from the top. You can see there are 4 rows, but the rows are moving. So when you go inside, you're actually moving 1 row from the other side, so you can circulate. And yeah, you can see some of the systems. So we kept a little bit of glass floor, so you will be able to see the system. Then if we need to change something like the filters for the water treatment, you can actually just open the trap. So it's very easy to access and to do some of the maintenance. And those are some of the plants we've been growing. So this is from the plant lab here, but you can see we have very successful been growing vanilla. So we have passion fruit here, vetiver, and more, and we're adding more and more. So now I want to jump, because I talked a lot about the terrestrial product, but I want to jump and tell you a little bit more about what we are planning in the coming years for our space program. So the second pod that we'll be developing, it's called the LEO pod. So we are not planning on doing the exterior structure. We'll leave that to an awesome company that are building those modules. What we are taking care of is inside. So it's the growing system inside, and how do we plug into the life support system of a station, but to bring the growing capabilities. Of course, there are still a lot of unknown, and there's still a lot of questions to answer, but it's very similar to a biopod. So it's it's a controlled environmental system where you have a bunch of sensors and a bunch of microcontrollers that are commanding the irrigation, commanding the lights, and also commanding the level of the atmospheric level. The good thing is that in a space station, the CO2 is very high, so you kind of like don't need really to enrich that CO2 level for plants to grow. But that's the first initial design of what we'll be working on for the coming years. That was actually the first render that we did at the time, and so we've been doing a couple of iteration on this design, but we will start more actively. We're not planning on building this, in the 2 coming years. It's more. I think it's more going to take us probably 4 years to get something up there. Well, we're also very dependent on other companies that are building station. We're not doing the module. We're really focusing on inside and how we can bring. So we call them space garden, but our vision for Leopard is 2 things. Is of course food production, but it's not designed to replace prepackaged food. It's designed to kind of help solving some of the the food gaps that NASA has identified. So it's going to be mostly microgreens and but specific one that are bringing more vitamins or more protein. And we also see Leopard more as bringing more capability to grow plants in LEO, so we can get more knowledge and more data. Because before we build a greenhouse on the moon, we really need to bring more, growing facility capability in space, because we actually lacking data. The second product we've been working on is actually a product we presented for the Deep Space Food Challenge that was launched by NASA, Nucleus. So we passed phase 1. Right now we're in phase 2, so we're working on the prototype, which will be installed at the Kennedy Space Center or Houston. We're still figuring this out, but before the end of this year. Very similar concept than Biopod, completely different form factor. This one is a combination of cubes inside which we control the same thing: humidity, temperature, light, a watering system. It's more of a passive watering system than the aeroponics that we're using on Earth.
Barbara, you have 15 minutes left. 15 minutes.
15 minutes left. Okay. And the way we envision Nucleus, so is really to be also deployed inside station, whether it's for in a station, but also it can be for long mission, long duration mission. And so the Nucleus, you can actually, this information is not confidential. You can find all of that on the NASA website regarding the Deep Space Food Challenge. But so it's designed to be a closed system. And so inside the Nucleus, we grow microgreens, mushrooms, and also insects. And the idea is really to leverage on what different plants or biological elements will require. So the mushroom, actually, they will need more oxygen, the plant will need more CO2, and the insect, they will deteriorate the non-edible parts that we have from the plant system, but also from the mushroom parts. So it's how we can recreate not a closed loop. It's not 100% closed loop, but it's a first step toward building, closed loop, a bioregenerative system where you actually leverage the biology and mix it with technology, so you can grow things that are fulfilling again, it's all goes back to the food gaps, and how can we provide astronauts in long duration mission with the nutrients that they will need over time, like vitamin C, like potassium, that are degrading over time for long mission during long travel. So we're super excited about this one. Been working and doing some tests, and we release the prototype very soon. The moon pod, so I don't have more renders to share with you guys today, but they will come soon. So as I said, we have a 5 years program, and hopefully within a few weeks we will be able to announce a strategic partnership with a big structure to adapt our system for lunar requirements. There's going to be a lot of design iteration between this version of the moon pod and what we want to do we want to do in the future. But most importantly, I think the first innovation will be in the membrane. We're probably going to reduce the amount of composite that we are using and working more towards having something that really adapts and fits inside the rockets. The deployability, we want to keep it with the inflatable parts, but the membrane is going to be very different. So as I said, we have only 2 layers on Earth. For space, it's going to be, for lunar, it's going to be at least at a minimum, I think, a 4-layer ETFE with one of the design that I really like, but we need to prove its feasibilities, is going to be so 2 layers of cushion instead of just one like we have on Earth, and 1 layer with air and the other layer with water, because water is really good to protect against solar radiation. And so that's one option. Of course, those spots that will not survive is that, if there is an asteroid that is just like crashing on them, and they're not designed they're not designed to support that. But we'll try to put them in in an area where there is less risk for them to be destroyed. I'm joking here because we're having a very often this question, like, are there asteroid proof? No, they're not, and there won't be. But so a lot of work on the membrane, on the watering system as well. The gravity is different than the gravity on Earth. So we will need to test. So we have experiments coming in 12 months-ish. So we need to test different watering system. It can be more active than the one that we have in the space station because the space station is like no gravity, so very passive. Or at least it's our approach. So no aeroponics and nothing like that. But for the moon, we don't know exactly what we will use. Not the aeroponics one that we designed for us, that's for sure. So adaptation of the membrane, adaptation of the watering system, and of course adaptation on how you control the atmosphere. This one will not take atmosphere from the lunar environment because there is not. So there is a certain volume of air that we will need at the beginning to inflate the structure, and the question is going to be, how do we maintain the equilibrium? The good thing is that we need CO2 and we produce oxygen. So a pod can be a very good source of oxygen for the crew, and we can take the CO2 that human exhale to actually boost the production of the biopod. So all of this integration will be studied by the team in the 5 coming years. And the design will change, but we will try to keep it very very similar to our first vision. I think it's important. I mean, at least this is our goal, is also trying to bring a new design to space products and to take design and you don't have to find the biopod beautiful. I mean, I think it is, but everyone has different tastes. But I think it's important to keep the design because it's also what's helped people getting vision for the future and projecting themselves. Of course, you have to be realistic and to be able to manufacture it, but it has to be very. I'm trying to be very. So I'm part of what is called the solar punk group, which is a very utopian vision of the future where technology and design actually fits with nature and help human live in harmony with their environments. So it's like the complete opposite of cyberpunk. Cyberpunk is very like VR and very dark. Solar punk is very solar and green and technology. And so we keep this design vision in also the product we're designing for space. The partners, but I think it's maybe less of our discussion interest here. But so, of course, NASA, the European Space Agency, we work a lot with Dassault system in simulation. So we simulate our product and I mean, we build them using a powerful software tool. Then we do a lot of simulation, so airflow and temperature, humidity, and inflation. And we do that, and it's allowing us to skip the step of building a lot of hardware prototypes. For the biopod, for instance, we did greenhouse prototype, but just for the software and the sensors and knowing how to grow the plants. We never did a mini version of the biopod because it's complex and it will require so much work compared to just running simulation, which is also hard, but makes it easier actually. And we backed by great investors. We're a startup. We went to 2 space incubation program. One is called Starburst. Another one is called Space Founders. And yeah, multiple prizes. NASA Deep Space Food Challenge, some startup pitch. But also it has been recognized in 2021 as a breakthrough in agtech. So we're like the most efficient, we number one for greenhouse production system. We have a lot of media, and it's a lot of people see a lot of my face because I'm the solo founder, but it's actually a teamwork. And so the credit goes back to, the team. There is a cool documentary on Netflix. It's a show called The Future Of, and the episode 5 is about space tourism. And so we talk a bit more about the deployability and the inflatable and why and I'm not, there are a bunch of different people talking about why inflatables are going to be the future, at least the first type of structure we'll put on the moon. There is Anousheh Ansari, CEO of X Prize and former astronaut, who's also giving our opinion on that and more NASA people. But yeah, and so that's us. That's the team here. Very young, but very, very, very passionate. We might look younger than actually the age we have. But thank you for your intention. I think I'm on time, and we might have sometimes for question, guys, if you want.
All right, great talk. Thank you so much, Barbara.
My pleasure.
We have several questions here, so I'm going to go ahead and just start from the beginning and see how many we get through. And if we don't, I can ask some more questions if you're able to stay on later, although that will be like 10 o'clock your time. So I'll get through as many as I can. So the first question was, what level of interaction is there between crops in Biopod? So is it like independent planting areas where they're not really interacting at all, or are there more interactions with co-planted crops? And then also, do you intend to facilitate closed ecological systems in later versions? I think I heard you talk about adding some more closure processes like decomposition into the other unit, but will that be incorporated into Biopod?
Yeah. Okay, very good question. So right now, the growing system we're using in the terrestrial biopod is based on aeroponics. So it's not hydro, it's aero. So we really mist very thin droplets of water mixed with nutrient solution, and there is a drainage system. And so the plants, when we do the plant selection, if you do like a pluriculture biopod, you can do monoculture, then you don't have you have the question after on the microorganism. But if we select different plants, we first make sure they like each other, because some plants don't like each other and they don't do well they don't do very well next to each other. So that's the first thing. But then we're not right now at the stage where we're making them collaborate. That said, when it comes to recreating ecological system, we are using insects and microorganisms to trigger to get rid of pathogen, to create the equilibrium with some of the microalgae that you can find. So we put bacteria that are taking place, so we don't have bacteria. So we play a little bit with that, with the microorganism, where it's in like R&D phase, and we run a lot of tests. And we are actually working also with insects, and especially when there is pollination involved. And so we work with bumblebees. So that's the state of art where we are right now. Next step for future version, we want to see how, we like aeroponics because it's pathogen. It's easier to get rid of pathogen with aeroponics and not to have pathogen. And also biopod is pathogen-free environments. But we want to run experiment with soil because there is so much interaction. So we need to understand a little bit more as well. But that's going to be more on the experimental level versus the solution we sell to customers going to be very aeroponics right now.
And what was the name for the Deep Space Food Challenge cube-shaped
Nucleus.
Nucleus. Okay, so the next question I think was probably answered by that, but Laura is asking how a biopod would handle inedible plant waste for nutrient recovery. So it sounds like Nucleus is heading in that direction.
Exactly. Nucleus is heading in that direction. So the first thing regarding the inedible part of the plant, for nucleus, the plant we selected, they have a very low ratio of inedible, edible, ratio. So we don't have that much to recycle. But the part that we have, we actually using the insects to eat it and to turn it. It's not completely closed loop, but it's heading toward this direction. So in the way we select plants, we also try to select plants that don't have a lot of inedible part. Like we don't like potatoes, but we like carrots. That's what I often say.
Couple of like more engineering related questions. One is about how to block cosmic radiation on the moon, particularly the moon. Mars probably not as much of a problem. And then another related to leakage. I understand that the design for the lunar environments for the enclosure is probably underway and not complete, but someone was asking about what kind of leakage rate biopod might have or how sealed it is. So sealing and cosmic radiation.
Okay, cool. So cosmic radiation, I don't have the answer right now, because we need to test a lot of different approach. One of the things we were thinking of, as I said, was this multi-layer membrane with the air cushion, water cushion, but also aerogel layer. So that's one of our that was one of our approach. In the coming years, we'll be more able to say that, but that's one case. Then we have different options. We'll test. Yeah, that's everything I can do about this topic right now, I can say.
Let's see. Another question was regarding the configuration of the growing space. So it seems like it's probably at least the nucleus is probably pretty efficient, but are there opinions on what you think the most efficient growing configurations are?
Sure. So currently the biopod is we have good optimization. So the growing area can be up to 2 times the floor. So the surface footprint of the biopod. Still, we will do much better on the next version. Now we can see it in real. We can see so much space that we can actually fill up. The shape of the biopod is actually pretty good compared to I've been asked the question of the shape of the biopod, but multiple reasons, because we do inflatable, it has to be a rounded or ellipse structure. And when you do circle, it's very hard to optimize growing space because you lose a lot on the angles. So on the ellipse one, it's great. The only thing is that because it's a little bit rounded, so you lose a bit of space. But we're coming with a second design iteration actually to optimize the space. And nucleus is much more efficient in terms of space than biopod so far. Yeah.
All right, thank you so much. This is all the time for questions that we have right now before our next presenter. But I would like to direct you, Barbara, to their Q&A box on Zoom. If you have a little while, feel free to answer some of those questions that were directed to you and you can type. Or if you're able to stay on for the rest of the duration, our last segment will be a brainstorming section where we can talk a little more.
Okay, cool. I'll try to make it. I'm not guaranteeing 100%, but at least I will answer to question on the Q&A.
Thank you very much.
Thank you, Chris. Thank you so much.
Okay, our next speaker. I'm very looking forward to this presentation from Cyrille Przybyla from excuse me, my voice, from the French National Institute for Ocean Science, and he's going to be presenting about the Lunar Hatch program. Are you able to share your screen, Cyrille?
Can you see my screen?
Yep, we can see.
Okay, perfect. Hi, everybody. My name is Cyrille Przybyla. I'm very excited to be here with you for the first ever Space Ecology Workshop. I'm sorry for the automatic translator. My English is not very good, and I have an accent from Europe. Sorry, Jane. I'm a marine biologist from the French Institute of Ocean Sciences. Firstly, I would like to thank the first Space Ecology Workshop team for inviting me, and I'm very proud to present to you the idea of space aquaculture. in the first time, and in the second time, I will introduce you the Lunar Hatch program.
We need to do a slideshow there,
Okay,
or we can see all your presenting notes.
it's a problem of slide. I don't understand. It's okay.
We can see your notes and
Okay.
There we go. Yep, that's right.
Okay. Okay, because I have note. Okay. Before we start, I would like to talk about my research center. It's a governmental institute with several research expertise, such as deep sea exploration, fisheries stock assessment, marine bioresources, and my particular field of research is the aquaculture in closed system. I've been intensively working on the marine fish breeding in closed system, and also controlled marine microalgae cultivation in a natural pond and fish nutrition. Our lab is located in the south of France, between Marseille and Barcelona in Spain, and between a natural lagoon, brackish water, and the Mediterranean Sea. This is all this part. My mission, my roadmap is the sustainable aquaculture, my mission on Earth. Sustainable on one end for the Earth environment and the biodiversity preservation, and in the second end for the food production system. In this scope of works, I have studied for 20 years the recirculating aquaculture system for fish. This system solves the water needs and the water parameter control. However, this system still continues to produce dissolved and particular waste. I want to focus on the system, what we do today. We have in the system dissolved waste. This is the metabolism of the fish, especially you have ammonia. The problem is that ammonia is dangerous for the fish, and we have to convert it using nitrification, a very simple bioreactor with nitrosomonas and nitrobacter, to convert the ammonia in nitrate. On the solid part, most of the solid parts are fecal matter, and it's very concentrated, and it's depend of the activity of the fish. Now, what you want to do is to close the loop, to use, for example, worms or sea cucumber to the degradation of the fecal matter. These sea cucumber worms do a mineralization also of the matter. This matter go to the algae compartment, and also all the dissolved waste go in the algae compartment. This algae compartment can convert nitrogen, can catch the nitrogen in phosphorus part, convert the CO2 in oxygen, and the flow water, the treated water, go back to the RAS system. But what happened about the algae production biomass? You can remove it and put it out the system, but we choose to study the opportunity to use this biomass to include it in the formulation of the fish feed. And we try to close the loop completely. To sum up the goals of the IMTA, because this system is called IMTA for Integrated Multitrophic Aquaculture, the goals are reusing and saving water, converting molecules from the system into several edible organisms, producing a feed source for fish, for example, worms or microalgae. The Food and Agriculture Organization, FAO, the part of the United Nations, is interested in IMTA because it could increase the global food security, and the impact on the environment is very limited. Now, what is the link with the space exploration and IMTA? In 2016, the European Space Agency published a call of idea for the European Moon Village. And I sent the idea to study the feasibility to build an integrated multitrophic aquaculture system in the most extreme environment with no B plan, the moon base. All the knowledge learned in this IMTA space program could be used for for the Earth application. As you know, it's not the cost of the meal which is the most expensive, but it's the cost of the transportation. In addition, if you think about an automatic cargo, a space cargo to the moon, every 4 or 6 months, we have to think about a partial food autonomy on the moon. There is already studies about agriculture on the moon, on ISS, in LEO. There have been studied, yes, these studies in LEO, lettuce with the NASA VG program, microalgae, and for the future BLSS, the Melissa pilot plant, and you already have a presentation on the biopod. But unfortunately, there are no animals included in this space agriculture at the moment. If we have to send a resident on the moon or on Mars, we have to select vegan astronaut. Why not? It could be the solution, but we can also think about something else. On Earth, human nutrition needs several interesting compounds provided by animal farming, such as EPA, DHA, omega-3, B12 vitamin. And fish can provide all these compounds for the human diet in space because it can store all this type of interesting compound. Compared to the other vertebrate on Earth, fish is the lowest oxygen consumer. As you know, in biology, one consumed oxygen mole produces 1 carbon dioxide mole. And compared to the other animals, fish is also the lowest CO2 producer. This is important because the future space IMTA oxygen could be produced by the microalgae. Aquaculture species such as shrimp, carp, trout, dorado have the lower FCR compared to the other animal. FCR is that means how many quantity of food you need to provide to the organism to increase the weight of 1 kilogram. On this drawing, you can observe that the beef, for example, you need 8 kilograms of food if you want 1 kilogram of growth. And all the aquatic organisms are around 1.5. It's important because fish feed production will be provided directly on the moon by the future IMTA. It's a nonsense to send feed to the fish on the moon. Very interesting thing too, it's a Brazilian study. Fish skin, the non-edible part of the fish, could be used as a bandage for burns, for example, radiation or something else. Fish and man and human skin collagen are compatible. And when the wounded skin is healed, the fish scale just falls off. The psychology aspect has to be also investigated. Farming fish on the moon could be a benefit for the moon resident welfare. For example, on Earth, a study held at the London Aquarium showed that as soon as visitors came in the aquarium, the blood pressure and the heartbeat decreased significantly. Briefly, here is a timeline of the history of the fish in low orbit. The first fish in orbit was a salt marsh fish found in the area of the launch pad of Cape Canaveral. Main fish studied in LEO were aquarium fish, like such as zebrafish or danio. Main fish studied in LEO were used for medicine studies, but not for aquaculture. Now, what is the situation of the space aquaculture today? Earth aquaculture and space aquaculture share the same goals: to convert waste from an aquatic closed loop into an edible organism. On Earth, why on Earth? On Earth to preserve the environment, of course, and the aquatic biodiversity, and also to increase the global food security. I show you the first slide on the IMTA. If with the same amount of water, the same volume of water, you can cultivate more edible organism. On the moon, the goal is to valorize all molecules imported on site, because this site is poor. Mars also, but a little bit less, and to provide food to the space resident, the idea of the partial food autonomy. Now the research on IMTA are still ongoing, sorry, and hasn't been finalized yet in both cases. And if you want to find a solution for the moon base, we will definitely find one for our planet. But after that, before to think about the space IMTA on the moon, we have to answer to one question. Can we send safely an aquaculture fish to the moon? This is the question for the Lunar program. The Lunar program proposed to send fish embryo to the moon, not juvenile, not adult fish. Why? Why? Because there is an autonomy stage. Embryo is very autonomous. We don't need a human intervention during the manned or unmanned cargo. Because there is a limited organism rate, we don't need to feed it. We don't need also to change the water because there are low gas exchange in it. Because there is all metabolites stored in chorion, water quality is easy to maintain. There is a limited space requirement for example, in a glass of 200 millimeter, you can have in it 200 small future fish. And of course, the payload is limited too. Now, let me see you what happened during the moon journey on the development of the embryo. In this stage, we are already on the journey, and it takes between 4 and 6 days. And at the end, we expect to arrive on the moon base at this stage, just before the hatching. Okay, now what are the questions for the Lunar program? We need to know which species to take on board for the lunar mission. We need to study the influence of the launcher vibration on the fish eggs integrity. We need also to know what is the influence of the hyper and microgravity on the fish embryogenesis. And also we have to study the secondary space radiation on the fish development. The first study was to select the fish candidate, fish aquaculture species candidate. On the left of the slide, you have the flight environment, like flight duration, radiation exposure, temperature variation during the journey to the moon, the launcher vibration. And on the right, you have the biological conditions. And we do an bibliography study with the knowledge we had for 40 years, and we have selected 20 aquaculture fish species already selected, and we have now to know which one is the best future fish or not. The first project of the program was LOVE project for the launch vibration on fish embryo. We want to know if the launch is a problem for the development of the embryo. The protocol was to expose a triplicate of 20 fish eggs to Soyuz vibration. Soyuz is the launcher which provides the worst vibration compares to the other rockets. And if the test succeed for this launcher, it will also succeed for the other, such as Ariane 6 or the SLS. In this study, the main parameters studied was the hatching rate compared to a control sample composed by sister and brothers fish, also in triplicate. I have a short movie to show you. This is the experiment. You can imagine that here you are on close to the launch pad, and we have several thousand of eggs, and we select the eggs to compose the sample. After that, we go to the institute to the simulation of the launch. We can imagine it's the rocket. We install on it accelerometer because we want to have the curves of the vibration, and then we start to test it, to test the Soyuz vibration on the fish eggs. You can see that it's a small tank, 200 millimeter almost, and this tank is inside a CubeSat. It's the model we choose. It's the CubeSat size. You can see the eggs, the fish eggs, in the window, and we stop the vibration. You can see the eggs, and we go back to the lab. And the experiment duration was 10 minutes because this is the time to be in orbit, 10 minutes. With the first minute are the vibration from the motors, and the other minutes it's the vibration from the air. And we put an incubator, all the experiment. We transfer eggs. You can see the small eggs after the simulation, and now you have larvae. After that, we take the sample and we count the number of larvae we have. The results show that there is no significant difference in the hatching rate between Soyuz samples and the control samples. We did the experiment at the third and at the two-thirds of the embryo development, and we also found no significant difference for the hatching rate. This fish stood this test. We did the same test with another species called migre. It's an aquaculture species. This species also stood the test, but it wasn't qualified as fish or not because its incubation time is shorter than the moon journey. That means that the hatching will occur during the journey. Now the gravity project for the study of different gravity. I can show you also a small video. This study is on hypergravity. It's the acceleration of the rocket. The ESA offered us the access to the ground-based facility dedicated to experiment with aquatic organisms. And the main parameter studied was also the hatching rate, but also the fish stressed post-hatching. Okay, we come from the 1G to 5G during 10 minutes. For the microgravity, it was the same facility. The experiment was carried out at the ESA facility using a random positioning machine, which imitated the microgravity. The eggs were exposed 2 days before the hatching. I cannot give more detail about the stress result because the analysis is still ongoing. But concerning the hatching rate, it was the same than the control for the both. Now, CORAFE project. The aim of the project is to study the influence of secondary cosmic radiation, neutron and proton. But today, this year, we focus on neutron. The experiment was carried out at the French Nuclear Center at Cadarache in France, using a particular accelerator which produces neutron. We focus on neutron in this experiment. The neutron dose exposure during the journée was calculated by the French Space Agency and the French Institute for the Radio Protection. Last month, we virtually succeeded to succeeded in making the journey to the moon in terms of neutron exposure. We will study the larvae DNA damage, the embryo stress, and the larvae integrity 10 days after hatching. Now, what is the future? We are just here. Next year, we submit a project for the proton study. Proton is a primary radiation, but also secondary vibration inside the vehicle. And we expect to add all the parameters together for a ground-based experiment to add vibration, gravity, and radiation. After that, we will be ready for a LEO experiment. And why not to a moon mission experiment. And in the coming years, we expect to design the Earth IMTA with algae, fish, and worms. And in 10 years, we have some discussion with HISA for be boarded in the EL3, the European Large Logistic Lander, for a biomission on the moon. Now it's my last slide. Why I believe in the space ecology? I think that we are nothing on Earth without our environment, and I think we have to think a large ecology, not only with vegetables, photosynthetic organisms, or bacteria. It's all the organisms are important, and the Biosphere 2 experiment is very inspiring for me, and we have to continue in this direction. I think that human presence outside Earth will be sustainable only if we export a balanced biosphere and in the respect of the ecological principle. Well, thank you for your attention, and at last, I would like to thank the team for the organization of this first Space Ecology Conference. If you have any question, please ask me slowly.
Thank you, Cyrille. This was a wonderful presentation and a different aspect, a different component of our ecological systems that we don't often talk about for space. So very excited to have you here. We have several questions from our audience. The first one is, do you anticipate freshwater or saltwater being most useful in microgravity aquaculture environment? Are there differences beyond species?
Okay, it's a very interesting question because at the moment we have select only 20 species. We don't know exactly the water quality on the moon and what type of profile of water. And in this short list, we have freshwater fish and marine fish. And we have also hot water fish and cold water fish. We have also flatfish and I say normal fish. We have different because in aquaculture there is a large biodiversity of species, and I'm sure that we will find the good species for the type of water we will have. It could be water from ice melt from lunar environment, but it could be also water from water waste treatment. We don't know for the moment.
Let's see. If you could bring a fish to space with you, not to eat, but to keep, what variety would you choose for your pet fish?
Not to eat, but to keep.
Yeah.
I don't know. All the fish are interesting. Yes. My job is to keep the fish alive.
Let's see. The next question was referencing the slides showing the waste to warmed algae flow charts. Not
Yes.
sure which slide that was.
First one? at the beginning?
Yeah, I think it was a flow chart. Yes. Yeah, I think that was the one. Have you looked at including copepods, rotifers in the system? Was the first question. There's a second part as well.
Okay, sorry, including what?
Oh, sorry. Copepods? I'm not sure if I'm pronouncing
Copepods.
that.
Yes.
Or
of course.
rotifers?
Yes, it's a very good. Yes, it's a very good question. Yes, it's not. This system is a concept. It's an agile concept. We can add a compartment, but we can also link all the compartments to the others. Of course, we can add a copepod. Of course, we can have also shrimps to treat the solid waste. And this drawing is just the concept, and the future allows us to imagine different compartments with different organisms.
Let's see. Nathan was also asking about the algae feed quality, so compatibility with the fish. Is there anything that needs to be added to algae to balance it for the fish, or can they just eat it directly?
No, we have to process it before. We have to process it. I used the algae cultivated on fish effluent. It was one of my research, and we have to process it, but it's a mix of different things, because you can use also worms, but you can also use the leaves of tomatoes, for example, that you don't eat. You can use different things, and you can. I have published about the topic to feed fish with moisture feed. You know, in the industry, it's a pellet. It's like for the pet, you know, but it's better to feed the fish with a moist feed. And we can imagine, like a fisherman, to have a kind of bowl with different compound, with algae mixed with worms, with something like this, and you can send it in water, and the fish can eat it easily.
Okay. Well, that will be interesting to see if we can develop the means of producing the fish food as well. Let's see. Another person has asked about how to prevent cannibalization of the young. And I was wondering also about reproduction. Do you capture and separate eggs into a separate hatching chamber? And are there microgravity complications?
Okay, there is two quick questions, I think. The cannibalism and the stage of the culture. Okay, this is right. Okay, for the cannibalism, it's very easy to fix. It's because we already do in the industry. You need to select fish with the same size, and we have no cannibalism in the most species. It depends on the species, but if you have cannibalism, even you have the same size, maybe it's not the good species to select to the moon mission of aquaculture. Concerning the sequence of the generation, the idea is to send one generation hatching on the moon, and 4 months later you have a new generation, and you have all different generation. When the first one is eaten, the second one is coming. And of course, in this system, the beginning is very long, but you can think about what we call an e-farm. That means a farm remotely from the Earth. You don't need to have an astronaut on the moon. Other thing automatically, feeding fish and the water treatment and water management. You can do it from Earth. And when the first generation of resident come, you have already the good size of the fish. I don't know if it's clear. You can imagine different generation.
That makes sense. How about the, propagation or reproductive processes? So egg production and hatching. Okay. How does all that work, and would microgravity be a problem?
Okay, for the moon case, we don't need to have breeders. We don't need to it's moon is very close. It's almost 4 days for the journey, between four days and 6 days for the journey to the moon. And we don't need to have breeders and reproducer on the moon, because it's very easy to have a reproducer close to the launch pad. For the Mars case, it's completely different. We have to think about this aspect, because on Mars it's 8 months almost for the trip, and we cannot send a cargo all the time. We have to a reflection of how could be the breeding and the fertilization and the parents to cultivate parents. I don't know if it's clear.
Mm-hmm.
Yeah. On the moon, we don't think about to have genitors. Okay, to have parents, fish parents on the moon. We don't think about it. We just send eggs from Earth because moon is very close. Okay, but for Mars, we need to study what is the fish reproduction on Mars. Yes.
Mm-hmm. Okay. Question from Frida. Mike. Yep.
We did studies back in the 1950s using tilapia, which were able to filter out microscopic algae. And the poor fish are really starving to death, because although we were feeding them 10 to the 6, 10 to the 7 cells per ml, each cell weighed like 10 to the minus 12 grams dry weight. And so we had to supplement with additional food. So there are fish that can take algae out of the water, but that was not adequate to support fish growth in our experiments.
Okay, I've read this publication. I think it's Gonzalez now, isn't it? The publication?
Gonzalez?
It's in my list, and I will send you the actual publications. Also, I don't know how many people have seen the videos of the first vertebrate mating in space, which of course were fish. That was a Japanese.
Yes. It's Dr. Shabani, Dr. Shabani.
Yeah, thank you.
Well, if anyone finds that video, perhaps
I have it at home.
Okay.
Yes.
But anyway, if it's available online, perhaps during our brainstorming session, we can show that. I would love to see it. Let's see. Another question here is about how many trophic levels would you consider? I think the question is, would you consider adding higher order plants to the system? You did mention adding shrimp is possible. A related question is introducing duckweed, which is, of course, Davy Souza asked that question, which is what I wanted to ask as well. And we thought about duckweed plants or other higher plants.
Okay, the IMTA, Integrated Multitrophic Aquaculture, run from the bacteria to the vertebrate. And we can add cell, algae cell, we can add worms, and I think we have to consider all the level trophic from the bacteria to the fish. But the question is the link with aquaponics, I think, no? Yes, of course, it could be interesting. It depends the quality of the water. Is the water compatible with the vegetable or photosynthetic organism? The key is the water quality. It's the same on Earth. The key is water quality, and of course energy, because this kind of system costs energy to the water circulation.
Well, partially in answer to Davy's question, at Space Lab, we're working on a duckweed production system for microgravity. So it would be nice perhaps to work with Cyrille on how to incorporate those aquatic plants into a production process. Let's see. Mr. Porterfield is asking what the biggest hardware challenges you see for environmental control. Are they related to maybe aeration, gas exchange? Are there other hardware challenges, technological challenges?
Technological challenges for me is to find to find a way to do this closed loop with the less energy as possible. That's mean gas injection or recovery. That's mean a pump on the moon. I don't know if the energy for water pump on the moon is the same than on Earth. I don't know. It could be R&D direction to try to do the most interesting system with the low energy consumption. But it's also a challenge for Earth. It's the same for Earth.
Okay, let's see. There's a question about aerobic organisms needing, of course, oxygen. And do you see problems with gas phases, gas separation, and microgravity? Let's see, there's more to this question. Okay, separation of not only gases, but solids, separation processes, I guess. Do you see a challenge with that?
In a total closed loop with water, you don't need to separate CO2 and oxygen, because in the turn of the loop, the CO2 and the oxygen is already dissolved. You don't have to separate. What we have to do is to collect particles and fecal materials. We have to collect it, but we don't separate it from the water because we use aquatic organism to the degradation. I'm clear or not?
Oh,
I don't need to separate. We don't need to separate in this case because everything is aquatic, and it's easy to do. That's what we do in my lab. The water is have a high CO2 concentration and go directly in the algae system, and we never had to strip the CO2 or the oxygen.
Okay. One question that I've been wondering is, with plant production systems, we're very worried about volume efficiency, of course. How much mass can we produce in the smallest space possible? And so I wonder about aquaculture as well. Are there some species that have a higher mass to water volume that they can tolerate, so that you would need the minimal water possible?
Yeah. It will depend on the species, but almost the fish is almost the same altogether. It depends on the feed ratio and the water quality. I think we have to disconnect the aquaculture industry from Earth to the space aquaculture, because on Earth we have to make money. We need to have a quick growth to have money. On the moon, it could be different. We need to have a fish alive at the edible size. We don't have to speed the growth with artificial things or something like this.
We have 5 minutes left. 5 minutes left.
Thank you, Sherry. I was also wondering about the embryos. That was really fascinating to me to see the vibration testing for embryos and the radiation testing. Other conditions you would need to maintain for transit, is there a wide temperature range or is it a very small temperature range? And the other question is microbial contamination a concern that you have to worry about?
Yes. Yes, there are two questions again.
Sorry, I keep doing that.
The condition in the species, the European sea bass, we need to keep the water between, I'm sorry, it's in degrees Celsius, between 30 degrees Celsius and 18. But different other species can have tolerance to another range. After that, we don't need to have light during the embryogenesis. It could be in the dark. It's not a problem. It's better. It's better to be in the dark. And yes, we are for the moment we haven't the technology to freeze the embryo. We don't know how to do. There are some research on it, but it's not in an industrial scale. Because if you send on the moon bacteria, you can use the dormants of the bacteria. If you send seed, you can use also the dormants and wake up the seed when you are on the moon. But concerning the vertebrate, it's complicated to freeze and wake up after on the moon. And sorry, the second question was?
Oh, I think you answered it. The other question I had was whether, microbial contamination is a concern.
We do the first experiment with the vibration, the Soyuz vibration. We test arsenic eggs versus eggs without treatment. We can treat the chorion of the eggs in the aim to avoid to have bacteria from the mother. And we found that there are no significant difference between the two conditions. We were afraid to have bacteria because bacteria consume oxygen. It could be a competitor with the eggs in the small tank. And but at the end, the bacteria activity is very low, and there are no impact on embryogenesis of the fish in the completely closed system.
I just have one final question for you from Joya Massa, where you were talking about similar sized fish being together. If we were harvesting continuously over a long time period, could you also operate with decreasing numbers of larger fish as you start harvesting? Would you start allowing
Yes, of course. Yes.
larger fish in smaller numbers and allowing them to
Yes, what we know is that, for example, you have 100 fish with the same size. We had a publication about this topic. Fish are not stressed. They are 100, they are all together, it's okay. The stress occurs when the fish are 10 or something like this. But you have a relation between the volume of the water and the fish. That means you have a relation between the density. If you decrease the volume of water, you keep a good stress. It's not a good stress. You keep the fish safe. But we have already studied this aspect.
So keeping density,
yes,
volume density considerations. Okay.
Yes.
Nice.
Yes.
All right. Well, that's all that we have time for right now. Thank you very much for a great presentation.
Thank you.
And our next speaker, we have Joel Cuello, who's a professor of biosystems engineering at the University of Arizona. And he's going to be talking about, oh, my apologies, Joel, I didn't update the title in the program.
That's all right.
You can tell us the title of your talk today. Go ahead and share your screen. And thank you for joining us.
All right. Shall I start now?
Oh, absolutely. Go for it. Looks like you can see your screen. Looks great.
Yes. So thank you very much. And thank you, Christine. Thank you, Patrick, and for the entire organizing committee for organizing this workshop. I'm happy to be here. So the title of my presentation is Going Subterranean, a strategy to a more resilient food production on the moon and Mars through vertical farming and the new food systems. So as my title indicates, I'm going to have two parts here. First, I'm going to talk about vertical farming, and then I'm going to switch to the new food systems, which of course I will be defining what it refers to. As Christine indicated in her introduction this morning, there is this renewed momentum right now in terms of space exploration, and not just in general, but really deploying crewed missions out there on the moon, Mars, And of course, in the future, we hope beyond those two bodies as well. And these are just some of the most active aeronautic companies that are very much involved in the development of new innovations and technologies to accomplish those missions. Of course, in cooperation with NASA, as well as other international space agencies. So this is an exciting time for space exploration, particularly on crewed missions. Now, of course, when we're talking about long duration crewed missions, either on the moon or Mars, and we can set aside interstellar explorations for now, there is a need for a lot of resources, and one of which, of course, is food, because we need to sustain life, including the life of astronauts who will be going boldly to those extraterrestrial environments. And as you know, the preceding discussions emphasized, this will have to take place within the framework, within the context of a circular bioeconomy scheme or a bioregenerative space life support. And as have been reviewed by a number of the preceding speakers, particularly Ray Wheeler, he kindly reviewed for us what's been done so far in terms of crop production in a space environment. And of course, most of them, or probably all of them at this time, have taken place at the International Space Station in terms of zero gravity or microgravity. And he cited the veggie and also the advanced plant habitat on the American side, and of course on the European and the Chinese, the Russian and other nations side, they have their own innovative crop production systems or growing systems. But anyhow, the goal really is to go beyond low Earth orbit and to go to the moon and to Mars. And this is just kind of refreshing your memory in terms of the relative distance of the Earth and Mars with respect to the sun. The distance of the Earth from the sun is 1 astronomical unit, and Mars is about 1.52 astronomical units. And so that has significant impact or consequences on solar radiation on the surface of both planets, as well as the amount of gravity as well. So my presentation applies to both the moon and Mars, but I'm going to focus more on Mars to avoid confusion. So it's challenging to grow anything or to preserve or maintain life on the surface of Mars because, number one, there's no available running water. The land is non-arable. You have extreme environmental conditions in terms of extreme swings in temperature between night and day. You've got deadly ionizing radiation that's bombarding the surface. You've got micrometeorites, you've got meteors. And then, of course, for Mars, you have dust storms. So dust is a critical and a crucial component of the Martian atmosphere. And depending on whether it's cooled or heated, it plays a key role in the circulation of the atmosphere around the planet. And that includes formation of dust storms. Now, dust storms on Mars could be local, it could be regional, or it could be planet encircling, as these dramatic photos here taken by Hubble telescope in 2001 shows us. So this was June 26, and that's pretty normal. And this is what it looks like when there is a planet encircling dust storm on Mars. And dust storms, particularly the planetary ones, the global ones, are more active during the fall and winter seasons on Mars. Okay, so considering all of those harsh environmental factors that are quite deadly to us, to living organisms, it really is a challenge to build a structure. And of course, it has to be a closed structure, has to be a controlled environment structure, it has to be a protected structure to be able to maintain life in that kind of harsh extraterrestrial environment. And certainly the first need for that would be having materials, structures that could withstand, the constant pressures applied by those environmental factors, from temperature to radiation to dust storms. So the question is, do we have those materials? And I'm sure that we do, and more materials are being developed. So I'm sure that, you know, something could be cobbled together to come up with something, a structure that would be resilient, but that's still a question mark, really. So again, I'm just highlighting here that we need advanced engineering materials and structures that could really withstand all of those environmental factor pressures. So this makes me or reminds me of this movie, which I thoroughly enjoyed, but of course, certain scenes really shocked me, like this greenhouse scene in the movie from The Martian back in 2015. So the first question I had is, well, what material was used actually to build this roof here that is supposed to be transparent to allow solar radiation to come through? Is that, you know, proof for UV radiation, for ionizing radiation? And by the look of it, probably not. And then there's this material here that's used as a curtain around the greenhouse. And at one point, it was actually flapping around and it's showing the exterior of the greenhouse, which is the outdoors in Mars. So of course, that's not possible because of the difference in atmospheric pressure and also the ionizing radiation. But this is a fictional, this is a movie. So I was willing to overlook that. And I still enjoy the movie. But that brings home the point of what I'm trying to say here, that building a protected and enclosed structure using the right materials in this harsh extraterrestrial environment is not an easy matter. And as one of the speakers earlier shared as well, we have to be mindful of the metrics that we have to constantly apply whenever we're designing a structure that is meant to sustain life in space on the Moon or Mars. So one is safety. The other one is efficiency for whatever task needs to be accomplished, in this case, food production. Crew time is extremely valuable. We don't want the crew to spend all of their time, for instance, tending to crops so that these crops can grow and that they could be fed. And then, of course, very critical are mass and energy concerns as well. So considering all of this, the solution that I and my collaborators, and in fact, a few of my grad students as well, came up with is the best way to go about this is to go subterranean. And that's either on the Moon or on Mars. And there was a precedent to this actually in my research experience when I was a young assistant professor at the University of Arizona. After I finished my postdoctoral fellowship at NASA Kennedy Space Center, I received this grant from NASA on a hybrid solar and electric lighting system. Actually, the first grant was not hybrid, it was just solar lighting system designed to supply available solar radiation for crop production that is meant to be located in a subterranean location. And I did this with my industry collaborator, Phil Sadler. This was around 1997. But then after that, I decided that really the best route is not to go pure solar, but to come up with a hybrid system involving solar and electric lighting system. To me, that's practical because it increases the resilience and reliability of the system. And at the same time, you're not 100% electric lighting. And so the demand for electric power that is intended for supplying the need of electric lamps is much reduced or significantly reduced. So again, this is just to remind you of the relative distance of the Earth and the relative distance of Mars from the Sun. And so there is significant amount of solar radiation that remains incident on the Martian surface, but of course, compared to that reaching the surface of the Earth, it's reduced. Of course, it varies. There is a range, but on average, it's about 43% of what's hitting the Earth. That's on top of the atmosphere of Mars. So 43% of what's reaching the top of the atmosphere of the Earth. So compared to the Earth, the solar radiation on the surface of Mars is much reduced. And again, that's a further rationale for going for a hybrid solar and electric lighting system. So we tried, various solar concentrators. This is a Japanese-made solar concentrator made of Fresnel lenses. And the brand name for this is Himawari, which is a Japanese word for sunflower because it has a tracker. So it automatically tracks the sun as it crosses the sky or the horizon. What we do is, what these Fresnel lenses do is it concentrates the solar radiation onto the tips of these fiber optic cables. And then the fiber optic cables transmit those concentrated solar radiation into the interior of this growth chamber, where we also have some polymer and fibrous fiber optics that then distribute the collected light over the canopy of the crops that we were growing at that time, including lettuce. We tried another solar concentrator. So instead of Fresnel, we also tried a mirror-based, parabolic in shape, solar concentrator. Again, the solar radiation hits the surface, it gets concentrated into the tip of this fiber optic cable, and then it gets transmitted into this chamber that is located in a subterranean crop facility. So in a way, this was really an introduction for me and my team in terms of the value of having a subterranean facility. But anyway, we tried light emitting diodes at that time. Light emitting diodes were already being commercialized. And this is just a comparison between, we tried other electric light sources as well, including xenon metal halide lamp, which is a white light. And this is the spectral distribution from about 400 to 700, which is the visible range, in comparison with an array of light that is made of red and blue LEDs, about 10% blue, 90% red. And this is the resulting spectral distribution. So one of my postdocs at that time, Ichi Ono and I, decided to do some calculations and simulations for using this hybrid solar and electric lighting system in the Martian environment. So we chose this site for the Viking Lander 1. So we were able to pinpoint that location on the planet, as well as the weather conditions or climatic conditions at that location. So that's the Chrysi Planitia. And so using our system at that time, so remember, this is around 1998, 1999. So technologies have advanced significantly today. So you would expect that the efficiencies that we have now are far greater than the efficiencies that we had then. So this is the solar irradiance collection, transmission, and distribution system. So that's the totality of the solar concentrator that we use, the particular cable optics that we used, and the length, the distance, and then the distribution system over the canopy of the crops we were growing in that controlled environment growth chamber. So these are the efficiencies that we obtained. And so if we had these efficiencies, these are then the energy savings that would be achieved if this system were used on the surface of Mars, supplying a photosynthetic photon flux of 200 micromoles per square meter per second for 16 hours. So this kind of lighting regime would be appropriate for salad type crops like lettuce, for instance. They'd be able to survive in that kind of lighting environment. And so at that time, we calculated that if we had a system that was 50% efficient, this is the amount of savings of energy that we would achieve for that crop production system on Mars. And that was about 42%. And of course, saving 42% of energy is quite significant. This is as opposed to using 100% electric lighting. And now again, I'm arguing that today with the advances in technologies and materials, I mean, certainly the efficiency that we would achieve is far greater than 50%, and therefore the energy savings we would achieve is far greater than 42%. So there is rationale then to go subterranean. And in this, it is within this context that my industry collaborators and I designed a vertical farming system or growing system that we call the boring vertical farm, which is really meant for space applications. Boring is exciting. Boring is just an acronym. It means the burrowing and regenerative in-ground vertical farm. And again, the boring vertical farm is meant for the moon, Mars, and beyond. I'd like to acknowledge my industry colleagues, Matteo Pietrabelli, who is a systems and structural engineer. Mackenzie McAleer, he's an expert on everything crypto, but at the same time, he developed a plant production system for space application that is soil-based, and I'll talk about that in a little bit. So as on Earth, on Mars, and on the moon, it's location, location, location. It really matters in terms of planning and strategizing to make systems for bioregenerative applications more resilient and even more sustainable. So the way to go about it is to take advantage of naturally occurring tunnels underneath the surface of the moon and Mars. And those are called lava tubes. And this is just a representation of what a lava tube is. So it's basically a tunnel, an empty void space underneath the surface. And sometimes there are breaks and they're referred to as skylights. All right, so lava tubes are formed when a lava flow, and of course, in the ancient times, there was active volcano on Mars as well as on the moon. And so when the upper portion or the top of the lava flow cools down, it then forms a crust. Whereas the lower portion, they remain molten and they continue flowing until, of course, the source of the lava gets exhausted. And that's how the lava tube is formed. Now, here on Earth, we have lava tubes. This is a picture of an actual lava tube. This is the Valentine Cave in Lava Beds National Monument in California. On the side, you can see these horizontal marks. They actually indicate the levels of the lava at certain periods of time that created this tube or underground tunnel. This one is in Hawaii, the Thurston Lava Tube at the Hawaii Volcanoes National Park, of course, in Hawaii. And you can see these horizontal marks as well. The longest lava tube on Earth is about 90 kilometers long. And again, this is kind of showing you the skylight in a lava tube. Of course, this one is on Earth. So this was a photograph that was taken by the European Space Agency Mars Express spacecraft in 2004 at the Pavonis Mons in the Tharsis Montes. So this is next to a Martian volcano. And these are lava tubes. And some are, have their roofs collapsed. So these are channels, really, not lava tubes, but the others are lava tubes. And this larger one is about 60 kilometers approximately in length. And the others are much longer. They're about 100 kilometers long. So they exist on Mars, in fact, in different places around the planet, on Mars, as well as on the moon. So the idea that we have for the boring vertical farm is that it's a tunneling type of vertical farm that is specifically situated in these lava tubes on the moon or on Mars. And there are advantages for doing that. Of course, it serves as and by the way, this is a graphic from the National Geographic magazine, where you have these surface infrastructures and facilities, but then you've got these habitats underneath in lava tubes. And so you can have a boring vertical farm there as well. So it makes sense to definitely seriously consider having these growing systems and even human habitats in a lava tube, because they serve as radiation shield or protection, dust storm shield and protection, and they're more thermally stable. So again, on the surface of Mars, in a Martian day or a lunar day on the moon, you can go from -Celsius to, hundreds of Celsius negative to higher temperature, but it's still negative. But it was estimated that in a Martian lava tube, it's more like -60 degrees Celsius, and it's pretty stable. So that is an environment that is much easier to handle than one that is wildly fluctuating and has a significant range of values. Okay, so for chamber configuration, of course, you can take a different type of shapes, whatever would fit in a particular situation, whatever is optimal. It can be single, you can arrange them in series or in parallel. And of course, you can have some wheels as well for motion, locomotion of these chambers. And the chamber material, of course, it's advisable that they should be made of light materials. So inflatable materials definitely are preferred. And inflatable materials now can be transported in a very compact manner or very small volume. And then once on the surface, it can be inflated. And then the material, which is originally soft, can be hardened using rigidizing methods. So this one is an example of a rigidized material as developed by Share a Space for their large integrated flexible environment chamber. And I like that acronym that they came up with, which is LIFE. So these are some of the materials that are being actively investigated and tested for materials for space applications. Now, in terms of growing systems, again, it's key that the crew time that is spent on growing food on Mars is minimized because there's more, their time is precious, and it would be better for their time to be used for some other equally crucial activities as well. And so with this, we are considering using a LifeGrow bot. This is just a hydroponic, or well, in this case, it must be aeroponic for it to work at microgravity. And it's sitting on top of a mover bot. And of course, it's possible for several of these LifeGrow bots to be sharing the same mover bot as well. And they could be arranged in particular arrangements or matrices so that the crop production system could be made very efficient, such as this automated circular operational protocol, where they go in circles. You have the germination room adjacent to the planting room, where the robots are planted with seedlings, and then they go to the growing room. Once they're mature, they go to the harvesting room, and then they get harvested, and then they recirculate. So there's a way to make this really efficient system. And this is just one possibility here. And again, this is the harvesting room, planting room, going in circles. But I kind of want to underscore one possibility here is that the growing room could actually divide it into two sections. The first one is designed so that the environmental conditions here are set and intended to really maximize the growth rate of the crops, so that by the time the LG bots reach the end of this section here, the plants are fully grown. And then they transfer to the other side of the room, and they go in the reverse direction. But in here, the environmental conditions are altered so that they are meant not for optimizing growth, because they're already full grown, but really to optimize their quality, their nutritional content, as well as their flavor profile. So by the time they reach the end here, they're ready for harvesting. And that definitely lends itself readily to data analytics and even artificial intelligence, because you can use sensors to monitor water requirement and pH and the light and the oxygen and so on and so forth. And then you can do an AI algorithm to really figure out what is the best combination of all of these levels so that you can achieve the maximum growth rate and the maximum quality, flavor profile, as well as nutritional content. Okay. And these are just some of the advantages. And I just want to acknowledge, this is a group of senior engineering students last semester. They worked a prototype for the LG ball, and they did that. And sorry, I don't have a picture here. The other one is a nutrient delivery system. Of course, it's got to work under microgravity condition. because, for instance, in the space station at low Earth orbit, there's zero or microgravity practically, and water is behaving very differently than it does here on Earth, because gravity is no longer the predominant force acting on the bulk water, but rather surface tension, and so the bulk water breaks up into these spherical water particles, and they float. And they tend to be stationary unless you apply force on them. So the typical hydroponic system here on Earth is not going to work well in that environment. And so we developed this intelligent aeroponic microgravity and Earth nutrient delivery system, or IMN system, where the way it works is you have these liquid emitters and air emitters. So the liquid emitters will fire essentially toward a root biomass, but then they'll break up into the spheres. And so you will need the air emitters to basically emit gusts of air toward these particles so that either they could break up or they can move forward to the next root biomass. And of course, we design various types of emitters. We have a single or individual liquid and air emitters, or they could be combined or compound. And you've got these rotational air emitters as well, which could be placed on the sides or on top, and they're essentially recirculating or moving. And again, that's targeted toward moving the particles. And again, I'd like to acknowledge my engineering students who worked on a prototype for this system. Then a special growing system here is a soil-based growing system called GeoGrow. And again, this is from my industry colleague, Mackenzie McAleer. In essence, it is a cylinder that is rotating. And there's a central axle, which really is a pressurized, water pressurized pipe inside. And it's got holes, or it's perforated, so the water will essentially flow into and diffuse into the soil that is contained with this within this cylindrical chamber. And then again, this chamber is turning, rotating, and the idea is that perhaps that could help even with the geotropic adaptation of the crops that are being grown. So it will be tested. And again, you can arrange them in various configurations that would be appropriate for whatever growing chamber is available on the Moon or Mars. In terms of chamber deployment, this is my industry colleague, Matteo Pietrabelli. One concept is, and this is for introducing this inflatable chamber inside a lava tube. So it could be initially folded, and then, of course, it has a way of moving, and then it will be moving toward a post, and then it will be secured to that post, and then it will move in the opposite direction, and then it will be unfolded, and then it can be rigidized. So these are just strategies for how to implement this in reality. And then there's a power supply system, as I mentioned, 2 decades ago. We worked on this hybrid solar and electric lighting system. But Matteo has already also designed this energy harvesting and storage system. In the case of the moon, of Mars, there is solar radiation available, and there's also wind. And so both sources of energy can be harnessed toward or for utilization in the boring vertical farm. Now, on the moon, of course, there is no wind on the surface of the moon, and so it will just be solar radiation on the moon. And
Joel, you have 15 minutes left. 15
yes,
minutes.
Thank you. Yeah. All right, and that one is for energy storage, but the details are shown here. I don't have the time to talk about it. All right, so now the second part is let's talk about alternative proteins. Because it's fine to grow crops on the moon and on Mars, and of course certain crops that could be grown are protein containing, like soybeans, but why not grow other forms of protein as well? And this is where the new food systems ideas come in. One large category of the new food systems is cellular agriculture, and one of which is cultivated meat. So this is real meat, except that on Earth, we grow animals, the whole animals, to grow them. And that results in intensification of agriculture, injecting them with antibiotics so that they don't get sick in the very confined and crowded environment. More than 75% of land use for food production goes to animal agriculture because you have to grow crops so that you can prepare the feed for the animals. And about a quarter of global greenhouse gas emissions are traceable to the food supply chain, and animal agriculture, unfortunately, accounts for about 3/4 of those emissions. So this is Mark Coase. He's a professor at Maastricht University, and he designed this, of course, not for space applications, but for Earth applications. So the idea is, instead of growing the entire animal, why not just take cells from the animals and grow the cells in cultures, in the lab, and in bioreactors in a natural setting. And these are some of the environmental savings that one achieves by adopting this method. You save on greenhouse gas emissions, on land use, on energy input. Now, of course, when this was first tried, of course, it was very expensive. It's about $18,000 to produce 1 pound of beef. That was 2013. Fast forward 2018, it's reduced $11 per pound. And it's now 4 years later, so you can expect that the price for a kilogram of beef produced in this manner is much lower now. And it was very expensive initially because of the required growing medium for growing these cells in culture. But now they came up with alternative media that are using lower cost ingredients. And of course, you could just grow them in bioreactors. So the world's first cultivated meat has been approved for commercial consumption and distribution as well. And this was in Singapore, and this was from a company, Eat Just, which actually is an American company based in San Francisco. This is Mark Coase. We met at a conference last year. The second one is fungal-based meat. So this is actually mushroom. And there's a company working on this called Mush Labs in Germany. So what we call mushroom is the fruiting body of this fungi. But in this method here, you don't grow the fruiting body, but you grow the fungi, the mycelium, and you can grow them in bioreactors. So this is fermentation. And these are the things that you essentially save on when you do it this way. Then you harvest the mycelia, and then you form them into your food products, such as a meatball. So it's not a real meat, it's made of mushroom. So I was invited to a science retreat in Hamburg last year, and that's where I met Mazen Brisk, who's the CEO and founder of Mush Labs. And finally, the algae-based meat, Sophie's Bionutrients, is a Singapore-based company. It's using algae to produce a burger patty. Of course, it's not made of meat, but of microalgae protein, as well as crab cakes. So I'm sure that there are already people working on cell cultures of actual fish to produce fish products or food. So anyway, chlorella is the algae that they're growing to produce the burger patty. And this has already been approved by the Singapore Food Agency. So it's legal in Singapore as well as in the European Union. So they grow the biomass in bioreactors, they harvest them, they dry them, they turn them into flour, and then they use extrusion methods to be able to produce their food products with the right texture. And of course, they work with the flavor as well as some other aspects. So in terms of nutrition, because this is made of chlorella, it's actually more nutritious than beef. It has 60 grams of patty, or each 60-gram patty delivers 24 grams of protein. It has all the 9 essential amino acids, including histidine and leucine, and it is high in B vitamins such as B12. And of course, I work on bioreactor design. So we have a whole suite of original bioreactors that we have developed for various types of cell cultures, including algae cells. And these have been patented by the University of Arizona, and these are perfect for these types of production. Anyway, I want to skip that. And also, I worked on bioreactors where we grow organ cultures. So instead of growing the entire plant, if your interests are just the roots, why not just grow the roots? Because in that case, your harvest index would be 100%, and you're going to save a lot of resources for growing the upper parts or the shoots of the plants that you're not going to be eating anyway. But of course, in a circular bioeconomy scheme, there's a way that you can make use of those shoots for recovering the nutrients for some other purposes as well. Anyway, I just want to mention Mackenzie and I, we have working with a great team as well, and we are working on sending an experimental version of the GeoGrow, which is soil-based, in the International Space Station, hopefully end of next year or beginning of 2024. All right, so I thank you. Thank you, Chris, again. And I'm happy to answer questions, of course.
Okay, great presentation.
Thank you.
Thanks, Joel. Oh, wow, a lot of different alternative food options that you're showing us there, which is great because we haven't heard much about those alternative sources today. So that was a great addition. Also, you've been very busy. You have a lot of projects going on, my goodness. I can feel the pain there. Let's see, starting, excuse me, with one question. Oh, back to the solar concentrators. Someone was asking about the efficiency. Is the efficiency of the concentrators limited only by surface area? Are there certain capacity considerations for fiber optics? Yeah, what are some of the factors that affect how efficient those solar concentrators are?
Yes. So the solar concentrator itself has its own efficiency. And in all honesty, these are decades ago. I haven't looked recently or read our papers, so I don't remember exactly. But I think the efficiency we were getting at that time was around 30% or 40%. Yes. That's in terms of the collection of the energy and concentrating it to the tip of the fiber optic cable. For the fiber optic cable, we use glass and we use polymer as well. Of course, glass has higher efficiency in terms of transmission of light compared with polymer. But with the advancement in material science now, I'm not going to be surprised if polymer cables, optical cables, are as efficient as glass now. Because even back then, it's like 90% or above 90%.
Yeah. The next question has to do with the whole aeroponics versus hydroponics aspect. So I understood from your presentation that aeroponics may be currently better suited for microgravity applications because hydroponics is still challenging for microgravity. But for Mars and Lunar, where you have partial gravity, do you still think that aeroponics is the better choice, or would you still choose it over hydroponics?
I would prefer, for Lunar and Martian, I still would prefer aeroponic because the gravity on the Moon and Mars are much reduced compared to that of the Earth. I'm sure water is still going to flow, but it's going to be in a weird way, if you will. The hydrodynamics are going to be quite different. And so it still needs external force or pressure to really get it to flow. So to be on the safe side, I would go with aeroponics. Okay, and then I guess I had another question related to that is what are any drawbacks that you see with aeroponics that it would be nice if hydroponics were to work, like would it overcome any challenges in the other direction? Is there any aeroponic challenges that are a problem? Oh, aeroponic challenges. Yeah, things that don't work as well for an aeroponic system in this environment. Practically speaking, I don't see any significant challenges with aeroponic systems, even as applied to extraterrestrial environments. No, I don't. And we have, really advanced emitters now, and they're smaller in size now. So there's really high flexibility now in terms of implementing this. Let's see. Oh, Joya is asking, she says, it seems like there's a lot of technologies under development that will impact the efficiency of the boring farm approaches, technologies that are being developed. Where do you see the lowest hanging fruit in those developments that'll have the biggest impacts on efficiency? On efficiency, you mean overall efficiency, I suppose? Yeah, I think any aspects of lower mass volume power, energy, crew time, all of those things, costs. Well, I would say Number one is the materials. I think that at this point in time, we pretty much have these advanced materials that are inflatable and then can be made rigid through rigidizing methods or processes. I think they're pretty much ready at this time, or at least we have some options that can actually be used for these applications, particularly that these are meant for below ground deployment or application. So there is significant radiation shield, there's significant thermal shield. And so the way that they're tested on the Earth, more than likely is going to hold for these environments because they're in a semi-protected setting. That's one. Then in terms of low-hanging fruits as well, again, we already have a lot of systems that have been developed in terms of production systems, whether it's hydroponics or whether they're aeroponics. So modifying them really is just a matter of making sure that they fit that particular structure that's going to be implemented. I don't think that's going to take a lot of design or a lot of modification or significant alterations. So the existing growing systems that we have, I think for the most part are considered applicable for these scenarios.
I had a question related back to the slide very close to the beginning about the light efficiency, the energy savings gained by using Martian sunlight, which I'm sure, of course, all those numbers change with more efficient LEDs now. But in that analysis, has your group conducted any analysis about the radiative heat loss and how that balances out with the energy savings from a transparent dome? And I asked that because we're also working on Mars greenhouse concepts, and I'm just wondering if there's any more data points.
Yeah, no, we certainly just consider this really for a subterranean situation. We were using exclusively solar concentrators for this.
Right, that's right.
Not using any transparent roof whereby radiation can penetrate and you could harness the sunlight that goes through.
I think we have about 2 minutes left. So maybe 2 more questions. Emily Matul is asking, how many of these alternative meats have you tried? And if you have, which one do you think tastes the best?
I'm sorry, alternative
meats? Yeah, alternative meats.
Let's see. Actually, none yet, honestly. So that's something that I would love to do. I mean, of course, there's the plant-based meat, which I didn't include here because the way they're produced is you have to use plant materials. And, if you're growing plant materials in space, might as well eat them directly rather than reconstitute and reform them into plant-based so-called meat. So I didn't include the Impossible and the Beyond Meat types of products here. But in reality, I haven't tasted yet the mushroom-based burger that is produced by that company, as well as the algae-based meat, as well as the the cellular meat, because they're not available here in the US yet.
Okay, and then one more question. Davey Souza was asking about microclimates in the robotic system and the rotating system, forgot the name. How is vapor pressure deficit dealt with? Are there considerations for airflow and all of that?
So we didn't look into that. However, we conceived the boring chamber to be either made of one whole chamber or could be subdivided into sub-chambers. And of course, there has to be environmental control implemented in each one of that sub-chamber or the entire chamber if it's going to be operated as one chamber. So definitely the calculations will have to be made in terms of how many LG bots are going to be accommodated by that chamber and what crops are going to be grown. And then of course their transpiration rate, and then what would be the resulting relative humidity, and what kind of controls will be made available to control the relative humidity, because that would affect the vapor pressure deficit.
Okay, I think that's all the questions we have time for right now. Joel, thank you very much.
Well, thank you very much.
And our next presentation, and actually final presentation for the day, we'll be hearing from Mr. John Hogan, who's the Bioengineering Branch Chief at NASA Ames Research Center, and he's going to talk to us today about the hybrid technical ecosystems for space life support. So thank you for coming. Thank you for being with us today, John.
My pleasure, Christine. Are you able to see yet?
Not yet.
Okay. Let's see here. Okay.
So when you go into the share, well, I don't know if your PowerPoint's started, but there's a little bottom right button to click share a final time. Sometimes I forget it.
Yeah. See, I'm not getting that. I think it has something to do with. Let me see if I move it down to a different screen if I get it. There we go. Okay, I needed a different.
There it is. We got you.
Let's see. Okay. Are you seeing my slides now?
We are. Just need to start the slideshow so we don't see all of your notes.
Oh. Okay.
There we go. Perfect.
Okay. Okay. All right. Thank you. Thank you for your patience here with Zoom. We in the government don't use Zoom very much, so I apologize. And I do want to thank Chris, Patrick, Freda, everybody who thought enough to pull this together. It has been a bit of a dry spell for biology in space, and it's very nice to see this upswell of interest and activity and to make this happen. So I really do appreciate that. And I appreciate the opportunity to talk with you. I am currently at NASA Ames Research Center. I am now a branch chief, but I've done a lot of work over the past 27 years in life support, bioregenerative life support, starting, as Ray Wheeler mentioned, a Rutgers University, it was called an NSCORT, and we had about 6.5 years learning about bioregenerative life support, and that got me pulled into this topic. I'm very interested. I'm an environmental scientist by training. So I have this sort of broad look that I'd like to share with you today, and then get into some details. My title is the Hybrid Technical Ecosystems for Space Life Support, and maybe hybrid technical is a little bit redundant, but every ecosystem that we're going to be using in space is obviously going to be technical in nature. But obviously, we're trying to support life, so it makes it hybrid. This is, one of the largest challenges that we're going to face. Besides learning how to live here on Earth very sustainably, trying to get off of Earth, which is difficult in the first place, and then go to these rather sparsely supportive environments. I mean, obviously, space itself is very difficult. But even when we get some gravity and a little bit of atmosphere on Mars or Moon, and we start to get some gravity and some resources, it is extremely difficult to sustain ourselves there for a long period of time. I also, Christine, wanted to define ecology because it is something that I studied quite a bit. And moving off the Earth, ecology has a very different feel for me. So ecology is a branch of science concerned with the interrelationships of organisms to their environment. And here we can see a crew member in ISS looking out the cupola, looking back at Earth, and I hear the crew spends a lot of time in the cupola, looking back at Earth, and we just get mesmerized and are truly affixed at just how wonderful and how beautiful their home planet is. And it's not really a surprise when we think about a baby being born. That baby is born for Earth and Earth circumstances. It's sort of an expectation of Earth. Lungs are formed in relation to the atmosphere that we have. The oxygen around 21%, inert nitrogen, carbon dioxide in certain concentrations, In the same way, they are also born in a way for psychological environments, for caring communities to be very supportive of them. This is, really important to understand when we're trying to leave home, is that, down to our DNA and our physical bodies and our 30 trillion cells, which have come together to learn how to live well on Earth, and now we're trying to leave, it becomes, a very difficult thing. So wherever we are, we need life support systems, right? We haven't found life growing yet in the vacuum of space, and we expect all life to require supportive conditions. So the Earth itself is, the most wonderful life support system for us humans and for life as we know it that we've ever found so far. And when we leave, we're trying to bring those life support functions with us, right? And very, and this suit is in a way a life support system, right? It allows this astronaut to float in space above that glowing blue planet below. And it's very temporary, right? This is not a long-term solution. This is so that they can go out and be mobile and do work outside. But they need to take care of certain things, even for this short, maybe 8-hour period, like pressure. They need, they need to create an atmospheric pressure inside. They need to supply water. There's even little food bars in there. Obviously, remove carbon dioxide, supply oxygen, keep the air and the humidity correct. The temperature, one side of the suit could be a couple of 100 degrees Celsius, and the back side of the suit could be minus a couple of 100 degrees Celsius. So thermal is very difficult. There is radiation protection at some level, at least UV. It's hard to do much with the cosmic radiation. They do their best with waste removal in that suit. Gravity. Now, there is no gravity in this particular circumstance, but they do need propulsion. So this is, sort of the fundamentals, the lowest level of life support that I think, people need, and it's for a short period of time. When we're trying to leave Earth and bring those conditions of Earth that would support humans long-term very well, for sustainably, for independence from Earth, we really need to understand Earth, right? This is our master life support system. Very intelligent, how the whole system's put together, and so I always enjoy being an environmental scientist, understanding Earth's life support systems and how they work, and very top level. We have what you could call the bones, so the rock that is underneath this, what we see, our biosphere. We are essentially the biosphere is a sheet of paper thickness on top of a basketball. So that's where we live. The rest of it is, creating gravity and radiation protection and redistributing minerals throughout the Earth with time. So we have this great resource diversity there's internal distribution and cycling. Our inner core, the nickel iron core that rotates, creates an electromagnetic sphere that keeps us from being stripped clean by the sun. We have this the gravitational field that allows life to evolve, though we know. The Earth has a stable rotation, which people believe was brought to us by the moon being formed, which was probably an impactor. Before that, it was too much of a wobble, and the seasons were so strong that it was likely that life wouldn't evolve. It has the proper light levels and proper atmospheric pressure, which is essentially our windshield, held on by gravity. So it's an amazing spaceship that's moving quite fast around the sun. On top of that is in this sheet of paper in our biosphere, we have a highly interactive and highly interdependent set of life support systems that are going on, that are given to us for free, essentially, by the Earth. These are natural processes going on long before we started setting foot on this planet. And in the Millennium Ecosystem Assessment, which was done at the turn of the millennium, brought together many scientists and identified what they considered to be, I think, 24 different ecosystem services, or you can think of these as life support systems, that are working on the Earth. Obviously, it's, you can debate the number and you can go into detail on a number of these. They did an assessment of how we're doing, as humans on this, and they found that essentially 16 of those are in strong decline. So we have a very difficult task in front of us of learning how to live sustainably here on such a remarkably resilient life support system. And it's, learning, and this is mainly because of our human life support systems that we have developed to maintain, done a lot of great things. However, to become in alignment with the Earth's life support systems, our human life support systems, need to match the needs of the Earth systems. So I'd like to just sort of briefly go over, so those of you who are familiar with Janine Benyus and biomimicry principles, these are, I always come back to these as really strong design drivers for me. These are essentially a list of different principles that the Earth uses to maintain itself. And nature optimizes rather than maximizes. It's extremely efficient. It runs on sunlight. You can debate that. There's a lot that runs on chemical energy underneath the ground. A lot of that was fed by the sun, but there is chemical energy. It uses only the energy materials it needs. It is extremely frugal. It fits the form to the function. When you look at a tree, it's an incredibly smart design. So are we. It recycles everything. It doesn't have a waste, right? It rewards cooperation. There's a tremendous amount of symbiosis. There's a tremendous amount of diversity, and it relies on that diversity for redundancy, much like we would rely on redundancy in our life support systems in space. And it demands local expertise. In other words, life and the system is adapted to the very local scenarios. Like, for instance, in redwood trees, they find species of ants that live nowhere else except at, 250 feet in that particular tree. They've been there for, centuries. So anyway, we go from nature to this. So we have, as the center of the universe, these little dark figures, which are us, and we are trying to figure out now a technical ecosystem that will allow us to live off of Earth. And so we go through a lot of trouble to create a system that is providing, I would say, a fraction, of what Earth is providing for us. And this is from the Advanced Life Support Reference Missions document, and it doesn't really matter the exactitude of this. I wanted to give you an idea of the level of detail that we need to go into to understand and create a system that is essentially an airtight system that's going to keep us alive for long periods of time, and to have regenerative life support systems where you can see all the lines going from one place to another, from the water system to the plant growth system to the thermal, the food, air, and the waste systems. That one person's feedstock is somebody else's waste. In creating these closed loop types of systems at a very small scale, so it begs the question of how do we do this at larger scales as well, especially as we're starting to do, what the current space companies want to do, which is not just have 4 people, boots on the ground. They want to have thousands or tens of thousands or more of people living on Mars or the Moon. So scaling these systems and understanding these systems is a major challenge, and we've had a lot of fantastic presentations today that support all of those types of activities which need to happen. I like this diagram because it really pulls together, for me, as an environmental scientist, before NASA, I was working in one of these sections and not really understanding that the lines coming in, the lines coming out, and those effects in a larger system. Once I tried to put my systems into a spacecraft, it made me realize that you're part of a community, you're part of a material community, an energy community that needs to work together. As we all know, we are moving from these short duration missions like ISS. When I mean short duration, I mean crew being there, usually a year at most. But we have frequent resupply of food, water, and emergency return to Earth. We don't have planetary protection requirements to really difficult extended crew durations, where we really, like on Mars, the possibility of resupply is near zil, we, understanding medical issues. There's no emergency return to Earth. It's going to take, currently in propulsion systems, 6 months. When you have the right, orbital timing going on, it'd be much longer if you try to do emergency return, and as well as strict planetary protection requirements. So I want to focus first on sort of this technical ecosystem thought of just looking at the major areas that we work in here at Ames. But this is, these are one of the three major areas for the, where you see physical chemical life support systems trying to create the processing, essentially, that here on Earth largely biological systems do. So in air revitalization, we're worried about removing the carbon dioxide. We have to compress it, we convert it back to other useful compounds like oxygen. How do we get oxygen and generate that? We have to worry about contaminants building up. In water, we have to take very dirty water and turn it into drinking water. So we have to disinfect it and control biofilms, remove organics, and do various conversions on them. Inorganics removal as well, and we want to maximize recovery, of course. Water is very heavy, it's one of the big issues. And waste management, as you can imagine, 4 people in a year, when you take out your garbage cans, imagine trying to keep a year's worth of garbage in your living room. Sorry about that. If you heard a siren, it's not for me. And waste management, you want to sanitize and microbially control what's going on inside the waste. You want to be able to volume reduce it and recover the, not only the water, the gases, the nutrients from it, and the building materials that you can make from it. So on space station, we do have regenerative systems. We do have regenerative water systems and regenerative CO2. We capture CO2 and get some of that back as oxygen when the Sabatier system is running. Nothing so much with waste. But as we move on to Moon, which is easier to still resupply, especially as ships get larger, but especially when we go to Mars, we're going to need deeper and deeper investments and capabilities with our regenerative life support, as well as, especially on Mars, in situ resource utilization, to be able to use the CO2 that's in the air, the water that's in the surface, and the regolith for building, all sorts of those things, to the greatest effect. And it's it's similar to, instead of that being a rover, imagine you have a horse and a covered wagon, and you're going west from east. And a long time ago here in America, you brought shovels and seeds, and you use what was local. So we're essentially in the same type of scenario. So looking at a little more detail in some of these areas, the atmosphere management, and you can see there that Mars actually does have an atmosphere. It is about 1%. It's about 95% CO2, a little bit of nitrogen, which is helpful. Like I mentioned, we have to worry about generating oxygen. removing the carbon dioxide, the contaminant control, and particulate control. And these are various scenarios through time. This is the Apollo 13 CO2 removal system that had trouble, and they were able to fix it. This is space station, non-regenerative lithium hydroxide canisters. And this is a trace contaminant control system to help particulates. And this is an oxygen generation system. When, here at Ames, we're working on a lot of CO2 removal systems, as well as some CO2 conversion systems, not so much to get oxygen back, but I'll talk more about that later in the biomanufacturing portion. But we're actually trying to use carbon to be able to feed biomanufacturing in new ways. There is an awful lot of research and going on. There's a lot of different technologies in this area that I don't have time to go into that we're developing to try to create very reliable systems. Many of these systems that we have currently right now do rely heavily on repair. So we do need to be able to have highly reliable systems, and that's a major thrust right now for a lot of our technology development. So although Mars, they really strongly believe was full of oceans at one time, probably a lack of a radiative protective core resulted in the loss of all of that. Maybe it went underground, maybe it was lost to the atmosphere, but it doesn't have much water. It's not super easy to get out. So we would still like to be able to recover our water, obviously. It's probably a lot cheaper than trying to get it from the surface. Again, we want to be able to disinfect it, remove organics and inorganics, and maximize the recovery. There's many different ways that we are looking at that. Here at Ames, there's a lot of membrane technologies that are being developed, forward and reverse osmosis technologies, thermal evaporative systems. These are pictures from down at Ray Wheeler's old, this is from quite a while ago, I think, Ray, but plants are also good at treating water if that water is not toxic to the plants, through evapotranspiration. So there's a number of different biological ways. There's a number of different biological, wastewater here on Earth is, the organics are treated primarily through biological systems, and there are people working on those types of systems as well. There's quite a few technologies in that realm, all working either with bacteria or algal systems, working towards that directive as well. Waste management is something that is really important. We don't want to create landfills. We want to be able to use everything we brought up and bring it back into a circulation. That was not been the case up to now. You can see here on the shuttle, this is how they would take care of waste. They put waste in a bag essentially and compress that bag, turn them into footballs, use duct tape to duct tape them up. And this is some pictures of people down actually Kennedy unraveling some of those wastes, and you can see things that people throw away. The toilet system, we get a lot of questions about toilets. I'm not going to go into the details, but essentially we collect that material, it's caught in essentially a can, and then it's just held and then brought back. So there is no real treatment, either currently or on shuttle. Space station is, very similar types of things. They have a lot more people, a lot more things, and a lot of waste end up in bags that then get brought back in return vehicles. There was this, on the shuttle, they did a trash compactor test at one point. These were manual handles that ended up breaking, so they took it out and brought it back home. Well, we have a very large effort right now that is, I'll talk about it on this slide, that is looking at a different type of compaction system. This is purely thermal processing and mechanical processing. It's a heat melt compactor that heats the waste to pull the water off, It sanitizes it, and then when it compresses it, when it's hot, and the plastic then cools and forms a hard puck. But obviously there's many other ways. This is a composting system that was run down at Kennedy. I also did a lot of composting work at Rutgers. That was my specialty. And so that's being able to take organic materials and turn that potentially into soil amendment. There's incinerators. This is also Kennedy research here. These are ways of treating inedible biomass to get nutrients back, as well as treating the organic matter in the water. Just going into a little more detail because I'm biased. This is where I did a lot of my work. You can see both the system that I worked in at Rutgers and the system at Kennedy. And there's anaerobic systems as well. So there's, the oxygen needed for composting plays a big role in all of this. If you don't need it, there's advantages, some disadvantages with anaerobic systems, of course. Looking at food production from waste, we had single cell protein and fungi. We have currently an STTR with a company called Nature's Find at this point, who have been working, and they have actually flown a fungal production system in space. They now have a fungal food company. It's growing, so that's very interesting. There's a continuously stirred tank reactor, which I showed before. Barry Finger, a long time ago, working on that. Conversion technologies. This was compost that we made and put in with fake regolith and simulant, and started growing plants in that. We've also done work here at Ames looking at microbial fuel cells. And this is some of the work at Kennedy looking at nutrient recovery. This is an extremely important facet, not only for space, but obviously here on Earth. Our current agricultural systems are just completely unsustainable the way that we handle nutrients right now. And so closed ecological systems are one way that we can start to increase that recovery. For integrated testing, Ray showed you some great pictures of the BPC. Human-rated tests were done at JSC. They tried to go a bit further. There was Bioplex and there's JSC integrity ideas. They have not been manifest yet, but obviously there's a new one called Chipia. It is at some level going after what these did intend to do. But obviously we need a lot of that kind of testing to test out the integration of systems as we move forward. So I just want to end up also with, I'm just talking about the in situ production emission products. We have obviously the ability to use in situ resources to be able to make a lot of the things that we need there. And obviously until we're self-replicating systems using local resources, we really can't be Earth independent. And so the projects that we have here at Ames look at biomanufacturing and biology. Obviously we have a lot of physical chemical processes that make most of the things that we use today. But we have biology can do a lot here, right? It can help with in situ resource utilization, in situ manufacturing, as it plays a lot of roles in closed loop life support, as you mentioned, as many people have mentioned, making medicine, obviously making food, and these systems done right can be very reliable and provide self-sustainability. The attributes are with biology as compared to chemical synthesis is that it's often very scalable, it's highly programmable, it can be very precise, and it can make, in some cases, it's the only way we know how to make certain things, like certain protein therapeutics. Very low temperature and pressure, regenerable, and it's a way to obtain or utilize into resources. We can make a number of different things here that we're interested in, and some of these are being made already. Food, vitamins, enzymes, therapeutics, polymers, fuels, primary chemicals as feedstocks to make secondary products, biocement and binders, and specialized function molecules. And we're like for carbonic anhydrase, we're making for a CO2 system we're developing, as well as for biosensors and probiotics. And we engineer the organism for this, but we have to also co-evolve the environment for that.
John,
So,
you have about 15 minutes left. 15 minutes.
Great, thank you. So scaling this and turning this into extraterrestrial biomanufacturing, creating a technical ecosystem that supports the biology is a complicated process. And we're doing a lot right now trying to figure out how to support this. There are many different feedstocks that will be available to us. We have a lot of challenges in our production on our bioreactors. We're going to be always at low gravity in comparison to Earth, at least for a long period of time. All the things that are hard to do here on Earth become much more difficult to do in space. Finding how to simplify that is important. Same with the harvesting system, and the same with the purification and preparation. Here on Earth, we have many resources at our disposal to do those things. In space, how do we use biology in the smartest way possible to give us the products we need with the least amount of processing, right? So if we're able to secrete products from microbes, say, and separate them very easily, we can bypass a lot of this. And then if we learn how to do that for there, that would make Earth processing very important. We're also going to need to worry about the environment, right? We mentioned that this is not Earth. We have a different environment. Even if we keep it in a pressurized system, radiation is going to be a real issue. It's a real issue for human health. It's a really strong issue for human health on these long missions. At Ames, I just wanted to mention that as part of this, they've developed, and it's actually it's on Artemis. Right now, it's the next launch that is going up has this on it, and it will put biology the furthest it's ever been away from Earth. And this is called Biosentinel. This is done at Ames Research Center. It is using yeast that have been engineered to serve as a sensor for radiation. So we can understand the effect of radiation on life using life as the metric rather than just a physical chemical radiation sensor. Another project that we know is part of my lab and we've been doing for a long time is bionutrients. Bionutrients is we've engineered yeast, baker's yeast, to make 2 different at this point 4 different nutrients on demand. And you can see here this little pack right here that we have essentially an edible media and our edible yeast, and the crew have been hydrating it. We have a 5-year experiment because we need a 5-year shelf life on food for Mars missions. And so we're trying to see if we can generate the nutrients that do degrade in the supplements and the stored foods. So that's going on right now. We're over 3 years into that project and going well. And you can see this dark orange. We're making carotenoids. So that's why carrots are that color. And that's why your mother tells you to eat your carrots, because you really need those nutrients. Also, we are instead of just bringing up, if you want to scale that process, and as referring back to the previous slide, we don't want to just bring the media up with us all the time. We want to be able to make media there. So if we're not using photosynthetic organisms, we're using heterotrophs like yeast. We want to be able to have a media available for us. So we convert, instead of a leaf converting it, we're using physical chemical systems to convert carbon dioxide and hydrogen to different compounds to make the organic substrates needed for heterotrophs to then make products and make things like bioplastics. So this idea is in contrast to how we do things here on Earth, which is we have carbon dioxide and water. Over months or years, it's made biomass, or millions of years it made oil and coal and methane, and then we take those organic products, go through chemical syntheses, and make our final products. Here, we're taking the carbon dioxide with the water using solar power, rapidly converting that carbon dioxide and water to building block molecules that are carbonaceous and the microbes can use to make our final products. So we're trying to speed this process as much as we can and make it as energy efficient along these steps as possible, trying to essentially beat photosynthesis. So in this process, what we've been doing is creating essentially a to Z process where we're looking at the CO2 conversion strategies. And there's many different types here, and carbon feedstock classes of different types of feedstocks that we can make, and then look at the specifics of what they are, and look at various host organisms to find out which of these they can natively use well and efficiently to make different products. We've been looking at electrochemical conversion systems to make acetate and formic acid. We've been working with a group at Stanford to be able to do that. We also had the NASA CO2 conversion challenge. We started that to be able to convert CO2 to sugars. You know, obviously we would love glucose. Many organisms use glucose and very efficient, very energy dense. And we did have 3 different winners there who were able to, without using biology, create sugars from carbon dioxide. So obviously we want to, keep working that type of those conversions to make that process more and more efficient. We have microbial food production and many different possibilities there. Dr. Cuello went through a number of these different possibilities, so I won't linger on this slide. We, as part of our bionutrients process, we are actually starting to make yogurt and kefir as well. So we have a Bionutrients 2 flight launching on SpaceX 26, so November 15th. We're also looking at making bread, all with these engineered organisms, to be able to make essentially nutrients and/or we also are making a pharmaceutical follistatin in these as well. So obviously looking at different probiotics, very important for human health, fungal foods. I mentioned some before, and also being able to make maybe non-human feed if we want to do other aspects. I'd like to sort of end by indicating if you're not familiar with cubes, this is it's what's called the Space Technology Research Institute. It's very similar to the NSQUARTS. This is the new version of the NSQUARTS. And CUBES is the Center for Utilization of Biological Engineering in Space. It's led by Berkeley, and it's has 5 years already. They just got a renewal for 3 more years. And they've been doing a fantastic job looking at biomanufacturing on Mars and different types of CO2 conversion systems to make acetate and other compounds, how to fix nitrogen. Ray mentioned the production of pharmaceuticals in plants, but also in cyanobacteria, microbiome engineering for plant growth, great advancements within the plant growth systems themselves and the lighting, as well as really great systems analysis. So if you haven't seen it, I recommend you go to CUBES and look at all the different areas that they've produced and many papers. They have at least 60 papers from that effort and done a lot in that, and we're really happy to have been able to facilitate that, and really grateful for them for all the great work. And I'd like to end on this slide again, just to remind ourselves that when we are developing these systems, we are definitely in a community, and we need to work with each other and think about ourselves as an optimization of an ecosystem, working with each other. With that, I'd like to thank you very much for this opportunity, and I'm happy to answer any questions.
Great presentation, John. Thank you so much. My first question actually related to your last comment about working together as we move into towards a commercial space industry, and also as we move towards commercialization of, I think, a lot of terrestrial technologies for sustainable life support, like vertical farming, et cetera. How do you see, or what do you see as the future for businesses and academia and government to be working together to realize these advances?
That's an excellent question, and one that I've been thinking about, and others within NASA have been thinking about for quite a bit. Commercialization has been brewing for a number of years, and I would say maybe in the past 5 years, it's become very clear that the future of space is going to be commercial companies. Rather than just the space agencies creating rockets and controlling all of that. And that's essentially what the government is supposed to do. We are there to help technologies develop, and then license technologies, make them available for companies to grow, and then play whatever role is necessary in that evolution. For example, the space station obviously it's been up for a long time. I'm not going to even predict, when it will, eventually be decommissioned. But there are 4 different space stations planned currently, and there's one that's going to be built using ISS as a system. There's 3 others. A lot of those have very strong science and, to be honest, biomanufacturing. Business plans, to be able to, start to facilitate off-world production, off-world science, to facilitate all of this. Obviously, the CLIPS landers for the moon and what would happen on Mars, very much evolving. So, within that realm, NASA's role, obviously, is to keep trying to understand and be, at the very least, a smart customer, but we're also developing, requirements. We're developing a lot of the medical issues that need to be understood and working on those. We're working on a lot of different technologies that the companies themselves may not have the financial resources to really solve. So I think NASA will, and other space agencies will continue to do that kind of work to facilitate that. And academia is right in there, right? I mean, we have only so much understanding. There's tremendous wealth of understanding in academia that we rely on heavily to help us out in this large community. So as it becomes more commercial, how does the money flow and where does it flow from? It's going to be a very interesting question, which I'm not a money person, so I'm not going to predict that. But I am very interested to see how that unfolds. So it was an excellent question, and your guess might be as good as mine.
Let's see, I have some more questions that I think we can talk about in the next segment, but my last one I think would be about waste processing, which we haven't talked much about today. And I know you have some experience with, excuse me, with both gray water and solid waste as well. What were some of the biggest lessons learned or surprising lessons learned for you with regard to waste processing? And can decomposition rates keep up with plant production, for example, for composting, bioreactors, incineration, all those different
Right.
methods? Any surprising lessons learned?
Yeah, it's a complex question.
Yeah.
So from a long time ago now, the composting, we grew plants, space type plants, took the inedible biomass, composted it with pretty much itself as a sole, feedstock, and turned it into a compost and tried to grow different seeds and plants on it. Those plants are, very heavy in nitrogen, and so when you compost it, a lot of ammonia gets released, and the pH goes up, and so we have some issues with pH issues So there would need to be, and composting gets complicated, and making something that you would every time be sure that your crop is going to work in this. And so I'll just say that was one thing that here on Earth you have so much to mix with, and so much a buffering capacity in soil when you add compost to it that the regolith doesn't have. That you can get away with things. It's worked out perfectly. I mean, that's the way the system was designed here, right? Those systems would require to become much larger. I did a lot of work on this, in terms of how much organic material you'd be able to have, what the input rates would be in order for that to work. And so that does become a matter of, in my mind, of size. It'll be a critical mass of doing this before I thought it would be a sustainable system. I think there's going to be a lot of waste is an incredibly complex thing. So I'll just leave it at that, because I know we're low on time. But
that's a good glimpse.
accumulating things that you don't want to in places you don't expect to are typically some of the large surprises.
Like volatiles as well.
Metals. Yeah.
Okay, I'm gonna go ahead and move us on into our next segment, and yeah, it's time. And then we can continue to, if the panelists are still online, we can continue the conversation, have some more questions for our speakers. In the meantime, John, if you could stop sharing for a moment, I'm going to switch over. To our activity for this afternoon, if I can find it. One moment. Okay, so today I think we've talked a lot about where we have been and where we probably still need to go. And I think all of us probably agree, many researchers agree on the need and feasibility of bioregenerative life support. And we have a fairly good idea of what the next steps are for development. We've written a lot about that on our mural today already. But the need also, one thing I think I would like to highlight a little more is technology maturation itself, which is very difficult. So we develop a new technology, but when you develop something for space, it takes a lot more time and a lot more money to test it out and prove that it's reliable in its relevant environment, such that often terrestrial technologies quickly outpace spaceborne technologies. So finding funding and platforms where you can do that maturation and testing and proof of concept for your tech is kind of difficult. So we need to really be able to prove out our readiness of these systems, not only on Earth, but then in space as well, to make sure that they're going to be there when we need them to. Excuse me. So one interesting development right now that's happening is a lot of these space platforms and commercial platforms in development, like the Lunar Gateway and commercial LEO space stations like Orbital Reef, or let's see, excuse me, some of the other commercial free flyers that are in development by various companies. These could be critical proving grounds for us and for near-term missions, where we can use these platforms to realize, to test things out, use them as proving grounds to realize our larger scale space farming systems for deep space, excuse me, not just space farming, but ecosystems for deep space exploration. So that's going to be an important step for the future, is how we work with these companies to be able to get some test platforms in an actual relevant environment. So with that in mind, today, for the next, what time is it? 2:05, 25 minutes. Am I doing my math right there, Patrick and Sherry? Yes, for the next 25 minutes, We'd like everyone, if possible, to go back to Mural. We'll put that link again in the chat in case you've lost it. And help us identify what you think some of the obstacles and solutions to closing these knowledge and technology gaps are. So I just mentioned one obstacle is technology maturation opportunities, and solutions maybe working with these companies that are building commercial space platforms as proving grounds. So that's one example. What do you think we need to do now? How can we all work together to realize operational space life support ecosystems? And here's some, on this slide here, just some categories that we've put up on the mural to consider. Are there ways that we can better communicate and collaborate between our ecology and bioastronautics professionals in government, industry, and academia? Are there ways that we can better share knowledge, and how do we get knowledge out on current R&D to those who need it? How do we train and inspire the next generation? Hopefully conferences like this might be a way to do that. And then of course, how do we fund all of this? What are some maybe agency advocates or business models for funding the R&D that needs to be done? And my biggest question for you all today is, given what you've heard and the conversations that we've had today, would you be interested in, or do you think that we should form a space ecology association or coalition, something to that effect? And if so, what do you think it should do? What do you think its vision would be? And we'll be also sending out a post-workshop survey on that question as well. So Patrick, I don't know if you were able to send, yep, there's the link in the chat to Mural. If everyone wants to go back to the Mural, you'll see on the outline on the right side of the screen, there's sections for these kind of future looking questions and what actions can we take on our R&D roadmap for space ecology. And to our organizers, did I miss anything? Is there any other comments?
No, I think that's everything. I mean, I guess maybe I do want people to participate on Mural, but I also acknowledge that some people are up quite late, and I would feel bad to keep them up too much later. So are there any like particular closing notes? I don't want people to like miss anything.
I think, yeah, if you need to head out before closing remarks, the biggest thing I would say is look out for the proceedings and the audio link. We'll be sending that. And also those bios in the who's who of space ecology, if you want to be included in that, that'll go out with the proceedings. So you could still send us your bio, tell us who you are and what you're working on. I think that's it. And it looks like it would be a good idea for me to give another little brief tutorial on murals, so I can do that. So those of you who weren't here in the first section of the day, you've got the mural link there. It's pretty fun and easy to use, actually. The first thing I would recommend is on the right side of your screen there. You'll see, I believe you'll see an outline, or at least an icon of an outline that can help you navigate to these different sections on the mural. And other ways to navigate around and move around the screen is basically you can just click and hold your mouse button, click and drag your screen around to navigate. And you can also use your mouse to zoom in and out, just scroll in and out. So there's many different ways to move yourself around the screen. So you can go ahead and play with that and get accustomed to it. To add an idea or a comment, the easiest thing is double click. Just double click on your screen and it'll add a sticky note. And also on the left here is an icon of a sticky note. You have other options there for adding text. So if you want to just add a little text box, you can use that there. The biggest advice for those of you who didn't use Mural earlier is just be really respectful and careful not to remove other people's content. So if you accidentally do that, there is an undo button. It's not shown on, oh, here we go. It's right here on my picture. For some reason, it's grayed out. You will have an undo button right here that you can use if you accidentally delete someone else's content. And yeah, so this is a way for us all to silently, since we have so many people on the line, be able to have a conversation in writing and put our thoughts down, and other people can then respond to your comments that you put down on the board, so that we can kind of interactively brainstorm silently and organically. So one more recommendation for you. There's a comment feature. You can use that, but we don't recommend it. It's just a little confusing. So probably just putting on stickies is the easiest thing to do. Hikaru, I think that hopefully answers your questions, but go ahead and chat any other questions you might have. Or also you can ask Patrick. Okay, great. Thanks. Okay, so who do we still have on the line? We've got, it looks like John. Joel, are you still there?
Yes, I am.
Okay, Cyrille, are you there?
I'm still here too.
Oh, hi, Bill. Good to have you. Looks like Barbara's still on the line as well. You're up late. Thank you. And, oh, Ray, you're still there as well, huh?
Yes, I'm here.
Okay. So I have a couple of questions. First for John that I didn't have a chance to get to. But before I start, Patrick, did you have any comments? We're good?
Yeah, I think we're good.
Okay. John, on your slides, back to the, oh, I forgot the name of the slide, but it was about the things that nature does that makes this all work so well. Let's see, let me stop sharing for a moment. Well, I think you'll remember. We all might remember. There was a bullet that said nature optimizes rather than maximizes.
Mm-hmm.
And another one had to do with efficiency. So one question we had was, what do we mean by nature optimizes rather than maximizes? So I was wondering what your thoughts were.
Yeah. So nature is never, I can put this in a context of right now in our biomanufacturing activities, we're engineering organisms that make a particular product for us, right? They're making beta carotene, zeaxanthin, pharmaceuticals. We are, trying to maximize in a way the amount of product that we can get out of, that organism. We are, in a way, you start to stress it and make more than it needs. It's as part of a metabolism, it will only make as many molecules of that as it needs, because if it makes as many as it possibly can, but it doesn't need it, then it's wasting a lot of energy and it's reducing its fitness, ecologically speaking. So, I mean, from a very simplistic point of view, that is, it's always, that metabolism, when you look at metabolic charts, it's very frugal, right? It's saying, these molecules that I'm converting to this, I'm going to do as little as possible there, because energy is so hard to get. So that happens at the cellular level, and that translates, I think, into the macro level as well. And that also sort of fits into the form, fits form to function, right? It doesn't make things that aren't required because of all the energy and the frugality required for fitness in an ecological system. So that's it. it's all wrapped up into one very interesting, process bundle at that level.
Let's see. I had a question that I think maybe was somewhat answered, but there was on the list of bioproducts, you included biosensors. And I was wondering if you could give an example of a biosensor.
Sure. Well, so the BioSentinel mission that I mentioned is a yeast that is it's able to be used as a sensor, right? It changes its characteristics in a way that you can visually understand how much radiation damage it's undergone. So from that particular point of view, that's a very specific biosensor that we could be making as we need, right? Very much in the bionutrients process that I mentioned before. You could be growing these organisms and having them constantly there and get an understanding of what is the real, life-altering dosage of radiation that we're getting here, and what's its effect. And so there's, what's required there, of course, is being able to correlate the damage in yeast to the damage in a human system. And that's part of that research and understanding, that correlation. Other biosensors are being developed into plants. For instance, the DoD is looking at plants that are able to detect various chemicals and for security purposes. But I think that there's quite a few different possibilities in this realm that I think people are just starting really to explore.
It's interesting. The next question I had was, or that we had from the group, was related to the biomanufacturing chains. What do you think is the most promising biomanufacturing chain, or the most critical to get started?
I'm sorry, biomanufacturing chain?
Or products, biomanufacturing product, I guess that's probably a better word, yeah.
So I'll give you the rationale for why I picked what I picked. The food system, right? I mean, so the food system right now is not ready for Mars. So if you talk to the representatives from the Human Research Program and the food systems, they will indicate that a 5-year shelf life is required for a 3 years Mars mission, and that's partly due to prepositioning and just delays that tend to happen. So they're working hard right now to understand and store foods for many years. They have done multiple year tests already, but more are on the way, and at at different temperatures, different storage conditions, to understand how nutrition is changed within that system. So both in supplements, vitamin pills, and in the foods themselves, certain vitamins tend to degrade significantly with time. And so they really run the risk of nutritional problems. It's all going to be very, very difficult for the crew as it is. The lack of gravity, the countermeasures that they're going to have to be doing, the long periods of isolation is already going to be very difficult on the immune system. So they want to make sure that the food system is something that they really enjoy eating. It's nutritionally dense and it provides everything. So they're working various ways to try to figure out how to work that system. Obviously, growing plants will be very helpful, and is a great way to produce a lot of different vitamins and nutrients and protein, all the things that you actually need for food. That is, those systems to do that at scale get big, and so we wanted to do something that was smaller, right? And so for a microbial biomanufacturing system, if we're able to take very small amounts of media with us, very small inoculum, and have small like food bags that essentially that you will hydrate and let it grow and let it produce those vitamins or the nutrients that do degrade with time, then it's a potential way of solving that problem. So that was our low-hanging fruit because of the minimal volume required and the magnitude of the problem. So the risk, the difficulties, that is a knowledge and a technology gap right now of how to keep food fresh and nutritious enough for everybody for 5 years. So that's what picked that. Now, on the and that's the same for pharmaceuticals. We're interested in pharmaceuticals because they have shelf lives as well. Some are a year or two, and some are less, and some are more. But being able to make pharmaceuticals on demand, really important as well. So that can be plants, or that could be microbe based system as well. Moving into larger scale systems like making bioplastics at scale for bulk systems such as binders for regolith to make landing pads or roads or construction walls, things like that, require quite large systems that will require very large bioprocessing. There are people at Cubes and other people looking at how to do that. We actually also have a scaled system that NASA is funding. It's called Mango Materials, the name of the company. And they are using methane from an anaerobic digester to make scaled bioplastic production. So we're trying to learn scaled systems from that company. But so in our lab, it made sense to go after small amounts of precious chemicals, and that's why we went there. But obviously, food in all the realms, including protein, fresh tasting, good protein. We're making yogurt as well and kefir, and we're starting to look at making bread and nutritionally optimized as well as a medium to grow things. So I hope that was a long-winded answer, but I hope that sort of answered your question.
That was a good answer. Thank you very much. There's another question related to biomanufacturing is, and I think I have my own answer, and I'm curious what yours is. Are there biomanufacturing processes that benefit from microgravity or even from vacuum? I guess is another interesting aspect.
I can tell you that our current results right now from the bionutrients experiment that our microbes go better in microgravity than they do in 1G, our ground controls. And we think mainly that's because we don't stir them, right? We don't have an agitation system right now. So, here on the ground, the liquid is static. In space, CO2 bubbles are being formed and actually move the liquid around. And you may have seen earlier, how the orange liquid, held to the side, that's filled with bubbles, CO2 bubbles inside. And so I think there's actually better mixing that happens in space. And I think that that's quite possibly a strong advantage, to be able to do that. I think keeping gases in contact and not, the bubbling aspect, it has that possibility. Using vacuum, I'm not quite sure. I haven't really thought about that. I don't have a good answer for that. But
I hadn't either. Nothing comes to mind.
So you had an answer as well? I'm interested.
I think you pretty much answered it. Is that, yes, there is indications that there are some benefits to microgravity on not only bacterial growth, but on protein crystallization is another area. So that there's this idea that maybe you could get more pure protein growth as well. So those are two that come to mind.
Another really important one, and I mentioned this before, is this biomanufacturing industry might be growing on these new space stations. Is largely around either growing tissue and not needing the scaffolding required to grow organs or tissue here on Earth. It could be amazingly important. And so, there is the idea of an industry of taking somebody's cells and now growing them a liver, or growing them a heart, or growing them a kidney in space, made directly from themselves, and now going in return to them for transplant or tissue, right? So I think that's an incredibly potentially important area that a lot of other people think so too. There's a recently, was it a $20 million gift was given to one of the UC systems for biomanufacturing in space that was specifically on tissue generation.
So with the last few minutes we have before we have some closing remarks, I'd like to ask a couple of questions of our speakers as a whole, and also invite our organizers.
Chris, could I have a question for John?
Oh, hi, Joel.
Hi. Can I have a quick question for John?
Yes, please.
Yeah, John, I really like that a project that you mentioned about producing feedstocks that are organic, derived from CO2, using physical chemical means, not biological means, producing sugars and alcohols, among other things. Can you give us an idea in terms of the energy requirement for that process, as well as to what scale that's been demonstrated, and the efficiency as well?
Yeah, so it really depends. It's a nascent field, and there are quite a few different companies starting to arise and make different products in different ways, from different feedstocks. The research that we funded was Matt Cannon at Stanford University, and he used electrochemical systems with gas diffusion electrodes to convert carbon monoxide, which is easy to make from carbon dioxide, to acetate, and got, I think, about Faraday efficiencies of close to 70% and similar first pass efficiencies in that conversion. Again, you're making a product, and then there is going to be essentially an energy chain, right, all the way from what it took to make the system, pretty electrochemical system, to run it, as well as then how much energy are you now giving the organisms from that product, right? There is one of the winning companies in the CO2 conversion challenge is a company called the Air Company, and their main product is not sugar, but is ethanol, and they make ethanol from carbon dioxide, and have been selling it as a alcoholic drink product, vodka. But ethanol is also a very viable substrate, for yeast and other organisms as well. So I don't know the energy efficiencies from those systems. I mentioned Pidong Yang's research at the university at UC Berkeley. I would recommend just looking at some of his work, and getting 3-4% efficiency in the CO2 conversion when you try to do the math end to end is, if a plant's 1%, it's really hard to compare these numbers, Joel, as you know, I'm sure, because of the vast systems that surround them in the envelope that you draw. So I think a lot of work needs to be done to fully understand all of that, but there is quite an interest now. I think that CO2 conversion challenge actually sparked a lot of industrial interest in terms of being able to produce a number of feedstocks that they normally get from biomass. And biomass is going to become a diminishing commodity. So to be able to make it straight from carbon dioxide is something that people are very, very interested in.
I think the implication of this new innovation, really, for space application, for bioregenerative space application, would be quite significant. So I'm excited about the project. Thanks.
Sure. Yeah, thanks for the question, Joel, and all your research. I really appreciate it.
Thank you.
Okay, I'd like to have maybe one more question for the panel of speakers as well as our facilitators. Actually, well, two, if I may. We'll see how this goes. Before doing some closing remarks for the day, looks like there's still some mural activity going on. The first one is, for me, it's a very loaded question. Engineers are notoriously cautious about the use of biological components because, in a few words, organisms die. So there tends to be a mindset that biological systems are less reliable than mechanical components. So what are everyone's thoughts on biological versus physical chemical reliability and maintenance requirements? And how do we put this issue to rest once and for all? I'll let anyone take that.
Well, just hearing the thought, when somebody's car stops working or food processor or whatever, we tend to say it died. And it seems to me that mechanical things in our everyday lives die much quicker than people. And so maybe the idea that the biology is not reliable because it dies is backwards.
Rita. Breweries, the liquor industry.
What do you?
They use biological processes. They make a big profit. Everybody uses their product. I think we can overcome the idea that biological systems are terribly unreliable by pointing to industries that use them reliably.
That is a wonderful point.
Ray, get them drunk.
and then have the discussion. Ray has his hand up. Go ahead, Ray.
Well, I saw Joya just typed a comment in, and I was going to say the same thing. Now, we weren't doing, I want to turn on my camera. We weren't doing sustained operations in our testing, but when we experienced failures, invariably it was a mechanical component, say in that chamber. A pump failed, the power went out, a blower failed, a sensor, and so forth. The biology was really quite predictable and sturdy. But it can fail. I mean, there are biological failures and there are environmental limits. One thing that the Russians touted for many years is that the biology is intrinsically self-regenerating if you have some propagules. If you plants, if you save some seeds, somewhere else, or you have some tissue cultures, if you have a failure in your system, you can grow a whole new one. If you have a Sabatier reactor that cracks or blows up or something like that, then you're now having to deal with welding metals and whatever and milling things and 3D printing stuff, and maybe it can be done. But yeah, I think it is something we sort of need to educate or disabuse the engineering community of that kind of notion. So I see Joel has his hand up.
Yeah, just quickly, I think that we're going to talk really about a robust bioregenerative ecosystem or life support system. We can't really talk about that without any biology involved in it. And I would argue that really some of the most resilient components of that ecosystem are biological. For instance, microbes. There are many microbes that are extremely resilient. So we need to harness that and make that part of the design of the entire working bioregenerative ecosystem. So really for me, to have a complete one and robust one and resilient one, it has to have a biological component. Without it, it's not complete.
Anyone else?
Yeah, I mean, I'd just like to sort of reiterate and say, I think probably the most reliable thing in a spacecraft is the human. You know, lifespan up to 80 years with, you know, you think about how little maintenance in a way other than food is required. And I agree completely with Ray. If, you know, if you are able to create with the physical chemical system, if you can create a very consistent supportive environment, biology is incredibly robust and adaptive in ways that mechanical systems are not. And I don't see how it could be seen otherwise once you understand those very, you know, fundamental basics.
Right. Looks like you have your
another
hand up.
sort of, yeah, I do. Another ancillary observation, relevant, I think, is that over the years, when I've say, for example, worked with chemical engineers, and they've wanted to model what's going on, say in a biological system, and you get to something like a bioreactor, and their tendency is to want to know equations and processes and every possible thing that's going on, and you tell them, well, we don't know what exactly is going on, but we know what the function is. And we know what the resilience limits are, and so that becomes sometimes a difficult thing for engineering communities to accept, because they want explanations for every process, every metabolic equation, and we know a lot, but in a bioreactor, I could be wrong, but I don't think we know exactly what's going on for every reaction in a bioreactor, and but we do understand its function, and we know how to manage that. So there's sort of a discomfort, I think, with the engineering community, with biology in that regard.
Frida.
We do have to acknowledge diseases, and I think that's one of the things that the engineers hold against the biologists, is that there's always the risk of a disease. The Russians had a lot of problems with one process accidentally inhibiting another process. They burned some of their straw one time as a way of getting rid of cellulose that was resistant to breaking down, and it killed a lot of the algal cultures. So, I mean, the brewery industry has pretty much designed their systems to have pretty high reliability, but they also have failures. And I think, you know, we can't ignore disease. And I think that's what the engineers hold against the biologists.
But that's also something that we can't hold back even in a mechanical system, because the microbial environment is there. You're not going to remove it. The biome is there. Emily Matula on the chat pointed out that we spend a lot of energy thinking about how to stop biology in a mechanical system because they're less adaptable. And Danielle actually put a nice, I think, final comment on the fact that it shouldn't be a bio versus physical chemical debate. Like a couple of you have said, we can't remove biology from the conversation. So we definitely should be exploiting both worlds. So some great comments there. Anyone else? Okay, one final question for the panel that I think maybe some on the line would really appreciate, especially some of our students, is whether you guys have any advice for a beginner in bioregenerative life support field in terms of, well, this question was what systems should they focus on first? But I would ask that as a broader question about where to start for curriculum or career direction or any of those things. The beginner in the field, where do you start?
I'd say start with your passion, because this is a multidisciplinary endeavor, and you can start at any entry points, whether it's biology, ecology, chemical engineering, biosystems engineering, industrial engineering. There's a way, there's a point of entry for you. That's your passion. But of course, it's good to have an appreciation as well of the comprehensive vision or view of what bioregenerative space support is trying to accomplish. Yes.
That's a great answer. Does anyone have anything else to add?
Well, I think agencies like NASA need to reinvest in the external community, the academic community, the business community, to create more entrance points for young and aspiring professionals.
Being recorded.
So,
recorded.
But getting NASA to cough up more money is a challenge, as we know. So anyway.
Frida.
So most professional organizations send out a request for sessions, for special sessions, sometimes a year before the meeting. And you submit to a committee, and you have to have, so for some meetings, it's a 2-hour session. Some meetings are morning or afternoon. You've got to have at least 3 to 5 committed speakers who will submit abstracts that they will definitely be willing to present. But certainly I have not seen very much acknowledgement among ecologists, limnologists, oceanographers, that their work is relevant, except when you get into extremophiles. So there's a lot of interest in studying extremophiles and how this may tell how life occurs on other environments. But that would be one possibility to let people know what's going on. And also it helps if there's a source of money that they can apply for.
Any other questions or comments from the speakers or organizers as we continue the discussion for these last few minutes?
Chris, I want to answer, question number 3 on the slide. Should we form a Space Ecology Association or coalition? My answer is absolutely yes. I think this is going to benefit quite a lot of young people and even professionals, really. And I really would like to reiterate what Ray just mentioned, that perhaps with this association, perhaps the association can lobby NASA to provide more opportunities for different entry points for college students and high school kids to really get into the design of a bioregenerative space life support for space applications. I think that's in education-wise and in terms of developing the future professionals and manpower for NASA, as well as the associated industry working together with NASA to realize this vision, I think that would go a long way.
Any other comments along the lines of, in response to Joel, or along the lines of an association and what we might do to continue from here?
There is a journal, they dropped the word gravitational, but they kept the word biological. So there is a journal that publishes space-related biological papers. I don't know whether forming an organization that might be linked to that publication would be an approach.
Yeah. And Ray, if I may have a follow-up question to you, what do you think is the best way, a strategy for, let's say, an association or group like Space Ecology to work with NASA to, persuade NASA and convince NASA to provide that funding that's needed to realize that provision of more diverse entry points for students.
I published them.
Well, a lot of it kind of has to be worked internally, where people like John and myself and Joya and others and other folks kind of push on our internal administration and program people to get into the budget planning, and you know, I've dropped the ball on some of that. I think there are NASA's life sciences program just went through what's called a decadal review. A lot of programs in NASA do this every 10 years. They convene a panel with the National Academy, and they solicit inputs, and then they distill them and they give them back to NASA, and they say, okay, here's our recommendations. And I've seen some of that, and I think there's some encouraging signs in some of those inputs we'll see. But one of them was to reinvigorate the ecological community in terms of space, engaging them in space. Mm-hmm. I hope those will survive and get through to the program. Right now, we're bioregenerative life support and the associated ecological research that goes with that is sort of orphaned in NASA. And it has largely to do with some programmatic shifts that occurred 10 years ago. And so that's kind of an internal thing that, and you can certainly contact people you know and say, look, you know, this is going on, and we're gonna pay the price if we don't get with it for future missions. John, any thoughts?
Yeah.
Yes, John, please.
Yeah, so I agree, Ray. My first thought was the decadals, and that's everybody's opportunity to jump in. Unfortunately, if you didn't participate in the latest one, and it'll be quite a while before they take inputs again. But they do have different types of decadals along the way. And I agree with Ray that I think that the inputs to the decadals are pushing towards biology. And the reason that this is so important is that when that NASA is trying to spend money on various areas, that we need to go back and say, does it match these roadmaps? It really needs to map one-to-one to these different areas. Now, it doesn't need to be exact, of course, but if they're not in those documents, it gets harder to do that. I will also say that I've been seeing in the current administration a lot of interest in climate change and the kinds of regenerative technologies in agriculture that would be very much in line with what needs to happen with space as well. So I think that there's ways that you could probably piggyback and look at a lot of those technologies and turning our agricultural systems into closed loop systems, are incredibly important. Same with our water systems, right? Being able to take our sewage and instead of putting it out to the ocean, finding ways to turn it back into useful water that we use. All of the in waste, obviously. These are all technologies which overlap dramatically with space, and so it's a way for you to be able to, leverage that money as well, and serve both objectives.
Right.
I had a similar thought that our viability and living on Earth is very tied to the topics that we're talking about. And there's a lot of people that are talking about climate change and species extinction and everything to do with why our environment is failing, and it's we're very interrelated.
Mm-hmm. Yeah. Right. Chris, just one quick comment. I completely agree, because these challenges that are being worked out right now in terms of the solutions and innovations for bioregenerative space life support, they are also directly applicable to the challenges that we're facing here on Earth. Because right now, with a changing climate, things have been so much disrupted. There's a lot of growers now, particularly in developing countries, they're at a loss because the seasonal patterns, the weathers have changed so dramatically. They're so disoriented in terms of their production system. And so this alternative closed environment, bioregenerative space life support systems that support that principle of the circular bioeconomy, they're needed not only for space, but for Earth. It's so relevant to what we need here on Earth today.
Because of the scale of planet Earth, hardly anybody realizes that it's a closed system.
Mm-hmm, yes. But now we're beginning to appreciate that more, that even though the Earth is so large in size, because our population has grown tremendously, now we're feeling the consequences of all of these disruptions that partly are caused by humans as well. And so the interconnections are becoming more manifest to most people, to a lot of people. And so in a way that's good, because then we can start designing innovations that are interlinked and that are more sustainable.
Was there anything that you wanted to add, Ray?
Yeah, well, exactly along this line, I think that unfortunately Christophe Lasseur has had to leave, but I think he's done a remarkable job with Melissa in keeping it going. That's no small feat to keep a program going in a space agency for 25 years. And what I think one of the strengths of the program has been exactly what we just talked about, is appealing to current issues that are pressing in society right now: circular economy, spinning off, investing in space, yes, but spinning off concepts, ideas, principles that help us on Earth. And so I think that would benefit all of us to keep thinking about that and let people know about it.
So. Yeah. And if there's the Space Ecology Association established, that would be the central focus of that association.
Yeah, definitely. Seems, judging by the mural anyway, sounds like we've got a lot of support for that idea. So that'll have to be something we follow up on.
Yes, absolutely.
John, did you have something to add?
Yeah, so it's a very old concept at this point, relatively speaking, is, spaceship Earth. What became clear to me when I started working with NASA is that, you're always going to be on a spaceship of some sort, wherever you are, in some habitat or spaceship, right? And so when you think about space ecology, Earth is space ecology.
Yes.
Right? I mean, we are an ecological system in space.
Right.
It just happens to have a large gravitational body and a lot of resources and very well-developed systems, when we entered the scene. And so I think it's important for us to understand the development of life support, universal life support.
Right.
That sort of follows those biomimicry principles of, we're local, right? We understand how to work with the systems that we have here to keep those systems healthy, as well as ourselves. And the way I like to put this is that humanity has, I think, been looking at Earth, maybe not even consciously, but as a life support system that's supporting us, But I think the large change that's going to be happening from this point forward is us learning how to become a life support system itself on planet Earth. You know, how do we humans, instead of living off of life support systems, how do we become a life support system for Earth? And I think when we look outward that way, we'll start to think differently. And I'll also say, the reason I ended on that slide with all the interconnections is because, just like nature, optimizes, we need to do that as well. And that very much is a systems thinking kind of perspective. And we can't just work on our own little siloed areas and hope that it works out for other people. We really do need to work together on that. And I think that, getting that message across and finding methods and opportunities for that to happen is very important. And it's all just part of life support wherever we are.
Mm. I like that.
Chris, you are muted if you were trying to say something.
Sorry about that. I think that's a great note to end on, John.
Absolutely.
Unless there's any other comments. Okay, so just some closing remarks before we leave you all today. Once again, thank you so much, everyone that participated and got out of bed very, very early or stayed up very, very late in order to be a part of this. We're so glad to have you with us. A reminder that if you'd like to be in the proceedings, in the who's who of space ecology, just go ahead and send us that bio on that PowerPoint template. And if you can't find it, just message Patrick for where to find it. And where do we go from here? Well, I think that from what I saw on the mural, there's a lot of interest in doing this more and in more detail and on a larger scale. So obviously, really that was one thing I was hoping to hear from everyone today, is that this was a good experience and something that we want to continue. So this workshop hopefully will be the first of many. If we were to do it again next year, perhaps we can do more in-depth breakout sessions for more detailed discussions of these various topics. Maybe on a larger scale. So it sounds like really the recommendation and consensus from the group is, yes, we want to do more of this and bigger and in more detail to involve more presentations and more knowledge sharing. So that's great to hear. And we look forward to hopefully organizing this again in the future. I think that is all I had for closing remarks from the group, but I'd like to open it up to the organizers for any final words from our organizers, excuse me.
I don't have anything to add. Everything has already been said. Really grateful that everyone could show up, and especially grateful to our speakers for offering their valuable time and expertise.
And it was amazing seeing everybody today, and especially hearing about the very diverse approaches to space ecology and to bioregenerative life support that have been done. And I'm looking forward to doing it again.
Yes.
And a thank you again to all of our speakers for giving us such a great, broad overview of the many aspects of this space ecology field.
And big thank you to you, Chris and Patrick, and all the members of the organizing team. Thank you.
Yes, absolutely. That was a big effort and valuable.
Well, I think we will be in touch with just about everyone in these post-workshop proceedings, and hopefully we'll have more things to talk about next time.
Yes.
I'm going to end the webinar now, so thanks again, everyone.
Thank you all.
Enjoy the rest of your weekend.
Bye, everybody.
Hey.
Thanks. Bye.
Bye-bye.
Thank you
Until
very
next
much.
year.